hygiene practices. As during the corona pandemic period, people were being taught basically about the health and hygiene issues, how to sanitize their hands, how to wash their hands. Even our leaders were promoting how to maintain health and hygiene, just because prevention is better than cure. So environmental health, the other aspect which this webinar is covering, that is environmental health. It includes hygiene, sanitation, and other aspects also, such as global warming, climate change, radiation, technology, flooding, and natural disasters. It also involves studying the environmental factors that affect health. So we see that health, hygiene, and environmental issues affect everybody's life in our society. So it is important for India also, and it is an aspect which covers the whole world. Climate change affects whole world. Flood and natural disasters or man-made disasters, they are affecting everybody's life. As we know, the pollutant contents is associated directly with chemistry, though it is affecting people's health. So in this 2 days webinars, I think all these aspects will be covered, and it will be very beneficial for the academicians also, because we will be listening to learned professors from all over world, and our students will also be benefited, and research scholars especially will come to know how to adopt research techniques in these issues and what are the areas which they can take up in their research. So these and other themes will be covered in 2 days deliberations, and hope we will come out with constructive ideas that will positively impact society. Because whether it is science or humanities or social sciences, main purpose is how it positively affects the society, whether it is national, we are talking about India, or international, because these things are universal. Whether we chemistry teachers will be more apt to it, that whether it is organic chemistry or inorganic chemistry, it is existing in everybody's life, and how we can educate people or educate our children to better their life through proper health and hygiene, and prevent ourselves from different diseases such as corona. As we know that some people say that even corona was in lab made. So chemical tests were being, I think, performed, and it generated. So it is just one aspect, whether it is true or myth, it's another aspect. But chemistry affects everybody's life. So it is a multidisciplinary approach, and I wish and congratulate my organizers of a department, especially Department of Chemistry, that they have come forward to organize this international webinar. My best wishes. Om Namah Shivaya.
Thank you so much. Thank you very much, ma'am, for your kind words. We, the Department of Chemistry, Navyuga Kanya Mahavidyalaya, are proud to host the first international conference, ICCHHE 2022, in collaboration with Chemical Research Society of India and Association of Chemistry Teachers, and they are known as CRSI and ACT respectively. CRSI and ACT, both organizations promote and organize seminars, symposia, workshops, conferences, and other related programs to facilitate and promote research in all branches of chemistry. The central idea behind the conference is to develop a mutually effective relationship among people of different streams. To accomplish our goal, we have come up with 13 different sub-themes related to the main concept of the conference. We have the eminent speakers, Dr. D.K. Upreti, Professor Satya Narayan, Professor Naveen Khare, Mr. Anand Pandey, Dr. Sanjay Kumar Pandey, Dr. Sanjeev Kumar Ojha, Dr. Alok Ranjan, Dr. Upendra Rao Gola, and Dr. Ranjana Kanchan with us to provide an insight on the topic and to share their valuable knowledge. Owing to the current pandemic, we are conducting the conference in hybrid mode. I hope that Chemistry for Health, Hygiene and Environment can play a major role to regenerate nature to everlasting extent with human interference, especially during COVID-19 times, for sustainable existence. I hope this conference will bring together researchers, academicians, students, and professionals on a common platform so as to exchange ideas and stimulate discussions on different sub-themes with a central focus on the importance of hygiene to maintain good health, and definitely it would sensitize us for a healthy and green environment. Now, I would like to recognize our collaborators, Chemical Research Society of India and Association of Chemistry Teachers. Without their support, this conference would not have been possible. In this sequence, I feel so privileged and honored to get the opportunity to invite Professor Vinod Kumar Singh ji, the President CRSI and the Chair Professor, Department of Chemistry, IIT Kanpur, for his formal address. I welcome you, sir, to the conference. May I request Professor Vinod Kumar Singh to please come for the formal address. I think sir has still not joined us. Professor Vinod Kumar Singh ji, are you there, sir? Okay, I think sir has got a little late. In the meantime, our second collaborator, Association of Chemistry Teachers, we are pleased and honored to welcome Professor Brajesh Pare, the President of Association of Chemistry Teachers, for his formal address and to release the most awaited e-souvenir. Over to you, Pare, sir.
Yeah, thank you, Dr. Neha. I'm audible?
Yes, sir.
Okay, thank you very much. It's a very good morning to all of you. It's a very auspicious day today, as the principal of the college has narrated. It's a Mahashivratri today, and it's a wonderful to be to the part of this program today. I congratulate the organizer, the Kanya Mahavidyalaya Lucknow, for organizing such a wonderful event, the International Conference, Chemistry for Health, Hygiene and Environment. And I have just listened, the battery of speakers going to be speak, in the next 2 days. So I congratulate you for organizing this program. Associate of Chemistry Teachers, we are 20 years old now. It was established somewhere in 2000, and we are now into 20th year, already completed. ICCHE has a mandate to popularize chemistry among the young students by doing many conferences, workshops, and various events we have been doing. I'm so happy that Dr. Neha and Dr. Sunita, they are associated very actively with various programs of ICCHE throughout the year, So, and we look forward to have many more such events together. Once again, I congratulate you, all the organizers, for the successful conduction and organization of this international conference. And I thank you for giving me this opportunity to declare your this booklet to open. I release it. I declare it's a release. Thank you very much.
Thank you very much, sir.
So are you going to show that the booklet?
Yes, sir.
Yeah, congratulations once again. It was really beautiful. And on behalf of SET and on my personal behalf to Professor Manjula, the principal, and Dr. Neha and Dr. Sunita. So all the best. And once again, congratulations. Have nice and wonderful 2 days ahead. Thank you.
Thank you so much, sir. I hope Professor Vinod Kumar Singh ji has joined us. Professor Vinod Kumar Singh ji, are you there? I think sir hasn't joined because he was supposed to join at 10:15 and he's very, very punctual to the timings. And today he's having flight. It might be possible because he's stuck somewhere on the way. But anyways, going ahead, now I would like to start the first session of ICCHHE. We'll be taking a break if he joins because he's specially managing to join us for the conference. So whenever he joins, we will just ask him to address. Now, may I please invite... Yes. May I please invite Dr. Sangeeta Kotwal to please start the first session and to welcome Professor Satya Narayan Singh ji.
Thank you, Neha. Good morning to all of you. Has Professor Satya Narayan joined?
Yes, ma'am. He's there.
Okay. Then thank you so much. Okay. On behalf of all of us here on this platform, I would like to take this opportunity to welcome Professor Satya Narayan, our keynote speaker today for the first session. And it is indeed my privilege and pleasure to introduce him to our audience. His list of achievements is very, very long, and I hope this brief introduction will do justice to it. So I'll just start. Professor Satya Narayan is currently a professor in the Department of Anatomy and Cell Biology, College of Medicine at University of Florida, Gainesville, Florida, USA. He obtained his PhD degree in biochemistry from Punjab University, Chandigarh, and MSc and BSc from University of Allahabad. He received his postdoctoral training at Louisiana State University, Baton Rouge, Louisiana, USA, and at University of Texas Medical Branch, Galveston, Texas, USA. He has also served as a chemist in charge at Curewell India Private Limited, Faridabad, Haryana. His research expertise includes DNA repair and transcriptional regulation mechanisms and their implication in therapeutic development for colorectal and breast cancers. His focus is to identify small molecules that can sensitize drug-resistant and cancer stem-like cell populations in tumors with minimum or no toxicity to normal cells. He has another research interest in cigarette smoke-induced breast carcinogenesis. He has described for the first time the role of Agnomatous polyposis coli, APC, in cigarette smoke-induced transformation of normal memory epithelial cells. He has published more than 100 research papers, including review articles and book chapters in these areas. His research work has been funded by the National Institutes of Health, Flight Attendants Medical Research Institute, American Cancer Society, and University of Florida. Dr. Narayan holds 4 patents to his scientific discoveries. He serves as an associate editor of Medical Genetics, Medical Genomics, Journal of Carcinogenesis, and is the editorial member of the International Journal of Biochemistry and Molecular Biology. He is actively involved in the medical science education at the University of Florida College of Medicine. He serves as a course director of Advanced Cell Biology and Tumor Biology, and as a co-director of Medical Cell Biology. He also teaches several other courses to biomedical sciences and dentistry students. Among several honors and awards, He has received 7 times the Exemplary Teacher Award by the College of Medicine, University of Florida. And sir, it is truly our honor to have you on this platform today as the keynote speaker, and a very, very warm welcome to you again. And now I would request you to address our participants who are eagerly looking forward to your speech. Thank you so much. Professor Satya Narayan, over to you, sir.
ॐ अज्ञानतिमिरिन्दास्य ज्ञानाञ्जनशलाकया चक्षुरुन्मिलितं येन तस्मै श्रीवे नमः। हे कृष्णकरुणा सिन्धो दीनबन्धो जगत्पते गोपेश गोपिकान्त श्रीराधाकान्त नमोस्तुते। Indeed, it's my great pleasure to be here today. I would like to thank Dr. Sangeeta Kotwal for the kind introduction. I do not really deserve for this. I don't know, this is a midnight here, whether I will be able to justify today my presence or not. But indeed, I'm very grateful to be here today. And I sincerely thank to Dr. Neha Agarwal for her kind invitation to participate in this conference. I didn't know about this conference. One day she called me and she asked me to join this, and I was very happy and thrilled to participate because I'm Indian. I'm born Indian, and my heart is always there. But with body, physically, I'm living in the United States, but with soul, I'm always in India. So also, I would like to extend my sincere thanks to Professor Manjula Upadhyay and Dr. Brajesh Pare and other distinguished guests and friends. I saw Dr. Sanjay Pandey has already joined here. I could not see others' names, so I don't know, but I welcome all of you to this wonderful conference, and I'm sure that everyone is going to actively get involved in this process and encouraged for the progress and establishment of this organization. So I'm going to talk about something which currently we are working on, which is related to drug development. So I would like to start with my slides, if I can. Can you see my slide? No, probably not, right? I need to share it first. Right? Share the screen. Can you see it now?
Yes, sir. It's visible now.
But I cannot see anything other than my screen. So at least can you see all these and can you hear me?
Yes, sir. It's clearly visible and we can hear you.
Okay, so because I'm not on my computer, I cannot see anything right now. I'm not seeing the audience. I'm not seeing anything. I'm just seeing the screen here. So,
all right.
So I will go blindly, not seeing anyone. I'm just going to begin my thoughts and ideas, what I have for you today. So as you can see from the first slide, that I'm going to speak about a novel therapeutic development for the colorectal cancer. And I'm going to try to start with the tone of this meeting, which is International Conference on Chemistry for Health, Hygiene, and Environment. And I would like to justify my presence here as a keynote speaker. And you can see even on the first slide that the good health, good hygiene is always smiling, and the bad health habits and our hygienic conditions, they are going to lead us to to shedding the tears. And in the middle is the consequence, which most of us are going to face if we are not very careful about our daily living habits. With that, I want to really justify my presence here, and I want to set up the foundation of this conference. And since my topic is related to cancer, I want to give you a brief understanding of the cancer. Cancer is a function of three very important factors: predisposition, environment, and aging. All these three components, they contribute in the development of cancer. Among these three, two are beyond our manipulations. We cannot do anything about predisposition. Predisposition means we are inheriting certain problems in our genome from our parents, and that we cannot correct by the means of any therapeutic approach. So if we are inheriting any defective gene from our parents, then sometimes during our lifespan, that defective gene is going to create the problem. It is going to show its phenotype. That means whatsoever the type of gene it is going to show the type of cancer in our lifetime. The third component is the aging. We cannot also do anything about the aging. We are going to grow old, and by growing old, so many things they are going to change in our body. And as the body weakens, the cancerous conditions or tumorigenic conditions, they also increase due to expand of our life. Only one thing among these three things is the one which we can manipulate, which we can handle, which we can take care. That is the environment. This is the only thing we can really manage. We cannot manage predisposition. We cannot manage aging. We can manage our eating habits. We can manage our drinking habits. We can create hygienic atmosphere around us so that the predisposition even we are carrying can be either minimized or can be prevented completely. For example, I'm going to talk about the colon cancer. The predisposition for the colon cancer occurs through the mutations in APC gene, which is called as adenomatous polyposis coli. So if a person is carrying the mutated gene of APC from one of his or her parents, then that person is going to be predisposed to colon cancer sometimes during their lifetime. Since the person is carrying only one mutated gene and another gene is still functional, the person will survive for a very long period of time until the wild type gene or the wild type allele which came from the normal parent is still active. But if our environmental conditions are not good, if we are exposed with environmental carcinogens, either through inhalation or ingestion or drinking or smoking, the smoke has 4,000 different types of chemicals. 74 of them are proven carcinogens. So these chemicals, when they enter in a predisposed person's body and they attack to the wild type DNA, which was coming as a good DNA, good gene from the wild type parent, becomes mutated. So under those conditions, if both the genes they get mutated, then person is definitely going to have the cancer. There is no prevention after that. So we can delay, we can prevent, we can stop even the carcinogenic process if we can manipulate the environment. We cannot do anything with predisposition, we cannot do anything with aging, but we can do a lot about the environment. So I think this conference is going to address all these, if not aging, then at least the the very important aspect of the environment, which is going to be very important for controlling some extent of aging and many aspects of the predisposition. So with this, I'm going to start my colon cancer and drug development projects. So colon cancer is a third major health problem worldwide, and major problem occurs during the metastasis stage of the colon cancer. So there is no really any improved therapy for this, and there is a high incidence of drug resistance during the metastasis. And the therapeutic plateau is in our clinical laboratories because the existing chemotherapeutic drugs are decades ago discovered, and still they are being used with different modifications. So there are no really new drugs, and in other words, we can say that there is no any appropriate drug which can we say that this is going to treat or this is going to cure the cancer. So there is no cure for the cancer yet. There is some delay in the progression of the cancer. There is some hope for the lifespan of a patient, but there is no cure because of the resistance, and there are no new avenues yet discovered for the colon cancer. So the first line chemotherapy for the metastatic tumors is the combination of three drugs, which is a 5-fluorouracil, leucovorin, and oxaliplatin. It is called as FOLFOX, and I will be using this term throughout my presentation. Even with this first line chemotherapy with FOLFOX, the 5-year survival of a colorectal cancer patient remains very low. That is only up to 14%. So that means we don't have really a good drug at this moment in our hands. So even these 14% cases, they do not go more than 5 years because the colon cancer cells, they develop resistant very quickly against the FOLFOX. So that's another problem which is created within our system. And at the same time, the FOLFOX regime leads to a significant toxicity. It is very costly, and it creates very much patient inconvenience. So the development for a second line chemotherapy, which can either improve the therapeutic efficacy of existing FOLFOX drug, and it can remove the drug resistance, is very much needed at this moment. Another component which I will be discussing today, apart from the FOLFOX resistance, is the TRAIL resistance in colorectal cancer cells. What is the TRAIL? It is a tumor necrosis factor related apoptosis inducing ligand. So this is the abbreviation TRAIL for this long name. And the TRAIL signaling is a physiological mean in our body system which kills very specifically the cancer cells, and it prevents the killing of the normal cells. So this is a natural defense system given to our body. Now the problem becomes, again, the limitation that the TRAIL therapy becomes resistant to the CRC, against the CRC cells. So two drugs which can be effectively used for the colorectal cancer therapy, they both develop or create resistance very quickly to the cancer cells. So what can be done? How we can use the TRAIL, which is a natural system in our body, to avoid this resistance and to improve the therapeutic efficacy? That's what is our purpose and the goal for the discovery of this project. So I'm going to give you little bit scientific background, or you can say that theoretical background of this whole proposal, which I'm going to talk about. So how the TRAIL develops or creates the resistance into the colon cancer cells. So TRAIL signaling, when is induced as a ligand, binds to its receptor DR5, and then it makes disc complex, which is a death-inducing signaling complex. Then other proteins like FAD and procaspase 8 bind to this complex, in which the procaspase 8 is activated into caspase 8, and caspase 8 then activates procaspase 3, and procaspase 3 is broken into the active caspase 3, and this active caspase 3 comes after the activation by caspase 8. And active caspase 3 cleaves ICCHE, which is a vital molecule, which is an inhibitor of caspase-dependent DNase. And once the DNase is active and the inhibitor is removed by the caspase 3, then it cleaves the chromatin, heterochromatins. And once the chromatins are broken, then the cell goes into cell death. That is called as drug sensitivity or sensitization of the cells, or cell death. So this is the trail-induced pathway. So why trail becomes resistant against these cells? Because if in the system, in the colon cancer or in any other cancers, if the P21, which is a cyclin-dependent kinase inhibitor, which is involved in the cell cycle regulation, becomes overexpressed due to certain reasons which we are going to discuss today, then it acts as an oncogene actually and becomes a resistant factor. And currently P21 has been targeted as a therapeutic target for developing new drugs. So how it happens? So when there is an increased level of P21, then it binds with procaspase 3 and makes procaspase 3 P21 inactive complex, and caspase 8 is unable to cleave or activate caspase 3 because of the P21. So if the procaspase 3 is not activated into caspase 3, then apoptotic pathway will inhibit right there. And that is the drug resistance, because any drug actually ultimately activates caspase 3. And the caspase 3 activation is necessary for inducing the cell death or abolishing the drug resistance and inducing the apoptosis. So if the procaspase 3 is bound with P21, then there will be resistance because caspase 3 is not available. The second pathway which I'm going to talk about is the Folfox resistance. So theoretically, the PP2A, which is a protein phosphatase 2A, so it is a phosphatase which dephosphorylates many oncoproteins, which are also the resistant factors which has been identified in colorectal cancer. So if there is a induced level of PP2A, then it will decrease AKT, which is another oncoprotein. So phospho-AKT level will be reduced if PP2A is more. But if PP2A is less, which is the case in colorectal cancer cells, then AKT will be more. There will be more mTORP, because mTOR is the substrate for the AKT. It will phosphorylate mTOR, and phosphorylated mTOR will phosphorylate 4-EBP1. So there are a lot of things you don't have to remember. You just understand that once the PP2A is down-regulated, there are cascade of kinases which are in one axis. They are going to be down-regulated. And they will be up-regulated if the PP2A is down-regulated. And the up-regulated concentration or amount of the 4-EBP1 phosphorylated form is involved in protein synthesis or translation of the protein. So the unphosphorylated form of 4-EBP it interacts with EIF4 translation initiation factor and makes it inactive. For the activation, you need EIF4E to complex with EIF4G. Then it makes active translation initiation complex. So when you have a phosphorylated form of 4-EBP, it will not interact with this complex, then it will form active cap-dependent P21. So there will be more P21 translation, and the increased P21 level will interact with procaspase 3 and will make it inactive. So you can, if you put these two pathways together, then you will see that the downregulation of PP2A is causing the increased level of P21. And even if you have a trail activation, it is not going to help anyway, because the caspase 8 will not be able to cleave procaspase 3 because of the presence of the P21. So our strategy is to downregulate this pathway so that we can increase this activity and synergize the whole thing. I will be talking about later on in the hypothesis. So we have identified that the decreased level of protein phosphatase 2A and an increased phosphorylation of AKT1, mTOR, 4-EBP1, and increased level of P21 in Fol-Fox resistant CRC cells are present. And we have identified for the first time P21 as an acquired Fol-Fox resistant factor in CRC cells. So this is our finding, and that's how we got into this project, to find out how we can downregulate P21 so that we can downregulate this entire pathway, and ultimately we can achieve the cleavage of procaspase 3 and induce the sensitization of the cancer cells. So we wanted to see that whether this hypothesis is going to be valid or applicable or not. Then we decided to check if there is any data available in the Cancer Genome Atlas by Bioinformatics Analysis. So we found 3 studies which has been submitted to NCI related to Fol-Fox responsive and non-responsive colonic tumors. When we analyzed the data, we were very happy to see that in the non-responsive colorectal tumors, the PP2A and PP2B, both subunits, they were decreased as compared to the responsive tumors. And when we look at AKT1, 4-EBP1, mTOR, and P21 genes, they all were increased significantly in the non-responsive tumors, which was our initial observation as well. And we also found out from the bioinformatic analysis a decreased level of PP2A and B subunits. In this example, I'm showing you the B subunit here, was significantly downregulated in tumor samples compared to the normals. Not only that, this decrease was progressively observed when the colonic tumor was progressing from normal to transformation status and ultimately going to the metastatic stage 4. So you can see that there is successive decrease in the PP2A levels as tumor progresses from normal colonic epithelial cells to adenoma to carcinoma to metastasis. And very interestingly, it is very nicely correlated with the overall survival of the colonic tumor patients. When you have a high level of PP2A, then the patient's survival is much better than those who have a low level of PP2A. This suggests that if we can somehow increase the activity or the level of PP2A, then we can increase the lifespan of CRC patients. So that has been our goal, that has been our objective. Can we increase the PP2A activity to this level where we can protect the cells from the resistance, as well as we can increase the sensitization and increase the survival of the patients for a longer period. So with that in mind, we discovered a small molecule known as NSC49L as a lead compound. I'm going to just state NSC49 throughout my presentation. It's a long story how we discovered this compound, but this is a valid PP2A agonist. We found out that in a reconstituted system, the PP2A, our small molecule, induces PP2A catalytic activity in a dose-dependent manner, and at a very low, like a nanomole range, is its catalytic constant, that is just 14 nanomoles. So the K activation is 14 nanomoles only. So that's a very good finding as far as a drug development is concerned. Then we also tested its activity within the cell by isolating the immunocomplex from the treated and untreated cells. And again, there was a dose-dependent increase in its activity. Then we used PP2A inhibitor, which is okadaic acid, and we found out that the cells which has been pre-treated with okadaic acid, they have desensitized the effect of NSC and TRAIL or their combination. So you can see that when the NSC has a IC50 of 4.2, and when you have okadaic acid, then it is 6.96. So I'm going to use this term IC50 in the later presentations, which is going to be the indication of cytotoxicity or sensitization of the cells. So lower the IC50, higher is the efficacy of the drug. So that means the drug is sensitizing the cells more effectively if they have lower IC50. And if I have a higher IC50, that means those drugs are being desensitized against those cells. So cells are desensitized if the IC50 is much higher. So like in the case of combination treatment, NSC and TRAIL, the IC50 is 1.39, but when you have a blockage of the PP2A activity, then it is desensitized almost sixfold. So we have to look on these parameters when we will be going during our presentation afterwards. This is just the control showing you that okadaic acid does not have any effect on the TRAIL IC50 alone, or when the TRAIL or okadaic acid, they are put together in the cells. So our hypothesis is basically that the stimulation of PP2A activity will inhibit AKT-mTOR4-ABP pathway, and it will reduce the level of P21. The decreased level of P21 will release procaspase 3 for TRAIL-dependent synergistic sensitization of FOLFOX-resistant CRC cells. So I already told you the FOLFOX-resistant component and the TRAIL-resistant components. So our hypothesis is to reverse this entire pathway. So if we have a small molecule which can induce the PP2A activity, then the induced activity of this phosphatase will downregulate the AKT1 activity, mTOR activity, 4-EBP1 activity, and once the phospho 4-EBP will be reduced, then unphosphorylated 4-EBP will bind to EIF4 translation factor, and will inhibit the P21 translation. I'm mentioning here P21, but this is going to be applicable to the global protein translation. We are going to see later on whether this compound NSC is specific for blocking the P21 translation or the global translation as well. In any case, at this stage, if the P21 translation is blocked, then we are proposing that the decreased level of P21 will release procaspase 3 from its complex, and then the procaspase 3 will be activated by trail pathway by caspase 8, and it will generate active caspase 3. The active caspase 3 will induce sensitization of the cells. So this is our hypothesis. So we have now a small molecule which is going to act through this pathway and is ultimately going to cause the sensitization of the. So now I'm going to show you some data, and this is going to be a very busy slide, so I don't want you to worry too much. I'm just going to the slide. So in this portion of the slide, this is the ICCHE-2022, that is the toxicity experiment, showing you that the trail, we are going to use 4 different colon cancer cell lines in this experiment. One is HCT116 cells, which is a trail sensitive cell line, HT29, which is a trail resistant cell line, and the FOLFOX resistant clones, which has been isolated from HCT116 and HT29 cells. So 4 cells, 2 wild type cells and 2 FOLFOX resistant cells coming out from these parental cells. Now you can say that this is the FHC cell line, which is the normal colonic epithelial cell lines. So trail does not has any effect on the normal colonic epithelial cells, as I mentioned you in the introduction also. But it very nicely sensitizes the HCT116 cells as it is expected, but it does not have much effect on the HT29 cells. Again, it was expected. But when the cells were selected against the FOLFOX resistance, then whether it is HT29, which is resistant to trail, or HCT116, which are sensitive to trail, both are going to, they became sensitive to the trail. Why and how this got, they got sensitized after the FOLFOX selection? That is the purpose of our discovery. And when we use the normal colonic epithelial cells and wanted to check whether our small molecule, whether alone or in combination with trail, induces the cytotoxicity, then it does, but at a very low level compared to the colonic tumor cells. This slide shows you the HCT116 and HT29. Again, you can see the trail in combination to NHC compound dose dependently induces the sensitization of the HCT116 cells. So from 7 to 1.5 or 8 to 1.5, that means lower the IC50, higher the toxicity or sensitization or apoptotic phenomenon. In HT29, you can see that there is no really much effect. The reason is that the concentration of the trail, which we are using here, is just 0.5 and 1 nanomoles, which was used against the trail sensitive cells. And the IC50 of the trail in HT29 almost is not detectable here. That means you have to use at least 10 nanomoles or more to see any effect here. So that's why you are not seeing any effect at this level. But when we use the FOLFOX resistant cells, either HCT116 or HT29, they both were dose dependently affected in combination effects. So when you combine trail with NSC, then you induce the cytotoxicity in both types of cells. So why these FOLFOX cells, they got sensitized by this combination strategy? I'm going to show you some data and will come to the conclusion of the hypothesis that why and how it has happened. We did in vivo experiments also using the PDX model of colorectal cancer stem cells, and you can see that these cells were derived from the human colonic tumor patients, and in the untreated tumor xenografts or stem cell tumor xenografts, there was a very rapid growth in the tumor. But when the 5-fluorouracil, which is the backbone of the FOLFOX therapy, was used, then up to 23 days, the tumor was regressed, but after that, regrowth started. So this is the resistant aspect of the 5FU. But when we use the NSC alone or in combination, then the regression of tumor was completely taken care up to the 33 days of the treatment, and the tumor were completely almost shrunken. So that means we have a novel compound which can be translated into the therapeutic development in coming future. Also, we did the cytotoxicity experiments and found out that these compounds are very minimally or no toxic to the mouse organs. We did the pharmacokinetic analysis also. So it has a very good absorbance and bioavailability. Only one drawback with this compound is that its plasma half-life is very low. So we are working at this moment to derivatize or develop the analogs of this molecule so that we can increase the plasma half-life and increase the bioavailability so that it can become a good druggable compound. And fortunately, we have already synthesized 3 analogs which are much better than the parent compound, and we are in the process of submitting the patent within 1 or 2 weeks. So now we did the bioinformatics analysis. We did the RNA-seq using the FOLFOX-HT29 cell mRNA, and we were very happy to see that in our combination treatments, we screened all kind of phosphatases and found out that both subunits of PP2A are the ones which are significantly increased, either NSC alone treatment or in combination. The trail has no effect and the vehicle control does not have any effect. And then we looked at the RNA-seq analysis of P53 and different cycle-independent kinase inhibitors, and we found out that P53, P63, P73 levels, mRNA levels are completely gone. There are no expression, but P21 in combination is highly overexpressed. And there is another kinase inhibitor, which is a P16, is also induced, but at this moment we are paying our attention to P21. And when we looked at the different cell lines, then we found out that those cell lines we have a lower expression of PP2A, which is a phosphatase. They have a reverse or inverse relationship with phospho-AKT. So there was a very increased amount of phospho-AKT, increased level of P21. So that means lower the PP2A, higher is the AKT1, and then higher is P21. And both these are the resistance factors for colorectal cancers. So this has been known for colorectal cancer, but not known for FOLFOX tumors. And P21 absolutely was not known as a resistant factor in FOLFOX cells. So then these are very busy slides. I don't want you to go too much into details. I'm just going to point out some important features about this. So this is a FOLFOX cell line, lane 5, and you can see that in the parental HCT116, AKT, phospho-AKT level is very much negligible. But when it goes to FOLFOX resistant clone, then there is a high expression of phospho-AKT. But when you do the combination treatment, then again, the AKT level is completely gone. And the P21, P53 is not expressed, as you can see at the mRNA level also. But P21, which is very low in the parental cell line, but very much increased in the FOLFOX selected clone, and combination treatment decreased its level. When these are decreased, then these are the apoptotic markers. Then the procaspase, cleaved procaspase 8, which is active caspase, is increased. Active caspase 3 is increased, and PARP1 is increased, which is all these are the apoptotic markers. And same thing has happened with FOLFOX HT29 also. So you can see that upregulation of AKT1, P21, and then downregulation when the combination treatment is done. And there is an increased level of the apoptotic markers. Then we looked at the AKT levels, and there are 3 AKT proteins: AKT1, AKT2, AKT3. So it is only AKT1 which is affected by the combination treatment, not other components. And then we looked at the mTOR. So it is the phospho-mTOR which is significantly downregulated, and phospho-4-EBP1 is significantly downregulated. And you can see this is correlated with the apoptotic markers. So anti-apoptotic proteins, they are decreased in combination treatment, and the pro-apoptotic proteins, their level is increased in the combination treatments. So this all puts together to our hypothesis, which I discussed to you earlier. This slide shows you that the apoptosis is increased in the combination treatment compared to the untreated cells, or when they are individually treated either with the NSC alone or the trail alone. There is increased apoptosis or significantly increased apoptosis when the NSC alone is treated, but it is much more induced when the combination treatment is done. And the same thing we observed with the phospho-HT29 cells as well. So now I want to talk about the important role of P21, because a whole thing is dependent upon the P21. So right now, up to this point, I was mentioning to you that downregulation of P21 is very important, and that is occurring by upregulating the P21, PP2A levels. Can we reverse this phenomenon? So we tried to use 2 different cell lines. One has the P21 wild type cells status, and another one is P21 knockout status. So if there is no P21, that means this cell will become much more sensitized. And you can see, if you look at just the trail activity, then the IC50 drops from 0.5 to 0.5. And similarly, when you determine the IC50 of the NSC compound, that is also very much reduced in the combination, and it provides the synergistic effect of the combination of the NSC and the trail when you use the P21 knockout cells. So if you downregulate P21, or if you remove somehow the P21, like in this case by knocking it down, then you can synergistically sensitize the cells in the combination therapy. To prove this point, then we overexpressed the P21 in the knockdown cell lines, and then you can see that this the desensitization occurred. So the trail IC50 is now much more increased when the P21 is overexpressed in the knockout cells. And the same thing has happened when we looked at the NSC dependent toxicity. And then you can see that when the P21 is knocked down, then the trail and NSC, they reduce the IC50 from 11 to 1. But when the P21 is overexpressed in the P21 minus cells, then even in the, so when you have the trail treatment, then you can see these P21 overexpressed cells, they are much more resistant compared to the compared to the trail and NSC treated cells. Similarly, when you determine the trail activity, then you can see that in the P21 knockout, there is a certain number, like 0.32 nanomoles of the activity. But when you have a trail plus NSC, then there is a further downregulation in the IC50. That means more sensitization is occurring. But when you overexpress the P21 under these two conditions, then you are protecting the cells from 0.3 to 1 and from 0.03 to 0.3. This means about 10-fold more desensitization is occurring when you overexpress the P21 back into P21 knockout cell lines. So question then arised. In the beginning, we saw that there was more P21 mRNA, but we are seeing the decreased protein level of P21 in colorectal, in Folfox resistant cells when they have been treated with combination treatments with NSC as well as with the TREL. So what could be the mechanism? So one thing is that the lot of mRNA is there in the Folfox cells. The P21 mRNA is in abundance after the NSC treatment. So whether it is at the level of protein degradation or protein synthesis, these are the only two parameters, because the transcriptional control is not a mechanism here. So we checked at the protein degradation of the P21, and we found out that protein is not really going through degradation. It is pretty much stable after the treatment. So the left parameter is remaining, that somehow our small molecule is blocking the translation of P21, and then we went ahead and looked at those parameters. So first thing, remember I told you that when there is a 4-EBP1 in its unphosphorylated state, then it interacts with EIF4E translation initiation factor. And the complex of these two molecules, they block the protein synthesis. And we did see that after NSC treatment, there is a increased co-localization of 4-EBP1 and EIF4E. That means the binding of these two proteins has been increased, and it will co-localize only when 4-EBP1 is unphosphorylated. To prove that whether this complex is involved in the protein translation, we did some set assay, and we found out that the NSC treatment by itself does under no conditions, this is the control, and there is a very good protein synthesis here. But when we used cycloheximide, which is a global protein synthesis inhibitor, then you can see that the protein synthesis is blocked. The good thing is that when we use the NSC treatment, then the global protein synthesis is not blocked. This suggests that if NSC compound is acting at a translational level on P21, then it could be very specific to P21 only, not inhibiting other protein syntheses as cycloheximide does, which is a global protein inhibitor. This small molecule is not a global, whether it is a global protein synthesis inhibitor or not, we cannot say at this stage, but it looks like that it is not. So we further went ahead and did immunoprecipitation from this complex and found out that indeed the NSC49 does not globally block the protein synthesis as cycloheximide does. It just inhibits the P21 only, and you can see the quantitation given here. So if you remember the hypothesis, then we can say that the NSC treatment basically blocks the synthesis of or the translation of P21, and this translation, blockage of the translation initiates at the PP2A level. So NSC49 induces the activity of PP2A that downregulates AKT1, and AKT1 downregulates mTOR and mTOR downregulates 4-EBP1. Once there is a 4-EB, so remember that all these are the phosphorylated forms. So when the phosphorylated form of 4-EBP1 is decreased, then unphosphorylated form of the 4-EBP1 is increased, which interacts with the translational initiation machinery and blocks the translation, and that's what it is happening here. So a small molecule is selectively blocking the translation of P21. Now, these are the controls to show you that our experimental conditions are valid and they are working. Just in a few minutes, I just want to conclude one more experiment showing you that can we reverse the caspase 3 effect and show again the desensitization against these drugs so that our entire pathway is valid and it is it can be translated into the therapeutic aspects. So if you look just the fall Fox cells and in not comparing to the HT29 because our main focus is these cells, you can see that it has a more expressed level of P21 compared to the parental cells, and NSC treatment downregulates the P21 level here, and the trail does not have any effect on the P21 level. But when you overlay these two, then it sees these kind of effects. And if you look at the procaspase 3, then its level is almost the same. But when you overlay or combine the P21 and procaspase 3, then you can see that there is a very nice co-localization of these two proteins, which is decreased after NSC treatment. And if you have a trail combination, then there is a further decrease, but almost 70-80% decrease has occurred already after the NSC treatment alone, because the NSC goes through the PP2A pathway and blocks the synthesis of P21. To test that, we used caspase 3 inhibitor, which is a ZDEBD. This is a caspase 3 inhibitor. So if we can inhibit the activity of the caspase, then we should decrease the sensitization of the Pol Fox cells against these drugs. And exactly that's what we observed. If you have the treatment of NSC and the trail together, then you can see the significant decrease in the IC50 compared to the control cells, untreated cells. But when you use the caspase 3 inhibitor, then you can desensitize these cells and increase the IC50 significantly from NSC and trail treated cells. So that means the last component, which is the activation of the caspase 3, is valid and it plays a role throughout in our hypothetical pathway. And not only that, you can see that it reverses the levels of P21. So this is a treated cells without caspase inhibitor, and this is the caspase inhibitor plus. So the P21 level comes back. The procaspase cleaved caspase 8, which was downregulated after the combination treatment, which was activated, actually its level was increased, because we are looking at the cleaved caspase. But when we have an inhibitor, caspase 3 inhibitor, then its level is decreased, because that pathway, entire pathway is going to be blocked, and the procaspase is blockage. P21 is novel analogs, which are much more potent and than this lead compound. And very soon, actually, to the treatment of this disease. Come to the conclusion now. Showed you that this small molecule is a very good, and its efficacy is very much validated in our experimental protocols, and we used it against in vivo and in vitro studies. And this has a very potent, it is a very potent agonist with a very low in nanomole range of catalytic activity. And we also showed you that this treatment inhibits AKT1, mTOR, 4EBP1 and P21 axis in FOLFOX resistant colorectal cancer cells. I showed you that the decreased level of P21 and increased level of cleaved caspase 8. Remember, cleaved caspase means active caspase, and cleaved caspase 3 and cleaved PARP1, they all were increased in the resistant cells. And then we also showed you that if you knock down the P21, then you synergistically enhance the TRAIL-induced NSC49 and TRAIL-induced cytotoxicity in knockout cells, and you can reverse it back by overexpressing the P21 in these knockout cells. I showed you that the NSC and the TRAIL treatments, they decrease the level of phospho-4EBP1 and increase the interaction of unphospho-4EBP1 with EIF4E translational initiation complex, which is an indicator of decreased protein synthesis. I also showed you that we can reverse this phenomenon by using the caspase 3 inhibitor. So we think that NSC is a potential phosphatase 2A agonist that enhances the therapeutic efficacy of TRAIL. Thus, our discovery proposes a novel analog that can be developed as a future second-line therapy for FOLFOX-resistant CRC cells by inhibiting AKT1, mTOR, 4EBP1, and P21 axis. These are the collaborators from our own University of Florida, different laboratories, and I have an active collaboration with Dr. Arun Sharma at the Penn State Cancer Institute in Hershey, Philadelphia. He's a medicinal chemist who is involved in the synthesis of analogs. And Dr. Jay Sharma from CellProGen Incorporated, Terence, he does some of our in vivo experiments. And then Dr. Adit Majumdar from the VA Medical Center in Detroit, he kindly provided us the FOLFOX-resistant colon cancer cells. Now, I would like to stop here with extending my sincere thanks to all of you for your time. I hope that I was able to communicate my message to you. If there has been any mistakes or shortcomings, please forgive me. I don't know if we have any time, but if you do, then I will be happy to answer any questions if you may have. Thank you so much. Have a happy Shivratri, Mahashivratri.
Thank you. Thank you so much, sir, for your valuable speech. And you managed to join us in spite of your odd timings between India and USA. Now this is the time for interactive session. If anyone wants to ask any question, you are kindly welcome to ask.
Good morning, sir.
Yes, sir.
Myself, HN Singh. I am grateful from patrons to participants and offering my gratitude to the team who is conducting and the speaker. And if anyone don't mind, I thanks to Corona also, because this has given birth to webinar so that so many being distant places we can interact. I am a physicist. My principal has not given me the leave to join it. So today is holiday, no problem. Tomorrow I will carry my mobile to college and do the office work with listening the lectures. So at least I may be aware about the things going on and done by our learned educationists, scientists, and so on. Sir, as a physicist, I have a conviction that our body is made up of around 33 trillion cells. and each cell has its own mind and body as a structure. Whatever the disease or happiness of the individual cell, we are looking for the external, resistive and treating. But what I have a conviction, may or may not be right like that for a physicist, but I have a conviction that source of the happiness or diseased part of the individual cell is with DNA. And if we identify and repair the DNA, which part of DNA is causing a part of the same cell to be diseased and the remaining is happy, and if we cure after some time, again it becomes diseased. So if there is some methodology coming in future, if we can identify in DNA what is the source of this cell, its happiness and designing the mind and structure, and if we repair there, the cell will be automatically correct forever as internal treatment. So this is just my conviction as it is.
No.
So, yes, sir.
So you are right, actually, and you know that cancer is a genetic disease, and it's
Right, sir.
with the defects in DNA. And
Yes, sir.
as my first slide in presentation, I mentioned that environment plays a very major role
Yes,
in
sir.
the disease development process.
Yes, sir.
We are drinking, eating so many, and we are exposed with the industrial pollutants, automobile exhaust, and so many of the environmental pollutants we are
Yes, sir.
inhaling or taking in any way in our body, and
Right,
it
sir.
is going to interact with our genome one way or other.
Right, sir.
Fortunate part is that our body has a very, very efficient DNA repair mechanism.
Right, sir.
So whatsoever the damage is occurring, it is
Yes, sir.
repairing at the same time.
Right, sir.
But if we are overwhelming our DNA repair system,
Right, sir.
then that's the problem starts.
Right,
So
sir.
cancer is not only a genetic disease, it is
Right,
a
sir.
clonal disease. So cancer
Achha.
is a clonal expansion.
Right, sir.
You mentioned that we have a 33
Trillion cells. Yes, sir.
it's fine, but only one cell which experiences the defect in its genome. If that particular cell was left without repair, then in the many cell divisions, that
Right,
one
sir.
particular cell is going
Right,
to
sir.
be full-grown tumor.
Yes, sir. Yes, sir. Yes, sir.
So we have to take care of ourselves.
Right,
We
sir.
have to try to manipulate our living conditions in such a way that
Right, sir.
any predisposed gene
Right,
which
sir.
is present in our body should
Right, sir.
not get the second hit.
Right, sir. Right, sir.
You know, cancer occurs
Absolutely. Yes,
by
sir.
second hit mutations.
Right,
If
sir.
the wild type allele also
Right, sir.
gets simultaneously mutated.
Right,
And
sir.
we can try. We cannot really say that, yes, I'm going to do it.
Right, sir.
It's all random phenomenon. It's not dependent upon us. Only
Right, sir.
in our hands is we can try. We can minimize. We can
Right, sir.
adopt a healthy life, healthy eating, healthy drinking, healthy
Right, sir.
sleeping. All these
Right, sir.
conditions, they are within our approach, within
Right,
our
sir.
reach. We can minimize the environmental pollution, but in
Right, sir.
industrialization, we are
Right,
living in
sir.
industrialized world.
Right, sir.
We are creating problems ourselves.
Right, sir.
So-called being a progressive people, we are going backwards.
Yes, sir. In that sense. Yes, sir. Yes, sir.
But we are not seeing how much behind we are going.
Right, sir.
And chemotherapy is a holistic treatment. We don't have any targeted therapy yet.
Right, sir.
So the entire body is being treated as a whole, although the tumor is present in very certain area in the body.
Right, sir. Sir, in some sense, nanotechnology may help in the coming future.
It can help to reduce the dosage effect. Like
Right, sir.
if a certain amount of the drug is needed to produce the therapeutic efficacy at certain level,
Right,
the
sir.
nanotechnology can reduce that dose.
Right, sir.
And sometimes if you can tag some specific marker to the nanoparticles, then it can
Right, sir.
deliver to the tumors also. So that is
Right, sir.
another
So targeted treatment. Yes, sir.
targeted delivery
Right, sir. Right, sir.
approach through nanotechnology as well. And these
Yes.
things are very aggressively researched these days.
Yes, sir. Yes, sir. Yes. Thank you.
We are still far away.
We will reach there. सर एक सर इस पे एक शेर है कि मंजिल तो मिलेगी भटक करके ही सही, गुमराह तो वो लोग हैं जो घर से निकले ही नहीं. तो we the scientists approaching, and definitely tomorrow is ours and we will solve the problem. Thank you so much, sir. Thank you so much.
आपने जो मंजिल की बात कही ना,
Right, sir.
दिनकर जी की एक कविता आ गई, एक लाइन
Yes,
सुना
sir.
देता हूं आपको।
Yes, sir.
एक दिन राह मंजिल बनेगी स्वयं। दिनकर जी
Right, sir.
कहते हैं,
Right, sir.
दिनकर जी हमारे राष्ट्रपति।
Right,
एक
sir.
दिन राह मंजिल बनेगी स्वयं,
Right, sir.
थक कर न राही अभी बीच में, सोच मत ले कि मंजिल बड़ी दूर है, सिर्फ इतना कि जो यह कदम चल रहा वो अधूरा रहा या कि भरपूर है.
Thank you so much, sir.
सामने देखिए धार बहती हुई, हाथ कहती हुई यूं चली जा रही, एक पल भी रुकी है नहीं इसलिए कि एक दिन सरित सागर बनेगी स्वयं.
Thank you so much, sir. Thank you so much, sir. Thank you so much, sir. I am grateful to the whole team. Thank you.
Sir, I have another question. Someone has sent me in the chat box. Dr. Sunita Singh has sent it to me. Is there any side effect which is associated with the usage of this drug, NSE49?
Yes. I showed you one slide. So far, up to the extent we have treated and up to the amount we have treated, we didn't see any significant side effects with 5-6 organs which we looked at the pathological signs. So it does not show any side effects yet. But it's a long study. It needs to be done at a large scale to definitely describe about these things, and we have to do the blood chemistry and all those things. Has not been done yet, but we are in the process of doing it.
Quick question. Narayan ji, very impressive talk.
Thank you so much.
Very, very nice talk. I really overwhelmed. So a quick question. For this small molecule that you have tested for CRC, for PP2A, are you looking for some other cancer type where PP2 is also implicated?
Yes. We have looked at in the pancreatic cancer. We have looked in glioblastoma. We have looked even in the breast cancer, and it works because PP2A is a ubiquitous molecule. It is present everywhere, but it will be more effective to those tumors where its level is down-regulated. That's where its efficacy is better.
Yes, fantastic. Congratulations.
Thank you.
Is there any question from the participants? Sir, I have another question.
Yeah.
As soon as the patient gets to know that the person has developed cancer, the rate at which it is spread in the body, that is doubled. So how can we get over with that feeling? And is there any, what medicine can do in that case?
Well, as I mentioned you that, again, the spread of tumors, it depends upon the type of organ where it is located. If it is present in the soft tissues, then it very rapidly grows, like in the pancreatic cancers or gallbladder cancers, they are so rapid in their growth. Once they are diagnosed, from diagnosis, then 6 months to 1 year is the lifespan. So that means if it is diagnosed at a very early stage, then surgery is the best bet for any cancer at this stage. The chemotherapy, they can reduce the growth and they can spend your lifetime for a few years, depending upon the type of cancer. But if you say that the chemotherapy is going to completely cure, it's not happening yet. It can expand your lifetime 5 years, 10 years, depending upon the type of cancer, and it can manage somehow. But if it is diagnosed early on, if surgically it is removed and then some chemotherapy or radiation therapy is applied, then that person can live longer life also. So it depends again where the tumor is, when it was diagnosed, and even after the diagnosis, whether we are, again, it comes to how we are taking care of those things. If the tumor is diagnosed and if our habits are not changing, if we are heavy smokers, if we are drunkards, and if we are meat eaters, and if we are abusing our body from all these exogenous agents, which are stimulators of those tumors, then the rate of, or the growth rate is going to be very rapid, of course. So we can try, if we live a healthy life, we can minimize the growth as well.
Sir, I hope one more last question is there. How is this drug different from the other drugs which are available for the treatment of cancer, colorectal cancer?
One different, you can see that other drugs which I mentioned, that the first line treatment drug is the FOLFOX, which is the 5-FU leucovorin oxaliplatin. This is the combination of the drug. There are other associated combinations also. Some monoclonal antibodies these days has been developed. They are also being used, but all these, they are developing resistance very rapidly. So once the resistance is developed, especially in the late stage colon cancers, when the tumor is already in the stage of metastasis, then these drugs, they don't really do much to the patient. So we are hoping, since FOLFOX creates resistance by inducing P21 in those tumors, so we are hoping that we can downregulate the P21 by our strategy, so that the FOLFOX, wherever there is a FOLFOX resistant, we can still use our strategy to sensitize those advanced stage tumors which are ineffective with the FOLFOX treatments. So that's why we think our strategy can be better or useful against and it can be developed as a second line treatment.
Thank you, sir. Thank you very much, sir, for being there for us. I hope. Yes, sir. Thank you so much. Now, as a formal, yes, sir, you want to say something, sir?
No, I was going to say that I may not be able to stay too long, but
yes.
but okay, it's been really a great pleasure, and I'm surprised myself that I was up and active until this point.
Yes, sir. Thank you so much. Actually, thank you. Such a prayer that is intangible.
Thank you, sir. Thank you.
Yeah. So I would like to, I'm so privileged to thank you today, and I'm feeling so honored to propose a vote of thanks to you, sir, on this special occasion. Thank you again.
Thank you so much for the invitation. It has been a great pleasure.
Thank you, sir. Now, as we move further, we would like to invite the most talented Dr. Sonal Agarwal to introduce this eminent speaker, Dr. Alok Ranjan. Over to you, Sonal.
Yes. A very good morning to everyone present in the conference today. And a very warm welcome from the entire Navyuga Kanya Mahavidyalaya family. I am Dr. Sonal Agarwal, Assistant Professor of Psychology in Navyuga Kanya Mahavidyalaya, Lucknow. And I feel delighted and honored to introduce the next speaker of the day, Dr. Alok Ranjan, sir. Dr. Ranjan is presently working as a staff scientist in the Department of Chemistry in Stanford University, Stanford, California, USA. He has completed his doctoral thesis from IIT Kanpur, IIT Kanpur, India, and has gone on to do his postdoctoral fellowship from UT Southwestern Medical Center, Dallas, Texas, USA, and also from the Department of Chemistry, Stanford University, Stanford, USA. Sir has published numerous research papers in both national and international journals with very high impact factors. He has received several awards and honors, including the fellowship from UGC and CSIR. He has qualified the National Eligibility Test and also qualified GATE examination conducted by IIT Kanpur. He has given several global talks and presented his research works in various conferences in both national and international forums. I invite you, sir, to enlighten us with your talk today on this very crucial topic of HIV/AIDS. As we all know, more than 70 million people across the world are infected with this disease, and it has severe effects on the quality of life. So it is imperative to understand the progress in the treatment of this disease, which is HIV/AIDS. So on that note, I welcome you, sir, Dr. Ranjan, to please enlighten us with your lecture today. And thank you for coming. So, over to you, sir. Hello?
Dr. Alok Ranjan, are you there?
I am audible?
Yes, sir.
Yes, sir. Yes, sir.
Yeah. So, thank you for your kind introduction. And I am Dr. Narayan. I listened it nicely, and I also used to work in that area in my previous institute. So, I had a question, but I can send you separately. So nice talk. So if you don't mind, I can close my, because still I am in office, and according to rule, still inside I have to wear mask. So few minutes I remove it, but maybe I will go off video, but I will present nicely. So don't worry. So let me share my presentation first. So you guys able to see my screen?
Not yet, sir.
Now?
It's not visible.
So because we used to use Zoom, this is completely different stuff. Not supporting. Let me see what I can do. It's showing some system preference. I do not know why.
You may have to use Chrome.
Yeah, I use Chrome.
So it should work.
Yeah, I already switched Safari to Chrome, so but still.
I tested this system before. I was so nervous myself because I was using first time.
Yeah.
I called my son and I did virtual with him, and he taught me how to use these things. I said, okay, that's great.
Option is coming, but it's not showing you denied the.
Not on the screen here.
Yeah. Showing you you denied the recording permissions. Kind of stuff is coming.
Yeah, you don't need recording yourself, but if you can get the access to share. Are you clicking the share screen?
Yeah.
Go to the presentation, right?
Yeah.
So if you go to presentation and then say your entire screen.
Yeah, I did that.
Yeah, so, and make sure that you are not using the multiple screens. You have only one screen, right?
Oh, no. I have multiple screens.
So switch that from that screen to your screen.
Okay.
So that's what it is happening.
Maybe that's the reason, because I'm in conference room.
Yeah, that's the reason. Just switch from your screen 1 to screen 2 or 2 to 1, whatsoever.
No, still it's not working.
I'll just check, sir.
Okay.
सत्य नारायण सर प्रणाम, वंदना द्विवेदी।
प्रणाम।
से आपको देख रहे थे लेकिन डिस्टर्ब नहीं करना चाहे, बहुत अच्छा लगा। और हम लोगों का बहुत सौभाग्य है कि आप हम लोगों से जुड़े, बहुत अच्छा रहा। वेरी नाइस इंफॉर्मेटिव सेशन।
ये मेरा सौभाग्य है कि हमको आप लोगों ने इनवाइट किया और अच्छा लगा, मुझे बहुत अच्छा लगा।
जी सर, बहुत बहुत धन्यवाद, बहुत अच्छा, थैंक यू सर।
Thank you so much. There is nothing to thank me.
Yeah. Sir, it was visible just a moment before. It was working.
So basically it will not present PowerPoint, and I have to go to PDF format.
Okay, sir, we can opt that PDF format also.
So Ranjan ji, looks like I may have to leave now because
sorry sir, I didn't know.
And tomorrow I have a class.
Yeah, I know.
I will
midnight here. Yeah.
Otherwise.
Would you like to send PDF to us so that we can show it from our side?
Yeah, I think that is going to happen, because here it's not denying me the permission. I do not know why.
But we have permitted from this side. Actually, there is no problem from this side. You kindly send us through email. We will show it from here.
Okay, आप सभी लोगों को बहुत बहुत धन्यवाद और नमस्कार।
Thank you, sir. Namaskar, sir. Namaskar.
आपको भी बहुत बहुत धन्यवाद।
Yes, sir.
Thank you very much.
राधे राधे।
राधे राधे।
So if it is not working, if you want, you can go for oral presentation.
Oh, no. I already sent to you. It's reached or not?
Sir, which mail ID?
The email ID you send me that.
I have not received it yet.
You received it or not?
Not yet, sir. Yeah, I've just received, yeah. Kindly give me a minute.
Okay. Because here I am able to screen only tab, not any PDF or anything. That's permission is denied to me. I do not know why.
Sir, can you do one thing? You please open your slide first and then you go to the share option.
I did that. I read things. Yeah.
Okay.
I used to present regularly. So, but problem
Okay.
is that it's not telling me your access is denied.
All right. So kindly give us a minute. We are trying to download and we are trying to share it from this side. I think we will do it in 1 minute.
Yes, आ गए. Share screen is.
So you can start now.
Yeah, thank you very much for this. So can you do in full screen?
Yeah, screen, yeah. Next slide, please.
No, I want to do in full screen. So this is full screen?
I'll just check, sir. Kindly make it full screen. Yeah, and kindly change the slide. Yes, Alok sir, you can start.
Yeah. Sorry for the inconvenience. I do not know why it didn't work. So I know majority of here is a UG BSc students. I go through your website and check everything. So I'm planning to present the way so kids can understand. So I divided my talk in following headings you already see here. I will go introduction, which majority of you guys knows. And then I move toward the current therapy. What is the medicine available? What is the effect? What is the side effect? And after that, we move toward the future direction. And among that also, I will basically discuss on the last topic called activation, elimination or kick and kill approach, because the area which I am working last 4 to 5 years. So let's move. Next slide.
Next slide, please. Sunilji, kindly move to next page.
Yeah, so introduction. So everybody is among you guys knows what is the HIV and how it works, where it came. Still, I want to give you some basic inputs. So everybody knows AIDS and HIV. And people think here HIV is a virus and AIDS is a disease. Yeah, you are true. 90% it's true, but it's not 100%. Like you guys know, COVID. COVID is virus, COVID-19, and COVID is disease also. Same thing apply with here also, with HIV. HIV is a virus, and if you are having it, it's called HIV positive. And if you become HIV positive, it's become a disease. So what is the difference between HIV positive and AIDS? HIV positive is the starting stage of the disease, and AIDS is the basically kind of last stage of disease. So when it's become at the last stage, it's called AIDS. When you are in starting, people can easily tell HIV. So when you guys read in as a school, you will know this is the virus and that is the disease. But it's not true. Both are the disease, only stage difference is there. So currently there is no effective cure. We are sitting in 2021, but there is no cure is there. But yeah, we can live with HIV, long life, 10, 20, 30, 40, 50 years, based on how nicely you are having the medicine and how controlled you are taking care of it. So getting a effective HIV treatment, because who want to live with the disease throughout their lives? Everybody want to get rid of any kind of problem they have. They want to get rid of completely. They do not want to live with that whole life. So next slide. So we are living a very interesting time called COVID. As everybody knows, COVID-19 found in 19, and in 19 everybody's know COVID is there because everybody infected. But let's see the life where our ancestors live. HIV virus, which earlier called in SIV, semen immunodeficiency virus, which live in chimpanzee, and from chimpanzee it jumped to human. When it's jumped to human, it's called human immunodeficiency virus, and it's jumped not today or tomorrow. It's 1800, and it's jumped in 1800, and people come to know it's 1970. Why? The reason is same, because now we are living in a global society. Every day, million people travel, and if they find a million people every day, their growth and mutation will happen very fast, and it can affect very fast. In 1800, people, it's found in Central Africa and live there. Slowly, when people start moving from Africa to US, Europe, and India, then this move with them. In starting, like COVID, it's also not that much. You just now saw the COVID phase. First mutation, second and third. When you got change the strain, intensity will change. Same thing happened with HIV. 1970 it's found in USA, and 1986 is detected in India. It's basically detected in Madras, so 86. And at present, estimate 5.1 million infection. The data is really old, it's 2014, and because they really didn't bring the new census. So next slide. So how do I know if I have the HIV? The same, everybody's like all the viral disease having the same symptoms. So don't go with symptoms ever. Always go with test. If you go with test, you will come to know what is. And here also swab test is possible, but don't go with swab. Always go with blood test. And called nucleic acid test or NAT. It's very effective. Antigen and antibody test is also available, and it will give you results within 2 to 4 weeks based on the where you live, and sometimes you will get 1 to 2 days also based on the risk. Yeah, next slide. Next slide. Yeah. So just now I already discussed with you HIV and AIDS. So HIV have stages. So if you have primary infection, is called stage 1 acute HIV infection. So when people having the HIV in their blood and they are not treating it, it's very contagious.
Krishna?
You have flu-like symptoms in your body, and natural response is taking care of it slow, but yeah, but it is acute. It's better to take the medicine as early as possible and stay with that. Never go out of the medicine, and test only used last. Second stage, you can change the slide or I can go away also. So second stage is chronic HIV infection. This is slightly worse than the earlier one. It's based on people to people. Sometimes if your immunity is really good, you will not feel it. If your immunity is not good, then yeah, you will find everything's present to you in front of you. In a way, it's good also. In a way, it's not good. In a way, it's good because if you saw the symptoms, you can go get the disease and you go and test it and get the treatment. If you are not able to see that, people think so, they don't have anything and then live with it. And it's happened with many cases. So what happened, till you have the good CD cells, it's called CD4 T cells. This is a kind of T cells. You already know your immune response. Maximum people know here. If not, I can go in detail in question answer. So CD4 T cells is responsible for your immune system. When the viral load will increase, the load on the CD4 T cell will become burden. And that case, they are going to infected and their percentage going to be reduced drastically. So go in third. Next slide. And this stage is called AIDS. Why this is different for earlier? In this case, what happened? This CD4 cell, which is become normal in first and second step, they are fighting nicely with disease. In this stage, they are going to be drastically low, become 50% of your earlier immune system. So if they are hundreds, they reach it to 50. So they are 200 cells per microliter or mm, as based on what measurement you are using. So basically, if your immune system CD4 cells reduce less than 50%, you think now you reach the AIDS condition. And in this condition, people have to be very, very vigilant, because if you have first 2 stage, it will take 10 stages to reach the stage level 3, because till that 10 years, your immune system will fight. It's depend upon person to person, but I kept 10 years for the average people. So you think 10 years. But if you reach to a stage after 2 to 3 years, if you are not taking the disease, it will end your life. And as you all know, AIDS is not a disease. Only you will going to kill by the opportunistic infection like lung infection, cancer, any coronary diseases, anything. What will get the opportunity first? It will hit you first. So that's. So next step. So when should I start treatment? It's obvious, as early as possible. Go ask your, take the condition, health provider, and go on the medicine, and stay as long as it's required. Next step. Next slide. Next slide. So here is the main slide. So if you, how you are going to treat the infection if you do not know what is the life cycle. So this is the life cycle of the normal HIV virus. So if you see, they are, in this figure, they are divided in 8 parts. Please see the number, because when I am going to talk detail in the treatment, you will see this number. So HIV first with your CD4 T cells. This is called a kind of somebody knock your door. When you open the door, people will enter inside. Same thing happen here. It will bind with your receptor, and it's called CD4 receptor. And they are having also co-receptor along with that. When they bind this receptor, inside your body are called cytoplasm. As you know, this is the CD4 cells, and this is the nucleus of CD4 cells. When they enter your cytoplasm, virus generate a enzyme called reverse transcriptage. That enzyme belongs to virus. It's help virus to convert RNA to a DNA. When earlier, everybody is not here, virus always have only RNA, not DNA. So here they converted their RNA into DNA. After converting and getting this DNA is then fusing or integrating the chromosome, go inside the nucleus, and it will integrate. The enzyme which is responsible for this is the integrase enzyme. If you see the white portion and the this orange portion, the orange portion is belong to the virus, and white portion belong to the CD4 T cell, and this is called infected CD4 cells. Here, with the help of this CD4 nucleus chromosome, it's going to transcription. Transcription that make a many messenger RNA, many copies, and that copies then go outside the this nucleus in cytoplasm. Here they will do translation. Translation, everybody knows, making the protein, and that protein divided in small part and generate a assembly or budding. And after budding is basically a small part of the virus, and then they go outside of the nucleus and infected the new CD4 T cells. Next slide. Next slide. So yeah. So how you are going to treat that? So see the number here, 1, 2, 3, 4, 5. So this is the figure here. So this figure responsible for you for the maximum things. So if you block any phases when they insert with your transcription or reverse transcription, translation, if any phase you disrupt the virus, you are able to hold the growth of the virus in your body, and that's the drug is doing. As already you told, there is no any drug which is going to cure the completely, eradicate the HIV, but they give you the maximum lifetimes. Like already told, HIV give you 10 to 13 years, but if you go on medicine, you can easily live for 40 to 50 years. You have the kid also, and kid also will be perfectly normal. So now lot of ART is coming here. So maximum first antiretroviral drug came is called reverse transcriptage enzyme. So you know this reverse transcriptase, this is the third phase, which is responsible basically when they convert their messenger RNA into DNA. If you are able to stop here, this DNA will not go to nucleus. If it will not go to nucleus, it will not infect the CD4 T cells. And it will not infect the CD4 T cells. CD4 cells will be remain healthy and it will fight back, and you can easily get rid of the virus. Problem will happen when this CD4 cells become infected. And how it's infected, I already told. When that DNA insert in the nucleus and it's bind with your chromosome. You see that is already the pink portion. Now its complete CD4 T cells is basically blackmailed by the HIV virus. And this whole virus at this stage called proviral DNA. This is called proviral DNA. This is not complete DNA, viral DNA, not complete CD4 DNA. This is in between. And that is the main reason till now people not able to eradicate the AIDS, because it's bind your immune system. If you want to eradicate it, you have to kill all your immune system. If you kill your all immune system, anyhow you are going to die. So it's not possible you are going to do that. So yeah, you can move to next slide. Yeah, so this is this 6, 7, 8, 10, these all are the stage where this antiretroviral drug works. So I already told stage 3 is very important, but this stage 6 and 7 is also important, playing role. If they are inside your, if you know already you are infected with DNA, then what's happened? Now you do not have any chance. Still you want to live with that virus. And there this antiretroviral drug came, which is going to stop this messenger RNA converting into the small wording virus, or any drug which is going to clean this virus or kill that virus. So that virus which are sitting in your body, they will be killed. Still you are having HIV virus, but because this virus is not going to infect in new cells, so still your CD count value will be more, and you can live healthy lives. 90% case people do not know also you have HIV or not. You can go 30, 40, 50 years based on the medicine you are and what type of medicine and how you are taking it. Next slide. Let's throw it on HIV and see. This way in 1980s, you got your first antiretroviral drugs called azithromycin. These drugs are called nucleoside reverse transcriptage inhibitor. So I already told the stage 3. They are halting the stage 3. Sorry, I am not able to see the slide. Hello. So slide is gone.
I'll just check, sir. So kindly give us a minute. I'm just trying again, sir. I'm just trying to download again.
So can you able to see me? So
yeah, I'm sharing. Okay, it's okay now. Yeah.
So can you see that slide? I shared just now. Can you able to see my slide?
Yes, sir. It's visible.
Yeah. Okay.
I think it's working from your side.
Yeah. Just now I share. Yeah.
Yeah. Okay. Fine.
Basically, problem is there. You stop. You did not give me permission to share, but you can give me permission to access your mail. So I re-downloaded from what I sent it to you. I do not know why this happened. Yeah. It's okay. Now it's nice. I am able to see that. So So after accessing this drug, the new drug, this gave us the lifeline. So based on this random trial, we got the idea. So it is possible we can get the more drug related to same. So for that, in NIH, one group is established called the AIDS Clinical Trial Group, or ACTG, which is basically based on this drug. And based on this drug, they started the retrial and hired the patient and throw many stuff there. So let's get if they are able to get some new drugs or not. So after that drugs, that is also having very limited work capacity and having very large side effects. People want to get rid of that. So they already told that group already started the work based on that result. In 1995, they found if you take two drugs.
So your slide is not moving actually.
It's moved?
No, it's not moving. We are seeing only the first page of your presentation. Yeah, now it's moving.
Okay. That's the problem.
Every problem gives a new learning, so never mind.
No, basically I can access only my mail, nothing else. If I download the slide, still I can able to share it. So
now it's working. Yeah. Please continue.
Yeah. So antiretroviral drug discovery, and then in 1995, based on that disease, they found what happened if we used two drugs. So till 1995, one new drug is called dideoxycytidine. The structure is here. This is also a kind of same molecule called NRTI, nucleotide reverse transcriptage inhibitor. It will inhibit bacteria to enter in the nucleus. If you are able to inhibit bacteria into nucleus, it's really good. So basically, you are having very few infected cells and lot of healthy cells. The lot of healthy cells can get rid of infected cells very easily, and you become healthy long times. So, but that is also having their limits. It's slightly better, but it's not as. Then in 1996, there is one more drugs appear. It's called protease inhibitor. This protease inhibitor is responsible for cleaning the virus, which is already produced by the infected cells. So, the protease, the budding cells, when they a small cell will then come out of the cells and infect in others. That cell which cleave by the protease inhibitor. And along with that, they also come with a new idea called triple drug therapy. Nowadays, everybody knows HART, highly effective antiretroviral therapy. This is at present the best therapy people have. So they are using 3 kind of drug, 1 protease drug and 2 different kind of drugs based on which condition you are and what you are. So like I want to discuss in detail. If a single male is there, they don't have any problem. If the female is HIV positive and they are pregnant and they want to be their kids not HIV positive or normal kids, it's possible. You have to talk to doctor, they will go back and change your drugs based on that. They will give you protease inhibitor or NRTI based on your condition, which condition and where you are. So this is, due to this, you have to be lifetime stay on your drug. So after that, people thought it's 3 drugs and really hectic to take it. So is it possible you can take all drug in one? And that's happened. So nowadays what happens, they will change the percentage and all the same 3 drugs they give you in single tablet. So if you take 1 tablet daily, you will live with that long life. At present, there is more than 30 antiretroviral drugs is there. And in 2007, there is new drug came, 2013 also come, and they are different. They are called Integrase Inhibitor. So basically, it will not allow you to integrate in the virus. So that's really halt the disease. So is it halting disease is okay for us or anybody? Everybody wants to piece up their mind. For that, they need to get rid of AIDS virus or HIV completely. So what is the problem? Why people is not able to do so? The reason behind is there is this figure. If you see this first figure, here what they have virus, they have short-lived infected cells, and they have long-lived infected cells. What happens if you do the highly effective antiretroviral therapy? They treat virus plus short-lived infected cell, but they are not going to treat the long-lived infected cell. R is called latently infected cell. And that's the problem with HIV. That is the reason people is not able to get rid of it. And when they stop the medicine, it will come back. So you will let me know the time because I already instructed. I will go fast. If you give me 10 minutes, I will try to finish it. It's fine? So why they are not able to treat that? The reason is behind this. You see, this is the basically cells called resting cells. If any cell is in resting stage, no infection is going to do anything to that. They are not prone to the infection. And when they got the some enzyme, and that enzyme going to activate it. So in that activated case, they are going to be prone to the infector and the virus can infect it. So when the virus going to infect it, so in that case, when you are giving the heart medicine, in case B, so this cell will die easily. But what happens, some cell, they are very, very intelligent. They see they are the infected cell and they are going to die. So what happens, they will jump back. Jump back means they are going to be inactive. They make themselves inactive, sitting like peacefully. And that cell is called latently infected memory cell. Because they are infected, they have memory of infected cell, but now they are inactive. They are not giving any antigen or proteins to which this antiretroviral therapy detect and kill it. So this is called the latently infected memory cell. And this cell is the reason where till now nobody able to completely get rid of the virus, because this cell can sit your body 10 year, 12 year, 13 year without reacting. After 13 year, when they will find any activation, and they will come back and restart their production of virus and reinfect you. So if you want to really get rid of the disease, so you have to be completely get rid of that cells. So that is called future direction for HIV treatment. Till now we talk where we are. So now we are going to talk the future direction. There is many ways you are going to treat it. First, if you delay the drug, now people are taking 1 capsule or 2 capsule a day, or based on their condition. If you delay the drug, if people can take a month 1 capsule or 2 month 1 capsule, that is also a good thing. That give you less neutralizing antibody. You direct inject the antibody in the body, and that going to take care of say. Our third is the gene therapy, and there is therapeutic vaccine or vaccine. So I'm not going to talk about therapeutic vaccine and vaccine. Till now there is no any vaccine. Therapeutic vaccine is vaccine which is you can insert after you get the disease for the treatment region. My work is basically based in gene therapy approach and the last step 6 called activation and elimination model. So I will discuss in detail gene therapy. You modify the cell, CD cell, make it to resistant, or give them something like HIV specific T cell receptor, and then insert in your body. When they will go body, then check where this HIV cell is there, and they will kill it. And they can kill in any position. You can send in the target that position, or target RNA, or inhibition position, or any place you can target. You will deliver there. Only you have to read proper delivery place. You have to search nice delivery way to deliver them, and they will take care of it. So this is my area. It's called kick and kill approach. This is, I already told, if you are get rid of this latent HIV provirus, then you can get rid of HIV. So you can eradicate HIV completely from Earth if you are able to get rid of that latent virus. Because you know, antiretroviral therapy, they can kill the virus, but they are not able to kill that latent HIV reservoirs. If they are going to kill that, they are going to kill your immune system and that things. So how you are going to take care of that? All that there is many way. I basically work on the protein kinase C modulators. A small molecule, like this is chemistry symposium, so like organic molecule which help you to activate protein kinase C modulator, which is giving you expression, oh, there is something there, like antigen expression, activate it. And when it will activate, then you can kill him by any way. Like you can kill by natural killer cells or antiretroviral therapy, yeah, viral cytopathic effects, anyway, it's up to you. What I am doing, I am using protein kinase C and a small molecule for the activation, and then I use later natural killer cells, NK cell, to kill them. So that's get rid of the HIV. So there is a lot of natural products is there, which is basically approved from 1981 to 2020. They all are not the natural products. Basically, they are the modified one. So they are isolated from the natural products, but they modified and make it kind of drugs. So like we have less time, so we are not going in details. So that is the molecule which I am working. It's called Briastatin 1, isolated from this plant called Bagulana retira. And they already completed the first stage, phase 1 of clinical trial of HIV/AIDS eradication. They are also treated in cancer immunotherapy, Alzheimer, and multiple sclerosis. So they are in different phases. So as for like, I'm a chemist, I did my PhD in chemistry. So I have to make this molecule, because when they isolated NIH and give us molecule, they isolated from Bagulana retina. From 3 African elephant, they got that 1 grams of these molecules. So there is lot of waste. So there is supply and demand option. They do not have much biostatin which can really treat in, we can try in patient. For that purpose, I already told they work like a protein kinase C modulator. So protein kinase C, they have many ways to do that, but I select protein kinase C. So I'm not going to discuss chemistry here because maximum people are the BSc and they do not want to go with complete organic synthesis. So there is very detailed mechanism which we already published in 2017. So you can go ahead and check it. So we already published this molecule and then we modify it. So in our next slide, you are going to write biostatin 1 and SUW133. SUW133 is same molecule except they do not have this methyl group here at C26. So we synthesized this molecule and then we started over the tracking here. So this is the results what we recently published. So you see here, this is humanized mice. As you know, humanized mice, if you add in mice any human substance, any organ or any bacteria. So here we have the humanized mice and we infected with HIV, this mice. And that HIV is barcoded. Why barcoded? So if they travel in body, we can see where it is and how they are working, and then we can completely control it. So what happened when we infected this HIV, you will see diverse barcode here. Different kind of barcode means different kind of bacteria, viruses is there. And then when we give that the antiretroviral therapy, HART or maximum people are antiretroviral therapy, this reservoir decreased a lot. Completely kind of decrease, reduce a less size like here. Only if 10 is there, you will become 2 or 3. 8 is disappear. The time you stop ART, they will completely rebound and they will reach in the earlier stage where you started the medicine. So what we do? Then we are added LRA. LRA is the same organic molecule which we synthesized like a drug molecule and giving that which ART. So this molecule, what will do? They will activate that latency cell and then ART will kill that. In that case, you have very few reservoir left. If you have very few reservoir left, so it will delay the rebounds. So if you do the 1-day medicine, now it will give you 1 week, 10 days or 20 days based on here. If you see, so if you take the medicine, it will give you around 30 days. So if you take LRA, then this drugs, so 30 days, you do not have the much reservoir for your drugs. So basically it will delay the dose. So the day and the pill you are now taking, now you can take the pill which is give you long life. This one pill now, one after a month. But it is only delay. It is not eradication of HIV. Then you guys ask why you are talking about eradication then. So that is the next question here. So what happened if you, I told SUW133, this is only going to reduce delay the disease, but it's not going to cure it. But what happened if you put something highly effective treatment, like here in our case, we use NK cell. In future, we are also planning to do the different kind of antiretroviral therapy, but here we use NK cell. So you will see the 3 graph here. Let's see here. If you use only NK cell, so after some times it will rebound. It will only delay after 8 weeks, and it will rebound. All 5 mouse. 5 means 5 is this is the 5 humanized mouse. All 5 will rebounds. If you clean with only my molecules, that is also only decrease the time. Like last slide we already discussed. Only they will delay your system, but it will rebound after sometimes 6 to 8 weeks. It will come back sometimes 2 weeks, sometimes 4 weeks. Different mice in different time it will show. It's always based on the bodies. So when we treated with both, latency reversal agent like SU-W133 plus NK cell, then here you will see there is among 10, 4 did not rebound, only 6 rebound, and that is also very limited. So that's the way is here. If you activate that cell and then you able to kill it, you are able to completely eradicate the HIV. So already told phase 1 clinical trial is done. Still we are waiting for the phase 2. It will take some time, but hope so in recent future we will see it. So last time we saw there is different work. One recent here is there, Moderna. Everybody is taking the COVID vaccine. Moderna 2020 in January only launched the vaccination phase 1 trial for the mRNA vaccines. That's also in phase 1 critical trial. So everybody finger crossed, if everything is good, you will have the drugs very soon in future. Thank you very much. I took your time due to technical problem. I'm happy to take any question you want.
Thank you very much, sir.
Sorry.
Now
Quickly.
now is the time to ask any question. If anyone has got any question, they are welcome to ask.
Yeah.
So I have a question.
Yes, Palakshi, you can ask. You can send the question in chat box. I'll ask on your behalf because there is some disturbance from your side. In the meantime, sir, we have got many questions from students of Navyuga Kanya Mahavidyalaya. So, like, बच्चे ये जानना चाहते हैं कि how did you start your journey from India to US? जो basic question बच्चे जानना चाहते हैं कि कैसे आप वहाँ पहुँचे? What were your aspirations? How did you achieve all that?
So basically, I also like same. I belong to Varanasi. Yeah. So I am from UP. So from Banaras, Varanasi. So I did my BSc MSc there. Then I thought, like everybody, what you want to do. So I qualified NET, did my PhD, IT Kanpur, given many places, interviews. So basically I did my intern in National Chemical Laboratory, Pune for a year. Then I qualified JRF, GATE, then IT Kanpur, finished my PhD there. And then I moved to University of Texas, Dallas, basically Southwestern Medical Center. There also I work, basically my whole work in PhD is chemistry. And then I moved towards biology. And there also I work on infectious diseases like COVID, HIV, they all are the part of infectious diseases. And from there I moved to Stanford and continue journey is going on. Still I am here as a staff scientist. Nobody knows where I will go to other.
So Palakshi was asking a question that was interrupted in between. She's asking how more effective are natural drugs in comparison to synthetic drugs for the treatment of HIV?
See, natural drugs means natural products, I think they are talking. So see, natural product is not designed to treat the human. Natural product isolated as a byproducts in nature. And now you are the human, we are the human. Our expectation and need are completely different from the tree or the plants or the bacteria with whom this natural product is isolated. So we have to modify them completely based on our response and needs. So if I am able to understand their question, they are talking about natural products and the medicine. So you have to go away because natural products having a lot of side effects also. You have to play with that also.
Okay. I think Sanjay sir is also want to ask any question. Sanjay sir.
Yeah, I was thinking about your targeting of PKC. Is it isoform specific or general PKC?
No, we did isoform specific also.
So how specific? Which isoform?
So basically, we did delta.
Delta?
So, yeah, still we are struggling to complete isolation like novel and conventional. So we have the selection between novel to conventional, but complete selectivity if you want alpha, beta, gamma, delta, still we are not able to get 100% selectivity.
Okay.
Yeah.
Good too. Very nice talk, Alok. By the way, I'm also from BHU chemistry.
Yeah.
Any other question, please? You're welcome to ask. All right. Thank you, Alokji. Thank you so much for being there for us. And we are
sorry
really
for this inconvenience.
No,
I do not know why, because I use always a lot of technology daily, but this time
see, this is not in anyone's hand. In fact, it might be a problem from our side as well, though we have tried our best to rectify it as soon as possible. But anyways, that does not make any difference. So I'm really sorry for that. This is the time for the vote of thanks now, because I think we have completed in time. So we are not running late. So this is the time to vote of thanks, to propose vote of thanks. So I'm feeling privileged and honored to get.
I presented very fast. That should not be there.
I understand, sir. But actually, it was very simple and understandable. Actually, the thing which you deliver, that should go to our children. That is the most important thing. And you fulfill all that criteria. Thank you so much. And later on, if we have any queries, students can always connect with you.
Yeah, always. Yeah.
Okay, thank you.
Thank you.
I would like to thank all the delegates and all those who are present here, who blessed us with their presence. I would not be taking much of time because we are running 15 minutes late. On behalf of 1 p.m. And the second session would be conducted by Dr. Pratima Ghosh. So thank you so much. Now let's meet at 1 p.m. We are not leaving the meeting. We are all here. And you all can get back at 1 p.m. Thank you.
afternoon, sir. Then I'll start the session now.
Yeah, absolutely. We still have a minute, I believe. You had taken a 10 minutes break, so we can wait for.
Good afternoon, sir.
I'll just start in 2 minutes.
Yeah, good afternoon. Good afternoon. Good afternoon, Mrs. Pandey. How is it going since morning?
Very well. Very well.
Yes. Great.
So I'm Dr. Neha and she's Dr. Pratima Pandey. She's the session coordinator this time.
Yeah, Dr. Pratima Ghosh, I believe.
Yes, sir.
Yeah, I'm Dr. Ghosh. I think Neha just got fumbled up there.
Yeah, I know. Relax, Neha.
I said Dr. Pratima.
Yeah, maybe I heard Dr. Pratima Pandey. That's okay. Neha, I don't have any presentation as such, so I'll just be talking maybe.
No issues, sir. Not at all.
Yeah.
We are very comfortable. Like with direct interaction, that is always better.
Yeah, absolutely. And are we having it interactive, or you want me to first say whatever I want to say and then maybe have questions later?
Sir, you will be delivering your lecture first.
Okay.
So I shall be starting.
I hope so, yeah.
Yeah, okay.
Namami Shamisham Nirvana Rupam Vibhum Vyapkam Brahma Veda Swarupam Nijam Nirgunam Nirvikalpam Niriham Chidakasham Akash Vasam Bhaje Aham. We started the session in the morning with the Tanda Shrot. Now I am starting the session 2 of our webinar with the Shiv Vandana which Sri Ram did, and just starting this auspicious occasion of on this Shivratri with welcoming our eminent guests to the session 2 of this our webinar. A very good afternoon and a very warm welcome to one and all present in the webinar of session 2 of this webinar. I take this opportunity to welcome Dr. Anand Pandey and Dr. Ojha and Dr. Seema Joshi in the second session of this webinar. To introduce Dr. Anand Pandey, I would like to say that he is a BE Honours in Chemical and MSc Honours in Chemistry. Mr. Anand Pandey is President and Chief of Operations in Jubilant Ingrevia Limited, covering 5 manufacturing units in India. In addition, he is leading manufacturing operations of Jubilant Generics Limited as well. Jubilant is a large integrated company manufacturing a wide range of chemical and pharmaceutical products. He has been instrumental in bringing up new technologies, developing managing projects, and developing new business opportunities. Prior to joining Jubilant, Anand worked for Atul Limited, Aditya Birla Group, in different geographies and business like carbon black in India and Egypt, and vegetable oils manufacturing in Malaysia and Philippines, as well as Continental Carbon India Limited. He worked as CCL operations. He has total work experience of 35 years. We are indeed very privileged to listen to you, sir. Over to Anand.
Thank you, Dr. Ghosh. Thank you so much for that lovely introduction. I would have loved to be a doctor, but unfortunately, I am not. I am just a chemical engineer. But it always feels good when somebody addresses you as a doctor. So thank you so much for that. Okay.
So your experience has made you a doctor.
Okay. Thank you so much. Thank you. So before I get on to the specific subject on sustainability and environment, first of all, thank you very much, Dr. Neha, Dr. Pratima, and everybody who's around for having me here in this session. A very pertinent topic, International Conference on Chemistry for Health, Hygiene and Environment. This is a topic which is so critical today with all that's going on around the world. And I being a chemistry postgraduate myself, have some sort of an affinity for such topics. So before I get on to environment and sustainability, which is critical these days, let me talk a bit about chemistry as such and its role in these fields of sustainability and environment. So as we know, chemistry is the biggest invention of mankind. It has multiple times rewritten history. It's ever evolving. I mean, hundreds of thousands of years back, chemistry started, and it has made possible the food we eat, the clothes we wear, gadgets we use every day, satellites sent to space and all. And many of you would be aware that the role of chemistry in all these, right from finding the first element to everything else. Now, the current challenge that we have as far as so chemistry has given us a lot of great new products, a great new way of living. In plastics, perhaps was one of the biggest inventions of chemistry. What has happened over the years is that the unabated use of this knowledge has somehow led us to a situation where we are in a dire strait today. So there has been, we are seeing major challenges related to pollution, major challenges related to the soil contamination, land contamination, rivers, seas getting polluted, drinking water getting polluted. And that brings us to a very, very critical and important area that something needs to be done about this. And that's where the world had moved towards talking about sustainability. So typically when we talk about sustainability, what is sustainability? It's basically, as we say, it is meeting the needs of the present generation without compromising the ability of the future generations to meet their own needs. So as we know, the resources which are available in the world are finite. Most of the resources are finite. Many of the resources are unlimited, like sun is unlimited. But many of the resources like fuel, the fossil fuels, which we burn to meet our fuel needs, to run our automobiles, are all right. And the way we are consuming these is definitely creating a major challenge for the environment. There are so many diseases which are therefore a result of all this uncontrolled use of fossil fuels, a very unsustainable lifestyle that we are all unfortunately leading at this point in time. So it is becoming so critical for us to do something about it. And the people sitting here, the chemistry students, the research scientists, etc., have a huge role to play. And therefore, this topic is becoming so critical and so important. It's also very hard to see that there are 13 sub-themes of the conference, which itself means talks about the vastness of the subject of chemistry. So how effectively we are impacting the sustainability and environment is very clear to all of us. We consume plastics and a whole lot of other things. Our industries consume a lot of resources, and these are finite resources which are limited in the world. So obviously, the focus therefore is on how do we generate newer and newer technologies, newer and newer processes, so that we are able to run in a sustainable manner. The world runs in a sustainable manner, so that the future generations also don't curse us. We don't leave a legacy that our predecessors enough, and today we are in a dire state. we already are. So, and a lot of this has happened in the last almost 30, 40 years because of the kind of advancement which has happened. And everything that is happening is consuming these resources, is creating pollution, is depleting the finite resources which are available. So it is very, very important for us to do something about it. Now, some of the areas where perhaps we can play a role, or the chemists can play a role, is in terms of developments which consume minimum resource. So if we look at industries, if we look at any product which is produced, it is produced with some, in all probabilities, except for physical processes, more or less, it is all chemical reactions which are involved. Every chemical reaction is consuming resources. The raw materials are getting converted into products, but the efficiency of the reactions, when we talk about how much of raw material is getting converted into product and how much of that is converting waste, which is finally finding its way into some body, either into the soil or water bodies or whatever, is the role where a major role is to be played by the chemists and the scientists to develop processes whereby we conserve maximum resources and generate maximum products. We can't say that the development should stop. So the development has to continue. The world has to grow. So we can't go back to the Stone Age where we don't consume any resources. The area where we perhaps need to work very, very seriously is on how do we conserve this. So typically some of the technologies or some of the concepts which are used in this world are, people call it 5 R's, people call it 3 R's. 5 R's is typically refuse, which is say no to items which are of single-use plastics, for example. I mean, this is a responsibility I think all of us have sitting here to say no to single-use plastics, because these are finding its way into garbage, into sea. Fishes are dying, aquatic life is dying, and there's a whole lot of pollution because these don't degrade. I mean, as chemists, you all know that plastics don't degrade easily. So first R which we talk about is obviously, so say no to single-use plastics. The other thing is reduce. Can we reduce the amount of waste we create? And this starts from home. This starts from our home. Many a times we leave food in our table, on our plates, and that goes as a waste. So right from our houses, into the industry, if we get into that, there is so much which can be done to reduce consumption of resources. We consume only that much which is giving us a good result. Again, chemistry has a big role to play here because, as I said earlier, chemical reaction is a matter of yield. From 1 mole of a particular product, 2 moles of product can be generated, then ideally we should be generating 2 moles, that's 100% conversion. But that doesn't happen. The world doesn't, because there are some side products, byproducts, waste which get generated. And that's where the role of chemists like us is that how do we make process efficient so that the efficient use of utilization of the resources is there. So this is the second R, which is reduce. The third R is reuse, which is when we talk about reuse, we are talking about say no to disposables and yes to reusables. So there are a whole lot of utensils. We all of us have get-togethers, parties at our place. Now hygiene is also important, so you prefer to use disposable glasses, disposable plates, etc., etc. But then this is also creating a lot of pollution. There are agencies who have come out these days who will supply clean stainless steel utensils for your parties, get-togethers, and they'll have it collected at a cost, obviously. Now, this is one probable opportunity, or there are many other opportunities like that where nowadays people have developed reusable plates or reusable, which doesn't get disposed and create environment pollution. So that's the third R. The fourth one is the recycle. So typically, when we talk about recycle, we talk about a material which is otherwise going to be becoming waste. Do we So this is again a role of the chemists, where we are talking about creating wealth from waste. So in industry, typically, if you see in our houses in older days, our grandmothers, mothers, and I'm sure many of you would also be doing this, that whatever is left over in terms of cloths and all, there are mats which are made at home, asan bana lete hain, kai sari cheezein. So, so many other things. Otherwise, this would all go into waste. We need to extrapolate the same logic into industry, into manufacturing of products, where whatever waste is getting generated, can we look at technologies, can we look at ways through which we can convert some of these waste material into value-added products. So that's what I mean when I say recycle. And the last part is rot. Fifth R is we many times throw our kitchen waste, etc. Now, many large societies, or I think many housing complexes, even in Lucknow perhaps, would have set up composting plants in their areas where the kitchen waste is also segregated and converted into compost, vermicompost, or any kind of compost which can then be used for growing vegetables, growing fruits, etc., etc. So many ways. But the two big areas where chemists and chemistry can play a big role is on the reduce, where we are talking about how do we improve the yields of our processes, and recycle, where we are talking about generating wealth from waste. So these are two areas I would say that where and industry has done a lot of work in this. Industry has done work, and I'll be absolutely upfront about it, more so because there's a value they see in this. Because you are getting a raw material which can create a product or something by which you can get value out of it. But it's a blessing in disguise. While it's a win-win situation for the industry as well as for the world, that you are not disposing of waste like this. There are so many concepts which world is working on, is in terms of, especially if I talk about manufacturing to the young chemists here. There are earlier, the concepts of cradle to grave, cradle to cradle, and now people are talking about circular economy. So circular economy, typically when one talks about circular economy, effectively we are talking about no waste. So whatever is waste gets recycled back, reused. So effectively there is no waste generated into the process. Now there are many companies. IKEA, for example, a large furniture manufacturer, has adopted circular economy. So they have said that we will not generate any waste. So they make furnitures, but whatever is the waste which is created, they make further furniture out of it. Some other furnitures, zero waste, absolutely zero. So that's what we are talking about when we talk about efficient utilization of the resources, etc. The environment pollution, emissions, is another big area. So many diseases are happening these days, we all know, which are airborne, pandemics are taking place. And here again, there is a big role we have to play in terms of conservation. So when we spoke about the finite resources of maybe fossil fuels and all, the world is now moving towards renewable forms of energy. So can we do something about solar power, wind power? India as a country has taken a target of converting all its power. 50% of the power will come from the renewables, is the mandate by 2030. Now this is a tough ask, and all of us have to play a role there. So industry is also doing its bit, because environment pollution is creating a major challenge. There are GHG emissions which are taking place, greenhouse gases I'm talking about, carbon dioxide, so many chlorofluorocarbons, etc., which is again automobile related challenges. So there is a lot of work which needs to be done and is being done on. Companies are now talking about net zero. So net zero, typically one talks about whatever carbon dioxide we are generating, the same amount of carbon dioxide we consume. So net zero GHG emissions. Now it's a difficult thing. There are technological issues. There are sort of cost-related issues, because if you have to implement some of these things in the industry, it has to be viable also. So no industry is there for doing a charity. So at the end of the day, the technology has to be viable. Again, chemists have a big role to play. How do we generate or develop processes which are efficient in nature, whereby we can, conserve these resources? And the waste generation is also happening because of the, the kind of changes which are happening in the world these days. If you see the pandemic time, the medicines for COVID-19 were developed within a very, very short time. Some of them were not even tested fully. Now, during this development process, and they have a finite life. The remdesivir, for example, was one such drug, which does generate waste. Now, by the time you develop a process for treating the waste, the medicine itself has gone out of market or would go out of market. So there is a huge challenge, and that's why the role of chemists becomes so much more important, that we do things at such a fast pace. So agility is becoming the buzzword these days, and I'm sure Navyuga Mahavidyalaya is developing and training its students to, and some of you, I'm sure, would be working in these areas for sustainability and environment, because this is definitely an area where chemistry can play a major role. And we know chemistry is gender neutral. So most of the areas you have either dominated by male or females, but chemistry is an area where anybody has contributed a lot of so it is gender neutral, and so there's a huge scope which we can all work on together. I would take a pause here. If there are any questions, we can take those. But thank you so much for giving this opportunity, and really do appreciate. Yes, so there is a question here. Biofuels are new kind of areas, and Deepthi had asked this question. I think it's a very pertinent question, Deepthi. The biofuels are an area which is definitely growing. India has mandated use of bioethanol in ethanol blending, and for which also there are greener technologies which are coming to develop ethanol. So I mean, you have to manufacture ethanol to get bioethanol, and India has taken a target of 20% blending of ethanol in petrol. We also have a target for blending biodiesel in diesel, but the petrol targets are stiff, and for which a huge amount of bioethanol is required. Now, bioethanol typically in India is produced from green raw material, which is molasses. The molasses comes from sugar industry. So there is always a balance between sugar and alcohol, because the food versus fuel debate continues all the time. But yes, biofuels is definitely an area which is growing. Even in industry, many people are evaluating and are doing, instead of burning fossil fuels, using biomass, which is available, which is a waste. So this is definitely an area which is growing and will grow, has to grow. Yeah.
Any other questions? Any other questions?
Sir, some of the students are asking a question, the same question they're asking. Since you are the president and chief of operations of such a big, renowned organization, so they want to know, how did you start your journey? From where did you start? And what were your aspirations? How did you achieve such a great success in your life? So kindly motivate them for this kind of career they can opt for future.
Okay, so there is no magic wand here. I mean that you think of something and it happens. I mean, it's so I did my engineering way back. I'm sure many of you would not have been born also. I passed out in 1985 from Birla Institute of Technology and Science, Pilani, and after that I got into industry, and then it's all hard work. I mean, effectively, it's all hard work, and I'm sure if you all decide that you have to achieve something, you would achieve, as long as you are focused on your approach. There will be opportunities which will come your way. You have to weigh very carefully what what needs to be done and where you would like to take those opportunities. So it's rather simple as such. Just grab your opportunities as and when they come. Keep looking for opportunities and keep working hard.
Sir, can you? I take the opportunity to ask a question, sir.
Yeah.
What are the economic viabilities in Uttar Pradesh itself in the area of biofuel generation?
Okay, so Dr. Ghosh, I come from an industry which is already into biofuel. So we have a large plant in a place called Gajraula, which is about 100 kilometers from Delhi, near Muradabad, before Muradabad, if any of you have heard that. So this company was earlier known as VAM Organic Chemicals, and we manufacture bioethanol, we produce bioethanol. And in terms of opportunities, if you say, there is a huge opportunity because there's a huge shortage of ethanol for blending in petrol. And that's why each company, every sugar mill, distillery is looking to set up ethanol plants. Now, ethanol primarily, if you look at bioethanol, it is produced either predominantly from molasses, which is a sugar byproduct, but that molasses is limited because sugarcane is limited. So the alternate alternate raw materials which are being used these days are manufacturing ethanol from grain. Primarily, the process, if we look at, this is fermentation of sugars into alcohol, and then you distill that alcohol and purify it. So typically, grains have got starch. Starch gets converted into sugar, and then sugar is then fermented to produce alcohol. So grain ethanol plants are coming up, a lot of them in the state of UP. The only challenge is on the availability of the waste grain, because because again that there's a debate between food versus fuel, because you use broken rice, you use broken grains for this, maize, etc. Now this goes for food application also. So primarily whatever is after the food application is going for ethanol, and a lot of companies, including ourselves, we are also setting up a large 300,000 liters per day of alcohol bioethanol plant at Gajraula, and there are many companies like ours who are looking to set this up. So there is a huge opportunity there.
Thank you, Sir Pandey. I would like any other questions if they are needed to be put up. Anybody else who? Okay. Thank you very much, Mr. Pandey. I would like to hand over the mic to Mrs. Seema Sarkar, Dr. Mrs. Seema Sarkar, to invite our next guest, Dr. Ojha.
So thank you so much.
Over to Dr. Sarkar.
Thank you, Pratima. And it's an honor and privilege to find Dr. Anand Pandey between us and Dr. Sanjeev Kumar Ojha also. And it's a very, very honor and privilege for me to introduce such a great personality. On this auspicious occasion of Mahashivratri, I first of all pray for the world peace, because the situations are really very, 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 very painful. So let's pray for the world peace first, and then let me introduce you Dr. Sanjeev Kumar Ojha. He is an Ayurvedic scholar and fellow of Society of Ethnobotany, working primarily on herbal Ayurvedic formulations, validation of traditional knowledge, developing novel compositions, formulations for better health care, as well as income generation through plant wealth since 2001. He has worked on the Ayurvedic Pharmacopoeia of India, Part 2, Volume 1, 2, and 3. First edition formulations, published by Government of India, Ministry of Health and Family Welfare. Recently, he has developed anti-diabetic formulation. That is, I think that's very, very interesting and important thing for NBRMAP BGR34, marketed by MS Amil Pharmaceutical, conceptual designing of formulation and development of composition of NBIRASOF herbal soft drink, the technology transferred to MS 3D Nutrients, Ratlam, MP in 2019. That's a very recent development. Recipient of Technology Award for Life Sciences, that is in 2016, by Ministry of Science and Technology, that is Government of India, New Delhi, for development of anti-diabetic formulation and Aryabhat Award in 2016, and Shri Dhanvantri Samman 2018, and he is also a winner of B.N. Mehrotra Medal 2020. Having more than 40 research publications, 4 chapters in the book, guided 1 PhD and 8 MD IU, M-Pharma, having several patents on herbal formulations. Also, he has served as consulting Ayurveda physician in CSIR dispensary. So I request and I humbly request Dr. Ojha to come and just enlighten us by his lecture. Dr. Ojha.
नमस्कार सबको मेरा, और काफी दिनों से मेरे को यहाँ पे लेक्चर एक देना था। पूर्व में पहले भी बात हो चुकी थी, हिंदी इंग्लिश मिला के बात करूंगा, थोड़ा बहुत संस्कृत भी बोलूंगा क्योंकि ट्रेडिशनल नॉलेज के लिए बहुत सारी चीजें संस्कृत में भी हैं। और फर्स्ट टाइम आप लोग ऐसा सोचेंगे कि इतना क्वालिफाइड लोगों के बीच में इस ढंग से बात रखनी है मुझे कि वो बात जो ट्रेडिशनल सिस्टम की है उसका साइंटिफिक एस्पेक्ट आपके सामने आ पाए। तो 1 मिनट में अपनी बेटी की जरा सा हेल्प ले रहा हूँ। ये उनका लैपटॉप है, ये प्रेजेंटेशन है। हाँ,
पूरा स्क्रीन सेलेक्ट करिए,
ये वाला
चेक करिए।
1 मिनट है।
हाँ, हो गया। पीपीटी पीपीटी
कहाँ पे?
ऊपर ऊपर ऊपर
करो बेटा। मेरा प्रेजेंटेशन आप सबको दिख रहा है?
Yes, sir, it is visible.
Yes, sir.
In today's world, we know several types of disorders, disease are there, but in ancient time, our Indian sages has discovered a system of medicine wherein they focus more on preventive aspect rather than curative aspect. So based upon those, they have made certain things which everyone knows, and most of our household ladies and dadi, nani, ma, everyone knows. But unfortunately, modern people, they are ignoring it and they are thinking that as if it is slightly way back system, or just like we are seeing here, पुराना पंडिताऊ सिस्टम है, हम लोग तो मॉलिकुलर पे बात करते हैं सारी बातें, लेकिन आज भी वो सारी बातें जो हैं देखेंगे कि कितनी अक्षरशः सत्य हैं। वो सारी बातें मैं इनमें आपके सामने रखना चाहूँगा। ये विलियम ऑसलर की बात मैं करना चाहता हूँ, जिन्होंने कहा था कि वन ऑफ द फर्स्ट ड्यूटी ऑफ फिजिशियन इज टू एजुकेट द मासेस नॉट टू टेक मेडिसिन। और उन्होंने इसका उत्तर भी दिया। वाई जब उनसे पूछा गया तो उन्होंने कहा द पर्सन हु टेक्स मेडिसिन मस्ट रिकवर ट्वाइस, वन फ्रॉम द डिसीज एंड अदर वन फ्रॉम द मेडिसिन। अब ये समझना है कि अगर आप दवा खाते हैं तो एक बार तो आपको बीमारी से निकलेंगे और दूसरी बार उस दवा के साइड इफेक्ट से आपको निकलने में समय लगेगा। आज की डेट में बहुत सारी ऐसी दवाएं हैं जिनका नाम अगर मैं लूं तो आप पाएंगे कि उसमें निरंतर साइड इफेक्ट्स ज़्यादा हैं, और डिसीज को किल करना तो एक बात हो गई, लेकिन तमाम सारी उसमें प्रॉब्लम क्रिएटिंग सब्सटेंसेस और ज़्यादा हो जाता है। तो इसीलिए एक बाद में 14वीं-15वीं शताब्दी के आसपास पार्सेलियस एक वैज्ञानिक हुए, उनका ऑल सब्सटेंसेस आर पॉइजन, देयर इज नन दैट इज नॉट ए पॉइजन, द राइट डोज डिफरेंशिएट ए पॉइजन एंड ए रेमेडी। तो ये एक बड़ी इंपॉर्टेंट बात उन्होंने कही कि आज की डेट में अगर हम पानी भी ज़्यादा पीते हैं, नमक भी ज़्यादा खाते हैं, चीनी भी ज़्यादा खाते हैं या घी भी ज़्यादा खाते हैं, तो वो उसको कहता है डोज की डिपेंडेंसी। तो वो एक चीज़ है कि हमको कब कितना कैसे लेना है, उसी के हिसाब से वो तय करते हैं कि क्या चीज़ एक हमारे शरीर के लिए जहर है या कौन सी चीज़ एक हमारे शरीर के लिए दवा बन सकती है। एक बात और शुरू करने से पहले मैं बताऊंगा, दलाई लामा साहब की बात थी, तो दलाई लामा साहब ने कहा कि आदमी जो है अपनी सेहत गंवा करके पैसे कमाता है और फिर पैसे गंवा करके सेहत खरीदना चाहता है। जीता ऐसे है जैसे कभी मरेगा ही नहीं और मर ऐसे जाता है जैसे कभी जिया ही नहीं। तो वास्तव में वो ना पास्ट में रहता है न फ्यूचर में रह पाता है, मतलब प्रेजेंट को एंजॉय नहीं करता है और बस चिंता में या फिर वरीज में, इन्हीं सब में रह जाता है, और या गिल्ट में रह जाता है। पास्ट लाइफ होगी तो गिल्ट होगा और फ्यूचर रहेगा तो एंजाइटी रहेगी। तो ये एक चीज उन्होंने बहुत अच्छी कही कि हमें वर्तमान में जीने की आदत डालनी चाहिए और कैसे हम आगे स्वस्थ हैं इसकी बात करते हैं। तो ये आयुर्वेद में जो बात हुई वो ज्यादातर उन्होंने प्लांट को एक एज ए सोर्स ऑफ मेडिसिन माना और लगभग 2500-3000 हजार ईसा पूर्व में इसका प्रादुर्भाव हुआ। मुख्य रूप से ये प्रिवेंटिव मेडिसिन थी जो कि लाइफ स्टाइल डिसऑर्डर्स के लिए दी गई थी, और 80% ऑफ द वर्ल्ड पॉपुलेशन रिलाई ऑन ट्रेडिशनल हर्बल मेडिसिन फॉर प्राइमरी हेल्थ केयर। ये WHO का स्टेटमेंट है, स्टैंडर्ड स्टेटमेंट है, हर जगह हम लोग इसको प्रेजेंटेशन में यूज करते हैं। लेकिन आजकल इसका जो ट्रेडिशनल मेडिसिन का बात बढ़ी है वो हुआ है कि अटेंशन गिवन बाय द गवर्नमेंट और इंक्रीज साइड इफेक्ट्स है, तमाम सारे एडवर्स ड्रग रिएक्शंस है मॉडर्न मेडिसिन के कि तमाम सारी दवा है। अगर मैं नाम लूँ, डिक्लोफिनेक है, से किडनी फेल करती है। भी पता लगा कि कहीं पे कोई मरे हुए जानवर को वल्चर्स या चील खा रहे थे तो उन चील की डेथ हो रही थी बिकॉज ऑफ किडनी फेलियर। इसी तरीके से तमाम सारी और है बीमारी की, हमने डिस्प्रिन ले लिया तो गैस्ट्राइटिस हो गई। तमाम सारी दवाओं के नाम हमें लेना नहीं चाहिए, लेकिन जस्ट एक इंडिकेशन के रूप में मैं ले रहा हूँ। और दूसरी चीज, हाई कॉस्ट ऑफ द मॉडर्न मेडिसिन, ये बड़ा इंपॉर्टेंट एस्पेक्ट है। आयुर्वेद में बहुत ही सरल, सहज और सुलभ तरीके से इलाज की बात की गई है। तो ये जो है हमें इस पे ध्यान देता है। और रिसेंटली मिनिस्ट्री ऑफ आयुष ने अपनी एडवाइजरी भी जारी की थी इम्यूनिटी के लिए, तो ये थी इसमें बातें। तो व्हाट इज आयुर्वेदा? आयुर्वेदा की जो डेफिनेशन दी गई थी उसमें एक श्लोक है, हिताहितम सुखम आयुतस दुखाहितम। तो मूल रूप से जो इसका जिस्ट है वो मैं आपके सामने रखता हूँ कि 4 स्टेट्स ऑफ लाइफ के बारे में बात करता है, अप्रोप्रिएट एंड इनअप्रोप्रिएट, उचित अनुचित की बात में, फिर हैप्पी एंड सॉरोफुल कंडीशंस, एंड अलॉन्ग विद व्हाट इज गुड एंड बैड फॉर लॉन्जेविटी। और अंत में ये एक जीवन का मापदंड है, मेजरमेंट ऑफ लाइफ है। तो इसलिए आयुर्वेद जिंदगी के बारे में जो भी है वो सारी बात करता है। इट इज साइंस ऑफ लाइफ एंड आर्ट ऑफ लिविंग। तो ये सारी चीजें इसमें जिस ढंग से भी बताई गई हैं वो हमारे जीवन का विज्ञान ही इसको कहना उचित रहेगा। तो ये केमिस्ट्री की कॉन्फ्रेंस है, तो मैं इसका ऐसे ही संस्कृत में इसका उद्भव के बारे में सोच रहा था कि कैसे इसकी निरुक्ति केमिस्ट्री में अगर संस्कृत का कोई आदमी होता तो बोलता किम इदम शास्त्रम। केमिस्ट्री को अगर हम लोग बोलेंगे किम, ये मतलब तत्व का बोध कराने वाला ना, ये क्या है, तो किम इदम, ये क्या है, उसका शास्त्र है। तो अगर हम फोनेटिक्स एंड लिंग्विस्टिक के बारे में थोड़ा बहुत जानकारी रखते हो तो देखेंगे कि हम अगर किम इदम शास्त्रम को 2-4 बार बोलेंगे तो केमिस्ट्री ही शब्द जैसा मिलता जुलता फोनेशन आएगा या आवाज आएगी। तो ये संस्कृत में इसकी निरुक्ति हो सकती है। और आयुर्वेदिक केमिस्ट्री जो है वो 5 महाभूतों पर आधारित है, जिसमें मूल रूप से आकाश, वायु, अग्नि, जल और पृथ्वी है, और उन्हीं से फिर वात और पित्त और कफ की बात करते हुए आगे बढ़ते हैं कि ये जो वात पित्त कफ होते हैं ये बॉडी में मैनिफेस्टेशंस हैं, लाक्षणिक रूप से दिखाई देता है, और इन्हीं का हम इन्हीं तत्वों के हिसाब से इलाज भी करते हैं। जैसे मान लीजिए कहीं पे कैटाबॉलिक एक्टिविटी या डायनेमिक है, या वात बहुत चल है, मतलब कहीं भी हवा होगी तो मूवमेंट होगा, कहीं पे हीट होगी तो पित्त होगा, कहीं पे ठंडा या स्थूल चीज होगा तो कफ होगा। ये अलग अलग ढंग से उसके लक्षण प्रकट होते हैं, और फिर उन्हीं को हम लक्षणों को प्रकृति के उन पौधों में देखते हैं और देखते हैं बीमारी में जिन चीजों की कमी है उनको उस तरीके से ला करके साम्य अवस्था में लाना ही आयुर्वेद का इलाज होता है। तो कुछ और भी है, नास्तो विद्यते भावो ना भावो विद्यते, मतलब जो दुनिया में है उसी ढंग से हम लोग उसको आयुर्वेद की उत्पत्ति के बारे में बात करते हैं, और अव्यक्त से, और मतलब ये आयुर्वेदिक कॉन्सेप्ट है। और 5 महाभूत थ्योरी है। फिर ये भी कहते हैं नान औषधि भूतम जगत किंचित, दुनिया में कोई भी ऐसी चीज नहीं है जिसमें औषधि गुण ना हो। ये थोड़ा फिलोसफिकल है तो इसलिए मैं थोड़ा जल्दी इसको पढ़ा रहा हूँ लेकिन ये समझना पड़ेगा, बिना इसके समझे हम लोग कोई भी चीज को जान नहीं सकते हैं। व्हाट इज लाइफ एक्चुअली? तो वहां पे जहां डेफिनेशन दे रहा था लाइफ की, तो वहां पे बोला इट इज डिपेंड अपॉन शरीर, मन और आत्मा, बॉडी, माइंड एंड सोल। इन तीनों का एक कॉम्बिनेशन है जिसमें कि आप कोई भी चीज इसमें से डिस्ट्रॉय हो जाती है तो लाइफ खत्म हो, मतलब उसका कोई मतलब नहीं रह जाता। मान लीजिए बॉडी नहीं है तब तो दिक्कत ही है, माइंड खराब हो जाता है तो भी उसकी कंप्लीटनेस खराब हो जाती है, इसका दिमाग का है वो मतलब साइकोलॉजिकल डिसऑर्डर्स के साथ में है। आत्मा निकल गई तब तो बॉडी का क्या मतलब ही रह जाता है। तो ये जो तीनों हैं, बॉडी, माइंड एंड सोल, तो बॉडी को नर्चर करने के लिए आहार है, और माइंड को नर्चरिंग करता है निद्रा से, और सोल को नर्चरिंग करता है ब्रह्मचर्य। ब्रह्मचर्य माने क्या है? वो अपने विचारों पे नियंत्रण करना और तमाम सारी उन इच्छाओं पे नियंत्रण करना है कि जिसको अनड्यू एडवांटेज है, कि जिसमें हम दूसरे के ऊपर या कई तमाम सारी चीजें रखते हैं, तो वो उसके लिए इट इज नॉट डायरेक्टली लिंक टू द सेलिबेसी इटसेल्फ। तो वो एक चीज है। इसके अलावा तमाम सारे इसके कैरेक्टरिस्टिक ऑफ लिविंग बीइंग्स हैं जो कि हम लोग अपने साइंस में बायोलॉजी में पढ़ते ही हैं, होमियोस्टेसिस, ऑर्गेनाइजेशन, मेटाबॉलिज्म, ग्रोथ, एडॉप्शन। तो व्हाट एक्चुअली इट इज लाइफ? लाइफ इट इज ए प्रोसेस, नॉट ए प्योर सब्सटेंस। हम ये नहीं कह सकते कि सीधी चीज जिंदगी है। जब शरीर से प्राण निकल जाते हैं या आत्मा निकल जाती है तो एक मटेरियल सब्सटेंस रह जाता है जिसको हम कहते हैं ये मिट्टी है, ये शरीर है, या ये बॉडी रह गई, तो वो तत्व निकल जाता है जीवन। तो ये एक समझना पड़ेगा, इट इज नॉट ए प्योर सब्सटेंस। एक चरक संहिता का इंपॉर्टेंट श्लोक आपके सामने रख रहा हूँ। ये आयुर्वेदिक नॉर्म्स फॉर स्टैंडर्ड ड्रग के बारे में है, कि जो अभी मैंने ड्रग के बारे में थोड़ी सी बात की कि तमाम सारी चीजें होती हैं। हमारे चरक ऋषि ने उनकी इतनी महत्वपूर्ण और सूक्ष्म दृष्टि थी कि एक एक चीज उन्होंने स्पष्ट की। और थोड़ा सा मैं पढ़ता हूँ। अल्पमात्रम महावेगम बहुदोषहरम सुखम लघु पाकम सुखा स्वादनम पीड़नम व्याधिनाशनम अविकारी च व्यापत्तो न अतिग्लानि कर्चयत गंधवर्ण रसोत्पेतम विद्यात मात्रा औषधम। इसको मैंने पब्लिश भी किया है, आर्यवैद्यन करके एक जर्नल आता है, उसमें आयुर्वेदिक नॉर्म्स फॉर स्टैंडर्ड ड्रग करके मैंने पब्लिश भी किया। ये एफिकेशियस इन स्मॉल क्वांटिटी, अल्प मात्रा, महावेग, इट मस्ट बी हैव ग्रेट पोटेंसी, किल वेरियस डिसऑर्डर, बहुदोषहरम, सुखम है ना, प्रोवाइड रिलीफ, इजी टू डाइजेस्ट, लघु पाकम, सुखा स्वादन, मतलब टेस्ट गुड होना चाहिए, अच्छा टेस्ट का होना चाहिए, एनर्जाइजर होना चाहिए, डिसीज किलर होना चाहिए। बहुत इंपॉर्टेंट बात यहाँ पे ये लिखी है, अविकारी च व्यापत्तो न अति ग्लानि कर्चय, नाइदर साइड इफेक्ट एंड नॉर हार्मफुल इफेक्ट। तो ये आज की डेट में सबसे बड़ा एक प्रश्न चिन्ह उठ के खड़ा होता है तमाम सारी दवाओं के बारे में, कि जब हम कोई दवा देते हैं तो उसमें कुछ ना कुछ साइड इफेक्ट या हार्मफुल इफेक्ट रहते हैं। तो ये हमें देखना चाहिए कि औषधि में ये एकदम नहीं हो तो सबसे अच्छा है। इसका एक दो उदाहरण भी मैं आगे दे सकता हूँ। जब हम लोग जानेंगे, थोड़ा सा भी जानने लगेंगे, तो हम पाएंगे अपने जीवन में, दैनिक जीवन में बहुत सारी चीज का इस्तेमाल रोज कर रहे हैं, नित्य, और औषधि रूप में कर रहे हैं, और हमें मालूम भी नहीं है, और खाने के साथ भी कर रहे हैं, और हमारे वो शरीर को, प्रतिरक्षा प्रणाली को सुधारते हैं। ये एक हाइपोथेटिकल चार्ट है जो कि मैंने समझाने के लिए बनाया कि कैसे हम लोगों के पास ये जो काम करने के लिए ऊर्जा आती है वो कैसे आती है। सूर्य से ऊर्जा आती है, सन इज द अल्टीमेट सोर्स ऑफ एनर्जी। लेकिन इसका मतलब ये नहीं है कि सूरज और भी नहीं है। सूरज एक स्टार है, हम लोग एक ग्रह में रहते हैं, प्लेनेट पे, तो यहाँ पे देखते हैं पौधों में एनर्जी को ट्रैप करने की क्षमता है। क्लोरोफिल के माध्यम से वो एडीपी को एटीपी में कन्वर्ट करते हैं। फिर फर्दर उससे दो तरीके के चीजें बनती है, एक जिनको हम खा सकते हैं, एक जिनको हम नहीं खा पाते हैं। फूड होता है, एक फॉडर हो जाता है। फूड हम लोग खाते हैं, फॉडर जानवर खा लेते हैं, और गाय अगर खाती है तो फिर उससे दूध भी निकलता है। उससे तमाम सारी चीजें प्रोसेस होकर के अंत में घी भी बनता है, जिससे जलाने पे वो दिए जैसी रोशनी करता है। वो उसकी रोशनी वैसे ही लगभग होती है जैसे सूरज की रोशनी होती है। खैर, यहाँ पे एक चीज और मैं बताऊंगा कि ये एक तरीके से एक सेकेंडरी प्रोसेस हुआ, और ये मधुमक्खी भी है जो पौधों से, प्लांट सैप से लेकर के शहद बनाती है। आयुर्वेद में शहद, गाय का दूध, गाय का घी, इन सब का बहुत ही इस्तेमाल है जड़ी बूटियों के साथ में देने में, अनुपान में। तो ये हम इसीलिए बताना चाह रहे थे कि जो इस तरीके से ऊर्जा का द्वितीयक प्रयोग होता है वो जो है वो बहुत महत्वपूर्ण होता है। वास्तव में ऋग्वेद में ही कह दिया था, सूर्यो आत्मा जगत सुयश, सूर्य ही इस जगत की आत्मा है। इसका मतलब ये था कि हम सब में जो भी ऊर्जा का स्रोत है वो वहीं से है। रॉबर्ट एंड्रू मिलिकन को नोबेल प्राइज मिला 1923 में। उन्होंने कहा मैन विल नेवर हैव एनी अदर सोर्स ऑफ एनर्जी देन सन। तो फिलहाल हम लोगों के लिए सूरज ही ये अनंतिम ऊर्जा का स्रोत है। और इसमें एक बार तो लोगों के मन में जिज्ञासा है साइंस के बारे में, लोगों को रहती है। एक प्लांट दिखा रहा हूँ जिसमें ऊपर टमाटो लगा हुआ है, नीचे आलू लगा है, टोमेटो करके, कि हम क्या ऐसी कोई विधा विकसित कर लें कि उसमें उसकी पूर्ण रूप से क्षमता का इस्तेमाल कर पाए। सोलोनेसी फैमिली में एक ही पौधा है ऊपर नीचे इस तरीके से कि जिससे सूर्य की ऊर्जा का पूरा इस्तेमाल हो पाए। इसके साथ ही एक और बात ये आगे आती है जो मैंने आकाश, वायु, अग्नि, जल और पृथ्वी कही थी, तो उसमें ये सूर्य की ऊर्जा अग्नि का द्योतक है। थोड़ा सा दिमाग में रखेंगे साइड में उसको भी। और आगे चलते हैं पृथ्वी के रूप में। जब हम देखते हैं तमाम सारे न्यूट्रिशनल कंटेंट जो कि पौधों में जाते हैं और उनको हम जब सेवन करते हैं तो वो हमारे शरीर में आते हैं। तो यहाँ हम देखेंगे कि तमाम सारे जो न्यूट्रिशनल कंटेंट अगर सॉइल में नहीं है, आजकल जैसे डिप्लीशन हो जाता है, तो बॉडी में भी हमारे रिफ्लेक्ट करते हैं। अगर इनका फॉडर जानवर चलता है तो जानवर का जो प्रोडक्ट है दूध है उसमें भी वो कमी महसूस होती है। इसीलिए आजकल ऑर्गेनिक फूड इस सब की बात होती है। तो ये एक टोटल एनर्जी साइकिल है, वो समझना है, जिसमें कि हम उस एनर्जी को ही करेंसी के फॉर्म में देखते हैं और सारा काम करते हैं। तो व्हाट एक्चुअली आयुर्वेदा, जब हम इसको ऑब्जेक्टिविटी वर्सेस सब्जेक्टिविटी, एक हाइपोथेटिकल कर्व है, तो ये एक देखते हैं, ये हाइपरबोलिक कर्व मिलता है कि अगर मान लीजिए कोई एक ऑब्जेक्ट है, ये मान लें हम मोनालिसा देख रहे हैं, तो सम ब्यूटीफुल पर्सन होगा, मान लीजिए हम किसी को लेते हैं कि ऐश्वर्या राय है या कोई है, कौन है वो एक, हेलो मेरी आवाज आ रही है?
यस सर,
यस सर, यस सर, यस
जी सर, जी सर।
ठीक, मैं मेरे को लगा कि मैं अकेला ही बोलता जा रहा हूँ तो वन वे ट्रैफिक लग रहा था। खैर तो ये मान लीजिए कि ऐश्वर्या राय है, तो मेरे को लगा कि सोसाइटल में इनकी वैल्यू है, अमिताभ बच्चन की बहू है और सुंदर है, साइकोलॉजिकली साउंड माइंड की है, बायोलॉजिकली ये होमोसेपियंस में फीमेल स्पीसीज है। और केमिस्ट्री में क्या है, अल्टीमेटली इनकर ये जला दी जाती है और कुछ भी होता है, राख बनता है, तो ये हम लोग कार्बन, हाइड्रोजन और ऑक्सीजन देखते हैं, जो कि मॉडर्न साइंस कहता है कि हम लोग इस तरीके से हैं। तो पूरा जो हम लोगों का शरीर है वो कार्बन, हाइड्रोजन और ऑक्सीजन और उसके बीच बने हुए बॉन्डिंग से ही बना हुआ है। और यही आयुर्वेद का है कि फुल ऑफ एस्थेटिक सेंस है, हाईली साइंटिफिक है, कंसीडर साइकी एज डिसीज फैक्टर है, ग्रेट सोसाइटल एक्सेप्टेंस है, वैल्यूलेस है, इफेक्टिव है और कमर्शियल भी है। लियोनार्डो दा विंची का ही कथन है, आई थॉट आई वॉज लर्निंग टू लिव, आई वॉज ओनली लर्निंग टू डाई। तो ये हम लोगों को समझना है आगे कि हम जो आयुर्वेद को कैसे अपनाएं और हम मतलब मरने की बजाय जीना सीखें। ये एक केमिकल स्टोरी लाइन का एक पेपर है, उसका मैं रेफरेंस दिखाना चाहता हूँ, जिसमें एलिमेंट्स ऑफ लाइफ का कैलकुलेशन किया गया, ऑक्सीजन, कार्बन और हाइड्रोजन और नाइट्रोजन की। हम लोगों में 60 किलो के आदमी में कौन कितना परसेंटेज है, तो वो परसेंटाइल है और वजन के हिसाब से भी है कि कितना किलो उसमें ऑक्सीजन होगा, कितना कार्बन होगा, कितना हाइड्रोजन होगा। ये वास्तव में आई ओपनर है। हम लोग आज हिंदू, मुसलमान, सिख, ईसाई तमाम सारी बातों पे झगड़ा करते हैं। वास्तव में अगर हम केमिस्ट्री की निगाह से देखें तो सारा झगड़ा ही मिट जाएगा, है ना। हमारा सारा झगड़ा खत्म हो जाएगा। ऊंच नीच, जात पात, धर्म, देश, कास्टिज्म, है ना, कॉकेशियन और इंडियन और आर्यन और द्रविडियन, सारे किस्से खत्म हो जाएंगे। हम लोग केवल कार्बन, हाइड्रोजन और ऑक्सीजन ही है। तो इसी से मैं आगे चलता हूँ। ये एक आप लोगों ने केमिस्ट्री में देखा ही होगा और इसमें टेबल में देखेंगे तो ये कुछ बल्क एलिमेंट्स और ट्रेस एलिमेंट्स हैं जो मैंने हाईलाइट किए हैं, जो बॉडी में हम लोगों को रिक्वायर होते हैं। और इनमें तमाम सारी इनकी डेफिशिएंसी से तमाम सारे डिसऑर्डर्स होते हैं, कैल्शियम, फास्फोरस, सोडियम, पोटेशियम, आयरन, आयोडीन, क्लोरीन, इन सब की कमी से होने वाले तमाम सारे। इसके माने कि सिर्फ ऐसा ही नहीं है कि कार्बन, हाइड्रोजन, ऑक्सीजन ही रिस्पॉन्सिबल है, तमाम सारे ट्रेस एलिमेंट्स भी बॉडी के लिए बहुत एसेंशियल होते हैं। एक ये एक्सपेरिमेंट आप लोगों ने देखा होगा, स्टेनले, मिलर और यूरे का एक्सपेरिमेंट था। तो इसमें ये था कि एक फ्लास्क में वैक्यूम रहता है और उसमें इलेक्ट्रोड लगा हुआ है, कुछ गैसेस, वैक्यूम नहीं, स्पेस है और गैसेस है, हाँ, और उसमें स्पार्क क्रिएट करते हैं तो वाटर और कुछ अमीनो एसिड्स बनते हैं। तो ये आयुर्वेद की दृष्टि से देखें तो ये जो जगह दिखा रहा है ये स्पेस है, आकाश है, और इसमें ये स्पार्क है, ये अग्नि है, और जो गैसेस हैं वो वायु है, और जो पानी बन रहा है नीचे तो ये डेफिनेटली जल है, और इसमें जो सब्सटेंस नीचे बनते हैं अमीनो एसिड्स वगैरह, तो उनमें मास गुरुत्व भी है, नीचे तलहटी में बैठते हैं। इसके मतलब है कि उसमें जिस चीज में गुरुत्व है तो पृथ्वी महाभूत का वो गुण है। तो वो आकाश, वायु, अग्नि, जल और पृथ्वी की दृष्टि से मैंने इसको देखने की कोशिश की, वो मैं इसके सामने रखा आपकी। एक ये गार्डन ऑफ ईडन है, इसमें बहुत सारी चीजें दिखाई दे रही हैं कि घोड़ा है, चिड़िया है, बर्ड है, तो ये सारा एक ही तरीके का बेसिक स्ट्रक्चरल सेल से बना हुआ है, सेम काइंड ऑफ मैक्रोमोलिक्यूल, डीएनए, आरएनए, प्रोटीन, सारी चीजें। लेकिन क्या है, उसके बावजूद सब अलग अलग हैं, सारे पक्षी अलग अलग हैं, सारा कुछ सेम सिमिलर टाइप का जेनेटिक कोड मटेरियल होगा, लेकिन उनका डाइवर्सिटी अलग अलग होगी। इसलिए ये लिविंग ऑर्गेनिज्म ये दिखाने के लिए इसको दिखाया। एक अभी जो बात कर रहे थे उधर पीछे, तमाम सारे लोग अभी देखते हैं उधर इस फोटो में कि खाने की दिक्कत होती है, कुछ लोग अंडर न्यूट्रिशन है, तो उसमें दोनों तरीके के लोग हैं। कुछ लोग जो हाई एनर्जी वाली चीजें खा तो रहे हैं, लेकिन उसमें प्रोटीन की डेफिशिएंसी है या आयरन की डेफिशिएंसी है। कुछ लोग ऐसे हैं जो ज्यादा खा करके मोटे भी हैं। तो दोनों तरीके के लोग जो हैं वो प्रॉब्लम क्रिएट करते हैं। मोटे लोगों में मेटाबॉलिक डिसऑर्डर, डायबिटीज, ओबेसिटी, ये सब होता है, अस्थमा, आर्थराइटिस, बीपी। और दुबले लोगों में अक्सर ये एनीमिया, डेफिशिएंसी, डायरिया, अपॉर्चुनिटी। इनके लिए क्या जरूरी है? इनके लिए बैलेंस डाइट जरूरी है। और इस तरीके का भी नहीं बनना है कि हम खा खा के मोटे हो जाएं और झंझट में आ जाएं। तो ये ना तो दुबले ही होना सही है, ना बहुत मोटा ही होना सही है। मध्यम मार्ग का अनुसरण करना है। भगवान बुद्ध ने कहा था मध्यम मार्ग का अनुसरण करें। तो उसके लिए आचार्य कौटिल्य का एक अर्थशास्त्र चाणक्य का है, आर्यभक्त करके इन्होंने एक डाइट बताई। आर्यभक्त माने जो आर्य क्या खाएगा, तो उसके लिए है कि कितना ओदन, सूप, लवण और घृत तेल, मतलब दाल, भात और नमक, इनका और तेल का, मतलब इसमें देखेंगे तो कितना कार्बोहाइड्रेट है और फैट है और प्रोटीन है और साल्ट है। एक बैलेंस डाइट का उन्होंने ये दिया, कॉन्सेप्ट दिया। तो ये बड़ा इंपॉर्टेंट था, आर्यभक्त करके इसको संज्ञा दी। तो ये एक हमारे ऋषि मुनियों का एंशिएंट टाइम से था कि जो पौधे हैं वो एनर्जी ट्रैप कर रहे हैं। वो दो ढंग से होते हैं, प्राइमरी मेटाबोलाइट जिनको हम लोग फैट, कार्बोहाइड्रेट और प्रोटीन कहते हैं, और जो आगे चल के होता है दूसरा होता है सेकेंडरी मेटाबोलाइट्स। ये हमारा फैट और प्रोटीन और कार्बोहाइड्रेट, ये जो आहार में हमारे आयुर्वेद में इसको आहार की संज्ञा दी गई, ये अधिक मात्रा में लिया जाता है। और जो सेकेंडरी मेटाबोलाइट्स हैं जिनमें फ्लेविनॉइड्स, एल्केनॉइड्स, ग्लाइकोसाइड्स, कुमारिन या स्टेरॉइड्स होते हैं, ये सारी चीजें सेकेंडरी मेटाबोलाइट होती हैं। ये इन पौधों की जरूरत हमें डेली जरूरत है, कम मात्रा में लेना है और हाईली पोटेंशिएट होते हैं। एक मैंने एग्जांपल देने की कोशिश की सामने कि जैसे गन्ने का रस और अदरक का रस। तो गन्ने का रस हम लोग पीते हैं तो 1 गिलास, अदरक का रस लेते हैं तो 1 चम्मच, दोनों ही प्लांट से ऑब्टेन है। हम 1 गिलास अदरक का रस नहीं पी सकते, 1 चम्मच गन्ने के रस से कोई फायदा नहीं होगा। तो ये जब हम लोग इसको आहार और औषधि के रूप में समझते हैं, दोनों अलग अलग हैं, लेकिन आहार औषधि समुच्चय का कॉन्सेप्ट है, दोनों मिला देते हैं। हम 1 गिलास गन्ने के रस में 1 चम्मच अदरक का रस मिला के पीते हैं तो नुकसान भी नहीं करेगा और उससे पेट भी भरेगा हमारा। तो ये आहार औषधि समुच्चय का जो कॉन्सेप्ट है आयुर्वेद में, ये बड़ा जबरदस्त है। हमारे यहाँ डे टू डे लाइफ में हम देखते होंगे अपने घर में भी कि तमाम सारी सब्जी बनती है तो उसका कॉम्बिनेशन यूज होता है कि घुइयां को अजवाइन से छौंक रहे हैं, लौकी को जीरा से छौंक रहे हैं, या बैगन को मेथी से छौंक रहे हैं, इस टाइप से है, रायता को राई से छौंक रहे हैं। तो ये जो कॉम्बिनेशन है दादी माँ, नानी माँ और घर परिवार में जो यूज हो रहा है, ये कहीं बुक में नहीं मेंशन्ड है। अभी हाल ही में इधर मकर संक्रांति में अभी सबने जो है गुड़ और तिल का सेवन किया और उसके लड्डू खाए। तो वो जो है इसमें देखेंगे जो आहार और औषधि की बात मैं कह रहा था, इसको अभी थोड़ा और मैं स्पष्ट करता हूँ। ये एक इसमें फूड कंपोनेंट है, जैसे गुड़ है वो फूड कंपोनेंट है, तिल जो है मेडिसिन का कंपोनेंट है। इसी तरीके से डेली हम लोग दाल खाते हैं, उसमें हल्दी पड़ी होती है, तो दाल जो है फूड कंपोनेंट है, हल्दी मेडिसिनल कंपोनेंट है। तो ऐसे ही बच्चा होने के बाद में हल्दी सौंठ और मेवे के लड्डू वगैरह दे दिए जाते हैं। ये कॉम्बिनेशन है, कॉम्बो, आहार औषधि फूड का जो कॉम्बो है वो दिया जाता है। और ये ऐसा नहीं है कि हम एक पर्टिकुलर मेडिसिन ही देंगे या पर्टिकुलर वो नहीं देंगे। जो हमारा ट्रेडिशनल नॉलेज है, दादी माँ, आज किसी के भी घर में कहीं भी बच्चा पैदा होता है और वो सौंठ वाले लड्डू, तिल, हल्दी वाले मिल जाते हैं ना, तो समझ में आता है कि इनके घर में बच्चा पैदा हुआ है। अदरवाइज क्या है कि वो बर्फी लड्डू कुछ भी ले आएंगे, लेकिन अगर मान लीजिए अगल बगल उसमें तो 4 घर में लड्डू भेजने का भी प्रचलन है कि भेज दो भैया, तो उनको पता लग जाएगा कि इनके घर में खुशखबरी आई है। एक मतलब मिला जुला समाज हमारा ऐसा था, हमारी संस्कृति की, कि हम उसको मिल बांट करके करते थे। तो वो जो है, और मैं यहाँ पे पूरा ऑल ओवर इंडिया मैंने इसको टूर किया, हर जगह मैंने पूछा भी लोगों से कि बच्चा होने के बाद हल्दी सौंठ देते हैं, तो सब जगह मोर और लेस इंडिया में पूरी जगह हल्दी सौंठ दिया जाता है। कहीं भी इसका कॉम्बिनेशन का ब्रेकअप नहीं है, कहीं कोई इसका वो नहीं है, और सभी लोग देते हैं, चाहे भाषाई रूप से कुछ भी हो, चाहे जाति रूप से कुछ हो, धर्म के रूप से कुछ हो, लेकिन हल्दी सौंठ और मेवे की चीजें प्रेगनेंसी के बाद हमेशा दी जाती हैं। एक आगे चलते हैं थोड़ा सा यहाँ से बात की, नान औषधि भूतम जगत किंचित, दुनिया में कोई भी ऐसी चीज नहीं है जिसमें कि मेडिसिनल प्रॉपर्टीज ना हो। तो एक ये चार्ट था हमारे पुष्पांगन सर थे डायरेक्टर सर, उन्होंने एक ऑल ओवर सर्वे करके, इंडिया सर्वे करके किया था, तो उसमें देखा था कितने प्लांट्स, लगभग उन्होंने 17,000 प्लांट्स की स्पीशीज के बात कही कि जिस के बारे में नॉलेज पता है। उसमें ट्रेडिशनल नॉलेज ओरल में 8,000 स्पीशीज है, आयुर्वेदा में लगभग 900 प्लांट्स के बारे में नॉलेज है, यूनानी में 700, ऐसे उन्होंने अलग अलग डिफरेंट सिस्टम में, मॉडर्न में 30 प्लांट्स में। लेकिन ये सारा बहुत लिमिटेड है, ये मतलब टिप ऑफ द आइसबर्ग। हो सकता है इस पृथ्वी पे 2.5 लाख से ज्यादा तरीके के पौधे हों, या और उससे ज्यादा हों, जो हम जिनके बारे में जानते ही नहीं है, या जो आयुर्वेद के क्षेत्र में नहीं है। तो हमारा जो ज्ञान है वो बहुत ही मतलब टिप ऑफ द आइसबर्ग है, बहुत सारी चीजें हम नहीं जानते। तो हमारे तमाम सारे बच्चों को वो सब चीजों को जानने के लिए प्रेरित होना चाहिए, और नोन टू अननोन की तरफ बढ़ने का एक कॉन्सेप्ट है, और ज्ञान का जो प्रकाश फैलाने के बाद है वो अस्तोमा सदगमय या तमसोमा ज्योतिर्गमय, ये सारी प्रेयर करते हैं, वो इसीलिए करते हैं। थोड़ा सा प्लांट्स की बात भी कर लें। एक ये सब आप स्क्रीन में देख रहे होंगे, मोस्ट इम्पॉर्टेंट पौधा है जिसका कि हम लोग इस्तेमाल करते हैं, ये क्वीन ऑफ हर्ब्स कहलाता है, और ये मोस्ट इम्पॉर्टेंट है। इसको हर घर में ऋषि मुनियों ने स्थापित किया आंगन में। इसको हम लोग तुलसी कहते हैं। तो गाना भी था, मैं तुलसी तेरे आंगन की। तो उसमें वास्तव में हुआ क्या कि आंगन में ही क्यों रखा जाए, ये था कि चूंकि चारों तरफ से घर घिरा रहेगा, फिर थोड़ा सा ऊंचे करके रखते हैं जिससे पैर से ठोकर ना लगे, फिर इसको रेस्पेक्टफुली, है ना, हम लोग इसको दिया दिखाते हैं और जल चढ़ाते हैं। तो एक है कि इस पौधे के प्रति सम्मान प्रकट करते हैं। वास्तव में एलिक्सिर ऑफ लाइफ है, इसमें एडॉप्टोजेन एंटीऑक्सीडेंट गुण है, यूजिनॉल होता है, इस पे इंडियन जर्नल ऑफ फिजियोफार्माकोलॉजी में पेपर भी छपा हुआ है कि कोलेस्ट्रॉल लोअरिंग और ब्लड ग्लूकोज लेवल कम करने वाला, यूवी रेडिएशन से बचाता है। तो ये तुलसी वास्तव में इतनी इंपॉर्टेंट हो कैसे गई? तो वास्तव में ये वन ऑफ द मोस्ट ब्लेस्ड प्लांट है। जैसे तेंदुलकर के बारे में हम सब जानते हैं, तेंदुलकर क्रिकेट खेलते हैं, तो क्या हम सब क्रिकेट खेलने वाले या क्रिकेट जानने वाले तेंदुलकर बन सकते हैं? नहीं बन सकते। क्यों? क्योंकि जैसे तेंदुलकर एक ब्लेस्ड चाइल्ड है ना, तो ऐसे इसके बारे में भी कहावत है, तुलसी तुलसी ही रहती जो वन की घास, भई कृपा रघुनाथ की हो गए तुलसीदास। रघुनाथ की कृपा मतलब गॉड ग्रेस से, गॉड की ब्लेसिंग थी। इसको हम लोग बहुत ही पूजा करके ऑस्पिशियस, अभी कार्तिक के महीने में पूरा महीना भर दिया दिखाते हैं। फिर उसके बाद एक चीज है, हम लोग इनका विवाह भी कराते हैं शालिग्राम जी से। विवाह दो जीवित चीजों की या चैतन्य चीजों के में होता है। तो पौधों में जीवन है, ये तो बहुत बाद में सर जेसी बोस ने बताया। हमारे ऋषि मुनियों की दृष्टि बहुत पहले ही थी कि जहां उन्होंने कहा कि पौधों में चेतना होती है। और इसी बहाने वो क्या करते थे, जब बारात निकलती थी तो जिस घर में तुलसी नहीं होती तो वहां तुलसी की स्थापना की जाती थी। तमाम सारे वैष्णव लोग इसको आरती उतारते हैं, तुलसी कृष्ण प्रेयसी नमो नमः और महाप्रसाद जन्नी। हाँ, महाप्रसाद जन्नी मतलब कोई भी चीज आप भगवान को देंगे तो उसमें जब तक तुलसी जी पढ़ नहीं जाएंगी तो भगवान को एक्सेप्ट नहीं होगा। इवन हमारा जो शरीर है वो भी तभी भगवान स्वीकार करते हैं जब उसमें तुलसी दल और गंगा जल पड़ जाए, तो भी वो भगवान उसको एक्सेप्ट करते हैं। तमाम सारी आदि व्याधि जरा मुक्तम तुलसी तुम नमोस्तुते करके इसकी पूजा की जाती है। और सर्दी जुकाम खांसी बुखार हो जाता है तो पहली चीज जो हम लोगों के मुंह से निकलती है वो ये नहीं निकलती कि डॉक्टर के पास जाओ, पर्चा लिखाओ, दवाई की दुकान जाओ, दवाई खरीद के लाओ। अरे भैया, सर्दी जुकाम हो गई है तो तुलसी अदरक काली मिर्च की चाय पिला दो, है ना। तो 2 दिन 1 दिन सुबह शाम चाय पी ली, थोड़ा ठीक हो जाएंगे। हमें मालूम है कि इस ढंग के वायरल आते रहे हैं, ट्रेडिशन में बहुत पहले से ही ये होता रहा और हम लोग उससे सामना करते रहे हैं। तो उसके लिए बचाव के लिए ऋषि मुनियों ने हर घर में इसकी स्थापना की और हम लोग तुलसी अदरक की चाय पीते रहे हैं। तो ये एक हमारा ट्रेडिशन है। बहुत इंपॉर्टेंट एक प्लांट की बात करने जा रहा हूँ जो आंगन में हमने अभी बता दिया है, तुलसी होती है, ये दरवाजे पे होता है, अभी बड़ा पेड़ होता है, ट्री प्लांट होता है। इसका नाम है अजाडिरेक्टिका इंडिका। इसके नाम में ही बहुत विशेषता है। इसके नाम में हम लोगों का जो अस्मिता कहते हैं, पहचान, वो है छिपी हुई। इंडिका माने, इंडिका अभी आपने कार सुनी होगी, टाटा जी ने निकाली थी, रतन टाटा जी की गाड़ी। तो वो एक ये था कि भारत से निकली। तो भारत के जितने पौधे हैं, बॉटनी वाले लोग जानते होंगे, उसमें इंडिका आता है, जैसे मैंजीफेरा इंडिका, मधुका इंडिका, अशोका, सराका इंडिका। तो वो इंडिका जो है भारत के पौधों के बारे में आता है। ये अजाडिरेक्टिका क्या है? अजाडिरेक्टिका जो पर्शियन इन्वेडर्स आए थे, तो यहाँ थोड़ा हिस्ट्री की चीज जाननी पड़ेगी। तो सिंधु घाटी, सिंधु नदी पार करके आए, तो सिंधु के उधर जो था वो भारत था। तो उनको मालूम नहीं था पर्शियन इन्वेडर्स को, और सिंधु को जब वो मतलब बोलना होता तो स से की जगह ह बोलते थे, तो उससे हिंदू हो गया। हिंदू धर्म की बजाय एक मतलब देश की बात आती, हिंदू एक क्षेत्र की बात है कि उस जगह पे रहने वाले सिंधु नदी के उधर रहने वाले सब हिंदू थे। तो हिंदू जो है, जब इंग्लैंड से अंग्रेज आए तो उनसे हा नहीं बोला गया, इंडू हो गया। इंडू, सिंधु, हिंदू, इंडू, इनसी से सिंधका, हिंदका, इंडिका। ये फोनेटिक्स एंड लिंग्विस्टिक्स है जो मैं आपको बता रहा हूँ कि कैसे कोई शब्द की उत्पत्ति और निरुक्ति होती है, कि कैसे कहाँ से कौन से शब्द निकलते हैं। साइंस की बात तो पता नहीं कितनी है, कितनी व्यापक है, मैं नहीं जानता कि सब लोग कितना जान पाएंगे कि नहीं, या मैं भी कुछ जान पाऊंगा कि नहीं। लेकिन ऐसे ही है कि इतिहास से जो चीज शुरू हुई, तो सिंधु से हिंदू, हिंदू से इंडू, इंदु से इंडिका, है ना, सिंधका। तो इसका जो आगे का नाम है आजाद दरख्त उल हिंद था। आजाद दरख्त पर्शियन नेम था जब पर्शियन इन्वेडर्स आए। आजाद माने प्लेंटी में जो पाया जाए, दरख्त माने पेड़, और हिंद का, तो वही आजाद हिंद का जो है, आजाद दरख्त उल हिंद जो है, वो आजाद डायरेक्ट का इंडिका बन गया। आजाद माने प्लेंटी में, दरख्त माने पेड़। जिस जगह पे नीम के पेड़ बहुतायत में पाए जाते हैं वो भारत है। ये हमारी अस्मिता की बात है। जैसे मान लीजिए कि अगर कोई हमारे बीच में सरदार बैठा हो, तो हम कह सकते हैं ना कि सरदार कौन है? जिसके सिर पे पगड़ी है। तो भारत कहाँ पे है? जहाँ पे नीम लगा हुआ है। तो ये हमारी अस्मिता की पहचान है। मैं इसको बहुत ही इंपॉर्टेंट देता हूँ, क्योंकि जैसे पुराने लोग होंगे, ट्रेडिशन वाले, ट्रेडिशनल लोग, देखे होंगे घर में आंगन में अय्या बैठती थी। अय्या माने दादी उदी बैठी रहती थी। वैसे अय्या की तरह हमारे यहाँ तुलसी माँ बैठ गई है ना, अम्मा। वो तुलसी को माँ ही बोलते हैं हम लोग। और दरवाजे पे कौन बैठ गए? नीम बाबा, है ना। बाबा बैठते थे, उसके नीचे चारपाई डाल करके, उसके नीचे जो है नीम बाबा के नीचे हम लोगों के बाबा ही बैठते थे सच में। तो ये वास्तव में बाबा ही हैं जो हम लोगों की देखभाल करते हैं। इनको नेचर ड्रग स्टोर है, विलेज फार्मेसी, पैनेसिया ऑफ ऑल डिसीज कहा गया। बहुत सारी स्किन डिसऑर्डर्स में है। ये करंट साइंस का एक पेपर है जिसमें हर पार्ट इसका यूजफुल बताया गया, और आप लोग चाहें तो उसको देख सकते हैं अपने से भी थोड़ा सा, कि सब चीज इसकी, पत्ती, छाल, कोई भी चीज इसकी। इंपॉर्टेंट चीज ये भी एक मैं बताना चाहूंगा कि अभी हाल ही में पेपर में छपता रहता है सल्फास जो एक दवा का नाम है गेहूं वगैरह को सुरक्षित रखने के लिए। तो अक्सर उसका नाम गलत चीजों के लिए जो है अखबार में छपता है, खाद्य सुसाइड किया। लेकिन अनाज को सुरक्षित रखने के ऑर्गेनिक मीन्स में हम लोग नीम की पत्ती को गेहूं के साथ में रखते थे और सुरक्षित रहता। तमाम सारे वायरल फीवर्स, मीजल्स और ये सब पॉक्स डिसऑर्डर्स, चिकन पॉक्स और ये होते थे, तो उसमें हम लोग आज भी यही रहता है कि गर्म पानी से में नीम के पत्ते खौला करके और उससे नहलाते हैं, और नीम के पत्तों का बेड लगा देते हैं, उस पर सोते हैं। कहीं फोड़े फुंसी हो जाता है तो नीम की छाल घिस के लगाते हैं। तमाम सारी चीजें नीम की इस्तेमाल होती है। बुखार होता है, पतली टहनी उबाल के पिला देते हैं। तो नीम बहुत ही इंपॉर्टेंट पौधा है। और उसके बाद आता है हल्दी। मैंने 2 चीजें बताई अभी तक, बार बार रिकैप कर रहा हूँ। एक तो आंगन में तुलसी, दरवाजे पे नीम, और रसोई में हल्दी। हल्दी एक ऐसी चीज है जो हम सुबह शाम डेली खाते हैं। आधा चम्मच रोज सुबह, आधा चम्मच शाम को, चाहे दाल चावल खाएं, चाहे रोटी सब्जी खाएं, चाहे अचार रोटी खाएं, चाहे वेज खाएं, चाहे नॉन वेज खाएं, कुछ भी खाएं, हल्दी खाएंगे, है ना। तो हल्दी खाएंगे तभी हेल्दी रहेंगे, क्योंकि जिस तरीके का हमारा ये कंट्री है इसमें तमाम सारे कीड़े मकोड़े, मच्छर मक्खी और ये सारे होते रहते हैं, तरह तरीके के मौसम के वेरिएशन है। तो बॉडी को जनरली इम्यून करते रहने के लिए एक ऋषि मुनियों ने इसको इंग्रेडिएंट को हमारे फूड में डाल दिया कि अब तो हल्दी खानी पड़ेगी, हल्दी खाओगे तभी सुरक्षित रहोगे, हेल्दी रहोगे, हल्दी खाओगे तो हेल्दी रहोगे। इसमें तमाम सारी चीजें ऐसी हैं। कुरकुमिन एक तत्व पाया जाता है, आजकल इस पे बहुत काम कर रहे हैं लोग। एंटीसेप्टिक है, कट एंड बर्न पे इसको यूज करते हैं, एंटीबैक्टीरियल एजेंट है। यहाँ पे एक हल्दी घाटी की लड़ाई हुई थी, एक तो जो अलग हुई थी वहाँ पे, उस लड़ाई के अलावा ये भी था, पेटेंट की लड़ाई हुई थी, उसे भी हल्दी घाटी की संज्ञा दी गई थी। इसको अमेरिका ने कहा था फ्रेश ब्लीडिंग वुंड्स में जो है इसका इस्तेमाल करते हैं, लेकिन हमारे यहाँ जब इसको माशेलकर साहब DGCSI आए थे तो उन्होंने देखा और पाया कि चरक सुश्रुत संहिता में आया था कि जहाँ पे बच्चे की नाल काटते हैं पैदा होते हुए बच्चे की, हल्दी लगा दी जाती। तो वो वहाँ पे फ्रेश ब्लीडिंग वुंड से उसको फ्रेश ब्लीडिंग को रोकने के लिए हल्दी का इस्तेमाल करते हैं। तो उससे वो ये पता लगा कि ये इंडियन ट्रेडिशनल नॉलेज है और इसका पेटेंट का एकाधिकार जो था वो US का वहां पे ब्रेक करने को मिला मौका। तो उससे क्या हुआ, इसको भी एक द्वितीय हल्दी घाटी की युद्ध की संज्ञा दी गई। एक ये एविडेंसेस ऑफ टर्मिनी की तमाम सारी बीमारियों में इसका इस्तेमाल होता रहता है, और सूजन में, दर्द में, तमाम सारी एक ये क्या हो गया, सरल प्रयोग भी है। मैं बता रहा हूँ हल्दी का हम लोग अक्सर कर सकते हैं। जैसे आजकल जंक फूड खाते हैं तो उससे अच्छा है कि रोटी की में पुरानी बासी रोटी रखी है तो उसमें हल्दी की बुकनी वगैरह लगा के करें। तो हल्दी, सौंठ, अजवाइन, हींग, काला नमक, सेंधा नमक सब मिला के माँ लोग अक्सर बना देते हैं, और उसको खाने से क्या है कि हमारा इम्यूनिटी भी अच्छा रहेगा, डाइजेशन भी ठीक रहेगा और कोई बीमारी उस ढंग से नहीं होगी, पेट भी भर जाएगा। बजाय एक ये कार्टून है कि शादी ही ऐसा जख्म है जिसमें चोट से पहले हल्दी लगती है। वास्तव में इसको समझना पड़ेगा। हल्दी की रस्म इतनी इंपॉर्टेंट हमारे यहाँ हल्दी है कि इसका एक्सटर्नल यूज भी है और इंटरनल भी है, ड्रग के रूप में भी और कॉस्मेटिक्स के रूप में, कॉस्मेस्यूटिकल्स के रूप में भी, फार्मास्यूटिकल्स के रूप में भी, जैसे भी इसको समझ लें, फूड में भी। तो ये जो है दो अननोन बॉडीज मिल करके एक होने जाती हैं। तो ये हल्दी की रस्म इसलिए डाल दी गई कि ये डिसइंफेक्टेंट है, ये एक दूसरे के इन्फेक्शन, बॉडीली इन्फेक्शन ना हो जाए। तो ये है कि हल्दी की, दूसरा ये है कि इससे त्वचा में निखार आता है। अभी मैं वो देखता हूँ, अक्सर हम लोग बचपन में देखते थे हल्दी चंदन के गुण इसमें समाये को टर्मरिक का कैसे आता था, वो सुंदर दिखने के लिए। ऐसे ही है तमाम सारी चीजों में। हल्दी और सौंठ का इस्तेमाल आर्थराइटिस में करते हैं, हल्दी और आंवले का इस्तेमाल डायबिटीज में करते हैं। हल्दी चूना शहद अगर कहीं पे गुम चोट लग जाती तो लगाते हैं। हल्दी प्याज की पुलटिस बांधते हैं। तो हल्दी के बहुत सारे एन नंबर ऑफ यूजेस हैं। हल्दी का लड्डू खिलाते हैं, वो मैंने बताया था, हल्दी सौंठ के लड्डू। तो रसोई में हल्दी एकदम माइंड में फिक्स करना पड़ेगा। आप जैसे ये इन चीजों की स्थापना की गई, इनके लिए किसी दवाई की दुकान की घोषणा नहीं की गई कि आप जो है दवाई की दुकान से और डॉक्टर की दुकान से जाकर के लेंगे और किसी बुद्धिमान आदमी को, नहीं दादी, सब कॉमन मैन। इन्होंने कितना मतलब सरलीकरण कर दिया। मैं अपने ऋषि मुनियों के प्रति नतमस्तक हूँ। और अश्वगंधा है ताकत के लिए, आजकल मार्केट इसकी बहुत ज्यादा हाई है, इंडियन जिनसेंग कहलाता है। अश्व जैसी शक्ति इसमें रहती है, पावर हाउस इसको कहा गया है, और शक्ति के लिए इसका इस्तेमाल होता है। घी को, और एक और है बहुत पॉपुलर है घृत कुमारी। ये दोनों का मार्केट है एक्चुअली। मैंने जो दिखाया इसलिए दिखाया कि कुमारी मतलब स्त्री के लिए है, और अश्वगंधा यूजुअली जो है मैन पावर के लिए मैं लोग यूज करते हैं। इसमें घृत कुमारी के बारे में बात करना चाहता हूँ। यहाँ भी थोड़ा सा एक चीज है जो पंच महाभूत से लिंक भी करूंगा। जैसे इसके अंदर भीतर जो होता है, मांसल पत्तियों के भीतर जेल होता है। सबने देखा होगा एलोवेरा की पत्ती अक्सर लोगों के घर में होती है, ये जेल होता है। तो जैसे मान लीजिए कहीं पे आग लगती है, तो आग लगती तो हम लोग कहते हैं जल गया। जल गया माने जल तत्व वहां से चला गया, है ना, क्योंकि वो पंच महाभूत से बना हुआ है। तो क्या करना है, उसको बराबर लाना है, साम्या और साम्या, क्या जल डालो, आग लगे तो क्या करते हैं, जल ही तो डालते हैं। तो वही चीज है कि जल गया तो जल डालो, है ना, या जेल लगा लो। तो अंग्रेज जो है उसे जल का बोला नहीं जा रहा था, जल से जेल, जल से जेल। ये फोनेटिक्स और लिंग्विस्टिक्स है। जब आप बोलेंगे, सीखेंगे, भाषा के बारे में सीखना समझेंगे कि कोई शब्द जेल कहाँ से आ गया, तो जल से ही वो आया हुआ है और उसका मॉडिफाइड वर्जन ही है। तमाम सारी जगहों पे कटने में, जलने में इसका इस्तेमाल होता है। अगर जल गया है तो या तो जल लगा लो या फिर जेल लगा लो। ये बहुत इंपॉर्टेंट है। और इसके अलावा इसमें तमाम सारा पौष्टिक तत्व भी होते हैं और जो कि इस्तेमाल किए जाते हैं। हलवा बनाते हैं इसका, उसको भी पोस्ट प्रेगनेंसी में देते हैं। एक आंवला होता है, इसके बारे में थोड़ा सा बात करना चाहता हूँ। ये आंवले की पहचान ऋषि मुनियों ने बहुत पहले ही कर ली थी। च्यवनप्राश बनाया तो आंवल की वय स्थापना नाम श्रेष्ठम, एज को स्टेबलाइज करती है। इसमें विटामिन सी कंटेंट बहुत ज्यादा होता है। आंवले में संतरे का लगभग 17 संतरे के बराबर एक आंवले में विटामिन सी कंटेंट होता है। और इसमें जो है ये सेंट गायरोगाई उनको नोबेल प्राइज एक मिला हुआ था इस बात के लिए कि जो उन्होंने कनेक्शन खोजा था बायोलॉजिकल कंबशन विद द रेफरेंस ऑफ दिस, इस जो इसमें एसिड्स होते हैं। तो ज्यादातर इसमें गैलिक एसिड आपको ये लंबा समय तक रख सकते हैं। दूसरी चीज है कि इसका जो विटामिन सी है वो पानी में गर्म करने पे सबसे नष्ट नहीं होता, तो वो बार बार यूज हो सकता है। मुरब्बा के रूप में यूज हो सकता है, चूरन के रूप में इस्तेमाल होता है, अचार के रूप में इस्तेमाल होता है, चटनी के रूप में इस्तेमाल होता है, बर्फी के रूप में इस्तेमाल होता है। ये सारी चीजें इसमें जो आंवले के तमाम सारे इस्तेमाल होते हैं। तो एक फल अगर रोज खाने को कहा जाए तो डेफिनेटली आप लोग आंवला चूज करिएगा। ये एक बेसिक इंसाइट मैंने जड़ी बूटियों के बारे में दी। थोड़ा सा आगे बात ट्विस्ट कर रहे हैं यहाँ से कि ये वाई टू डू रिसर्च इन आयुर्वेदा, क्योंकि आज की डेट में इतना ग्लोबलाइजेशन हो गया, इंडस्ट्रियलाइजेशन हो गया। पहले था कि आयुर्वेद एक कस्टमाइज सिस्टम था, 1 आदमी 2 आदमी 10 आदमी 20 आदमी। अब ये कि हर आदमी को वैसा ही चाहिए। फिर आज की डेट में साइंस का बात आ गई तो हर चीज का एविडेंस चाहिए, एविडेंस बेस है कि नहीं, उसका प्रमाण है कि नहीं, डेटा पब्लिश है या नहीं। तो फिर सेफ्टी ऑफ ड्रग की भी बात आती है, फिर कन्वीनियंस कैसी है, फिर ड्रग स्टैंडर्डाइजेशन एंड क्वालिटी एश्योरेंस, सही दवा बनी है कि नहीं, स्टैंडर्ड ड्रग है कि नहीं, काम करेगी कि नहीं करेगी। फिर तमाम सारी नई दवाएं, नए फॉर्मुलेशन, नए इंडिकेशन, तो सब चीजों के लिए रिसर्च की बहुत जरूरत है। फंडामेंटल प्रिंसिपल्स भी समझना चाहिए आयुर्वेद का। और इसके लिए एक ये है कि फ्यूचर में एक आयुर्वेद का एक करिकुलम में डालना चाहिए। हर आदमी को आयुर्वेद पढ़ना चाहिए। इसलिए नहीं पढ़ना चाहिए कि डॉक्टरी करनी है, इसलिए पढ़ना चाहिए कि आप अपने पुरानी ज्ञान को जाने, संजोएं और अपने जो वर्तमान किसी भी क्षेत्र के लोग हैं वो उसके बारे में कैसे उसमें योगदान दे सकते हैं, एक रिसर्च कर सकते हैं। इसलिए आयुर्वेद को जानना जरूरी है। फिर इसमें क्लिनिकल ट्रायल की भी बात आती है, तो रिवैलिडेशन, डोज, ड्यूरेशन, इंडिकेशन, साइड इफेक्ट्स, इन तमाम सारी चीजों के लिए, या न्यू सोर्स मटेरियल, मैंने जैसे कहा कि प्लांट तो बहुत ज्यादा ग्रुप है, उसमें से हम कुछ ही केवल इस्तेमाल कर पा रहे हैं, क्या हम सारे पौधों का दोहन कर पा रहे हैं कि नहीं, इसके लिए हमें तमाम सारी चीजों के ट्रायल्स की जरूरत पड़ती है। ये जो रिसर्च प्रोसेस है, ये है क्या? रिसर्च प्रोसेस क्या होता है? हम लोग डेटा जनरेट करते हैं, फिर उस डेटा को इंफॉर्मेशन में कन्वर्ट करते हैं, इंफॉर्मेशन को नॉलेज में, नॉलेज को विजडम में, और विजडम को टेक्नोलॉजी में, और टेक्नोलॉजी से फिर हम वेल्थ या पैसा कमाते हैं। ये एक रूट है जो हमें समझना है कि हम रिसर्च करके आखिर करेंगे क्या। बहुत सारे लोग पूछते हैं, अरे सब इतना तो हम जानते हैं, सब ये तो हमें पता ही। नहीं, जितना हम जानते हैं उतना अच्छा है, लेकिन हो सकता है मैं बार बार कह रहा हूँ, टिप ऑफ द आइसबर्ग जानते हो, बाकी जो अज्ञात है उसका तो हमें पता ही नहीं है। तो वो हमें मतलब ज्ञात से अज्ञात की तरफ जाना है ना, तमसो मां ज्योतिर्गमय, तो वो एक समझना है। फिर इस तरीके से तमाम सारी चीजें हैं जो ये लगभग लास्ट स्लाइड के आसपास पहुंच रहा हूँ और मैं सम अप करूंगा। ये पौधों में तमाम सारी चीजें, एक्टिव इंग्रेडिएंट्स होते हैं, क्या एक टेबल है। ये वो पौधे हैं जो कि रसायन के रूप में हम लोग यूज करते हैं डे टू डे लाइफ में। तो उसमें सतावर भी है, दारू है, केवाच है, पीपली है, पीपर जिसको हम लोग कहते हैं, आंवला। एक हर्बलिज्म के ऊपर बात आती है कि कन्वेंशनल हर्बलिज्म था जिसमें सिंगल ड्रग या कॉम्बिनेशन यूज होता है। नियो हर्बलिज्म है कि जो उसमें एक्सट्रैक्ट बेस है, अब सारी चीजें एक्सट्रैक्ट बेस तमाम सारी यूज होने लगी। एक चीज मैं आजकल स्यूडो हर्बलिज्म भी कहने लगा हूँ कि आजकल मार्केट को एनकैश करने के लिए ना पेस्ट में भी डाल देंगे, शैम्पू में भी डाल देंगे, सोप में भी डाल देंगे, कंज्यूमर गुड्स किसी भी चीज में कोई एक हर्ब का छोटा सा टुकड़ा डाल दिया, कहने ये जो है हर्बल हो गया है, या ये स्यूडो हर्बलिज्म है। मोस्ट ऑफ देम थिंग्स आर 90% हो सकता है उसमें केमिकल हो, या उससे कम ही, बहुत ही कम हर्ब्स हो, लेकिन उसको हर्ब्स की तरफ मानेंगे। एक बात को समझाने के लिए ट्रेडिशनल मेडिसिन में क्या हुआ कि ये 3 टाइम होता है, सुबह दोपहर शाम। तो उसमें आजकल क्या हो गया कि सुबह हम लोग चाय पीते हैं, दोपहर में धुआं पीते हैं, चाहे सिगरेट हो या गाड़ी का धुआं हो, शाम को शराब पिया। आयुर्वेद के अनुसार क्या होता है, सुबह हम लोग जल लेते हैं या प्राणवायु लेते हैं, दोपहर में खाने के साथ मट्ठा लेते हैं, और रात में जो है खाने के बाद दूध लेते हैं। ये एक सिस्टम है, ये हमें डेवलप करना पड़ेगा हेल्दी लिविंग के लिए। ये सारी चीजें छोड़नी पड़ेंगी, इसको अडॉप्ट करना पड़ेगा। ऐसे ही देखेंगे, बड़ा इंपॉर्टेंट है, गुड़ के साथ विभिन्न प्रयोग है चूर्ण के। तो ये वास्तव में है कि बहुत छोटे छोटे प्रयोग हैं और बहुत अच्छे हैं, उपयोगी रहते हैं। कुछ सामान्य औषधियों के यहाँ पे योग भी मैंने बताया, त्रिफला है, हरड़ बहेड़ा आंवला, त्रिकटु सौंठ पीपल काली मिर्च। थोड़ा समय की कमी हो रही है, योगा है, ये भी करना चाहिए, हम लोगों को देखना चाहिए। एक अभी हाल ही में सर्केडियन रिदम पे, इस पे भी फिजियोलॉजी का नोबेल प्राइज मिला। ये आयुर्वेद में पहले से ही दिया हुआ था कि किस समय उसमें इसकी बात की गई है। तो ये पित्त और वात पित्त कफ के हिसाब से ही। हेलो,
I just want to say we have sufficient time. आप बहुत ही अच्छा मतलब हम लोग एक्सप्लेनेशन दे रहे हैं, तो काइंडली इलैबोरेट।
इस वाले को या किसको कहना है, इसको?
हाँ, मतलब ओवरऑल, मतलब आप टाइम का ऐसा कोई इशू नहीं है सर, तो आप जो भी एक्सप्लेन कर रहे हैं तो
काइंडली टेक योर ओन टाइम।
अच्छा, तो ये जो बात मैं कह रहा था, यहाँ पे थोड़ी सी बात आ रही थी, जैसे हम लोग गुड़ और सौंठ है, म्यूकस आता है लोगों को, तो अब थोड़ा डॉक्टरी अपनी बता रहा हूँ। तो उसमें गुड़ और पीपली है, ये भूख न लगने पे या ऐसे दिया, या यूरिन में बर्निंग मिक्सचुरेशन में जीरा का इस्तेमाल करते हैं। पाइल्स वगैरह में एक हरड़ होती है, टर्मिनेलिया चिबला, उसको यूज करते हैं। ये सारा गुड़ और काली मिर्च खांसी में इस्तेमाल करते हैं। ये एक सिस्टम है, मतलब तमाम सारे ऐसे कॉम्बिनेशंस हैं जो मैंने इसमें भी बता रहा हूँ कि अक्सर हम लोग नाम सुनते हैं। बहुत सारे हमारे लोगों के बीच में जोड़ों में दर्द या गठिया की शिकायत होगी, तो ये चतुर्बीज है, ये घर में बना सकते हैं, मेथी, अजवाइन, मंगरेल और चमसूर। तो ये आप नोट भी कर सकते हैं, या बाद में कभी इसको स्लाइड को हम दे भी सकता हूँ, या नोट भी कर सकते हैं। तो उसको कोई दिक्कत नहीं है कि हम लोग इसको बना लें और खा लें। या अभी चाय बनाते हैं तो उसमें सौंठ, पीपल, काली मिर्च डालते हैं, साथ में तुलसी ऐड कर लेते हैं। तो खांसी में हमारा जो आजकल आयुष काढ़ा वगैरह चल रहा है, वो सब इसी के ही मॉडिफिकेशंस हैं। ऐसी त्रिफला है, हरड़ बहेड़ा आंवला। तो आम, अमरूद, केला भी 3 फल है, तो क्या वो भी त्रिफला है? नहीं, वो आहार वाले में है, ये औषधि वाले फल है। तो ये समझना है, आहार वाले फल जो है वो पेट भरने के लिए होते हैं, औषधि वाले फल जो होते हैं वो थोड़ी मात्रा में, कम मात्रा में यूज होते हैं। ऐसे ही यहाँ पे गिनती के हिसाब से पंचकोल है, षट सखार है, रासना सप्तक है, हींग वाष्टक है, नवायस है, दशमूल है। तो ये पौधों के सेट बनाए हुए। दशमूल अक्सर जो है पोस्ट डिलीवरी पर्प्यूरियल फीवर होता है, बुखार आता है, उसमें हम लोग हर घर में देखते हैं दशमूला रिस्ट पिलाया जाता है। अगर नॉलेज ही नहीं होगी तो हम कैसे देंगे? हम कहेंगे अरे नहीं नहीं नहीं, कहीं नुकसान कर जाए। अरे भैया, जो चीज नुकसान करने वाली है वो तो तुम खा रहे हो, वो तुम ले रहे हो। लोग अक्सर मेरे पास आते हैं, वो मुंह में पान मसाला तंबाकू भरे रहते हैं, मेरे से कहेंगे, अगर मैंने किसी को अश्वगंधा लिख दिया या कोई चीज लिख दी, गिलोय लिख दी, तो कहेंगे कि ये नुकसान नहीं करेगा। अरे तुम्हें अगर ये होता नफा नुकसान का, तो तुम मसाला और तंबाकू खा के आते। तो वो कैसे हम लोगों को समझा पाते हैं, कैसे कन्विंस कर पाते हैं क्या सही है क्या गलत। पहले समझाती क्या गुड है और बैड के लिए। जितने बच्चे हैं यहाँ, आपको पता लगे कि व्हाट शुड बी योर गाइडिंग फोर्स। खुद को अपने और देखो, बना कि अगर गुड है तो यू डू, छोटा नहीं है। देयर इज नो अदर ऑप्शन। तो वो में कौन सा जज कर सकते हो, और वही चीज आयुर्वेद का है। आयुर्वेद कहीं अलग से कोई चीज नहीं है ना। आयुर्वेद में बहुत सारी चीजें हिस्ट्री है, बहुत सारी चीजें इतिहास है, बहुत सारा दर्शन है, बहुत सारा विज्ञान है, इतना कम्प्रिहेंसिवनेस है कि अब पौधों के बारे में तमाम सारी बातें कही गई हैं। दशमूल है तो उसमें 2 तरीके के सेट हैं, लघु पंचमूल और वृहद पौधे हैं, उनकी जड़ है, और पंचमूल छोटे, और वृहद पंचमूल में बेल, अग्निमन, श्योना, हमारे आज फॉर्चुनेटली महाशिव की पत्ती, बेल और जड़ी बूटियों के अलावा, मतलब बेल का फल जो वो दवाई के रूप में काम आता है, बेल का पका हुआ फल के रूप में लिया जाता है। तो ऐसे ही देखें कि उन चीजों को एम्फेसाइज किया गया, लेकिन हम युक्त है बेल का पेड़ देखते उसको नोचने लग जाता है। अरे भैया, जहां भी बेल हो तुम वहीं पे जल चढ़ा दो, शंकर जी वहीं मिल जाएंगे, है ना। तो वो एक माइंडसेट योग की बात है। अभी सब जगह योग से अब योगा भी हो गया, तो ध्यान, समाधि, ये सारा आजकल चल रहा है हेल्दी के लिए। आप दवा सुनने लगे, पैसा रहेगा। नॉक्टर्नल एनिमल्स, तो जो रात में जाते जिन्हें पीछे सो गए, जीवन को नियमित करिए, है ना। जो शरीर की कंडीशनिंग है, जो दिमाग का वो है, वो हमारा सनराइज से ही सेट है हम लोगों की जो बायोलॉजिकल क्लॉक है, और कि हम सूर्य का प्रकाश से हमारा दिमाग विकसित होता है, खुलता है। विकसित होने के बाद खुलेगा, ओपन होगा, हम तमाम सारी चीजें जानेंगे। तो ब्रह्म मुहूर्त में उठते थे, आयुर्वेद उसको कहता है ब्रह्म मुहूर्त उत्तिष्ठेत। तो वहां पे ब्रह्म मुहूर्त उत्तिष्ठेत क्या, ब्रह्म मुहूर्त में जाकर के लोग, जिनको हमें समझना चाहिए। और अभी हाल ही में जो बीमारी हुई उसमें ये सब था कि हैंड हाइजीन, सोशल डिस्टेंसिंग और वार्म वाटर और स्टीम भाप लेना, ये सारी चीजें, गर्म पानी पीना कितना लाभप्रद है। बिना पैसे के भी इससे हम लोग बचाव कर पाए हैं। और अगर सिर्फ यही सोचें कि मास्क हो, सैनिटाइजर हो, तमाम मतलब मार्केट ड्रिवन, अगेन वो सारा चीज आप अपने दिमाग में सोचिए। हम लोग नमस्ते करते हैं, है ना, हाथ नहीं मिलाते हैं। देखिए हमारी परंपरा और ट्रेडिशनल सिस्टम कि हम लोगों वो, हमें मालूम है कि एक दूसरे से छूने से लग सकता है तो छुएंगे ही नहीं, अननेसेसरी क्यों छुएंगे। तो ये चीजें अवॉइड करते थे। वो ये जो है हमें देखनी चाहिए कि हम कैसे अपने आप को स्वस्थ रख सकें, कहाँ पे पैसे अननेसेसरी ना मार्केट में दें। एक नर्सरी राइम की बात करते हैं कि बेस्ट 6 डॉक्टर क्या है, तो उसके बारे में कहता है कि ये सनशाइन, वाटर, रेस्ट, एयर, एक्सरसाइज एंड डाइट, ये भी बड़े इंपॉर्टेंट है। अगर हमने इन चीजों को 6 चीजों को मैनेज कर लिया तो ये नर्सरी राइम हमारे पढ़ाई नहीं जाती, ये सब पढ़ाया जाना चाहिए। द बेस्ट 6 डॉक्टर्स एनीवेयर, नो वन कैन डिनाई इट आर सनशाइन, वाटर, रेस्ट, एयर, एक्सरसाइज एंड डाइट, these 6 will ready you attend। ये सब चीजें हम लोगों को बताया जाना चाहिए कि तुम थोड़ा सनशाइन भी ले लो। विटामिन डी की कमी के आजकल इतने पेशेंट आ रहे हैं कि मैं बता नहीं सकता। और फिर पानी की कमी के हैं, तमाम लोग पानी ही नहीं पीते हैं कि टॉयलेट जाना पड़ेगा, पानी भी पियो। फिर रेस्ट नहीं करते हैं, है ना, एंजाइटी और डिप्रेशन, रात भर मोबाइल खोल करके लेटे रहते हैं फालतू फेसबुक और व्हाट्सएप बाजी में। एयर, फ्रेश एयर नहीं लेना चाहते, हाथ पैर हिलाते ही नहीं, ब्लड सर्कुलेशन होगा ही नहीं, प्रॉपर डाइट लेंगे ही नहीं, जंक फूड खाएंगे। तो भैया जब आप बीमारी की तरफ कूद ही रहे हो तो फिर किसी व्यवस्था या सिस्टम को दोष ना दें कि आयुर्वेद जो है काम नहीं करता, या दवा काम नहीं करती, या ये डॉक्टर ठीक नहीं है, या वो डॉक्टर ठीक नहीं है। हो सकता है आप ही डिजर्व ना करते हैं उस इलाज को। ये मेरा अपना फीलिंग है, गट फीलिंग मैंने ऐसे व्यक्त किया। तो ये लास्ट में मैं ये कहना चाहता हूँ कि जब किसी को आप देखें तो ऐसे ना सोचिए कि जैसे एक वो है ना कि 6 ब्लाइंड पर्सन थे और उनको एक एलिफेंट को दे दिया गया, दिखा दिया गया, तो जिसने जो पार्ट पकड़ा, पूंछ पकड़ा तो कहा रस्सी है, और पेट पकड़ा कहा दीवाल है, कान पकड़ा तो कहा फैन है। तो ऐसे ही किसी ने कुछ कहा, सूंड पकड़ी तो कहा ये तो अजगर है, या तो वो नहीं होना है। आपको टोटलिटी में दृश्य को देखना है, हर एंगल से देखिए आप, बायोलॉजी से भी देखिए, केमिस्ट्री से भी देखिए, संस्कृत से भी देखिए, हिस्ट्री से भी देखिए, है ना। तो एक ये इसका ही है जो कंक्लूजन दिया उन्होंने, दीज मेन डिस्प्यूटेड लाउड एंड लॉन्ग, ईच इन देयर ओन ओपिनियन एक्सीडिंग स्टिफ एंड स्ट्रॉन्ग, दो ईच वर पार्टली राइट एंड ऑल वर रॉन्ग। तो सब के सब गलत होंगे क्योंकि वो सही चीज जान ही नहीं पाएंगे, टोटलिटी में जान ही नहीं पाएंगे, और हम लोग ऐसे ही लड़ते झगड़ते रहेंगे। ये एक कैरी होम मैसेज है कि चोर इसे चुरा नहीं सकता, राजा इसे हार नहीं सकता है, और कोई इसे बंटवारा नहीं करता। व्यय कृते वर्धित, मतलब ये खर्च करने से बढ़ता है। नित्यम विद्याधन सर्वधन प्रधानम, ये जो खर्च करने से बढ़ने वाला गुण है सभी शिक्षकों के बीच में हूँ, और ज्ञान बांटने से बढ़ता है। यही बात कह के मैं अपनी बात समाप्त करता हूँ। ये हमारे बॉटनिकल गार्डन का एक दृश्य है, और मैं आभार व्यक्त करता हूँ, थैंक यू कहता हूँ सबको जिन्होंने मुझे मौका दिया यहाँ पे बोलने का।
बहुत बहुत धन्यवाद डॉक्टर संजीव ओझा जी, आपने हम लोगों को इतनी ज्ञानवर्धक बातें बताई। गाय का दूध, शहद, घी, सूर्य की अनंतिम ऊर्जा, पोमैटो प्लांट, इन सब के बारे में जानकर वास्तव में हम लोगों को बहुत बहुत अच्छा लगा, और अपनी भारतीय संस्कृति के बारे में और इसकी ऊर्जावान तत्वों के बारे में जानकर तो हम लोग मतलब पहले इसके बारे में कुछ पता भी नहीं था। तो इतनी सारी चीजें जो हम लोग अब आपसे हम लोग ने जानी, बहुत 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 ज्ञानवर्धक थी। आहार औषधि सामुच्चय की सार्थकता का भी आपने वर्णन किया, और इन सब के बारे में जानकारी देने के लिए बहुत बहुत धन्यवाद सर। नाउ I would like to thank our both the guests and convey my heartiest thanks to the valuable thoughts shared by Mr. Anand Pandey and Dr. Sanjeev Oza. According to Mr. Anand Pandey, we heard about the areas chemists should play their role for a sustainable development. According to him, chemists should develop processes to conserve resources. He talked about 5 R's. He talked about say no to single-use plastics, reduce, recycle, reuse, rots. He also talked about composting plants, generating wealth from waste, and he also talked about circular economy. I would definitely like to thank and would have the pleasure to meet Mr. Pandey and to know about the economic viabilities of all these thoughts which he shared to us. Thank you very much, Mr. Pandey. And संजीव ओझा जी के बारे में भर दिया, और आपकी जो वक्तव्य है उसको पूर्ण रूप थैंक्स देने के लिए मैं यहाँ पर डॉक्टर सीमा सरकार को आमंत्रित करती हूँ। मैम,
एक बात मैं बताने वाला हूँ कि मैडम पांडे, ललिता पांडे मैडम दिख रही है मुझे, तो एक बार उन्होंने इनवाइट किया था, लेकिन अनफॉर्चुनेटली मैं कॉलेज में आ नहीं पाया था। तो मेरे मन में था कि मुझे जो है इस बार अपनी प्रॉमिस को रखना है। रॉबर्ट फ्रॉस्ट की कविता है।
थैंक यू सर, थैंक यू सो मच।
नहीं सही थी, बुखार था, लेकिन मैंने कहा कि मैं आज अपना प्रॉमिस रखता हूँ।
बहुत बहुत भाग्य सर, अहो भाग्य हमारा। बहुत बहुत कहेंगे आपके आभारी हैं कि आज जैसा लेक्चर तो शायद हम मिस कर जाते अगर आप हमारे बीच ना होते तो वाकई में आपने आज, मैं सुन रही थी और मैं गुन रही थी, और मुझे लग रहा था कि मैं प्योर ह्यूमैनिटीज की हूँ, और इतना इंटरेस्टिंग लेक्चर आज साइंस का हो रहा है। तो जो हम एक हर लोग भी इसमें जुड़ा हुआ है इस समय, सब उसको जो है समझ रहा था, और बहुत ही इंटरेस्टिंग वे में आपने प्रेजेंट किया। मतलब आपने कोई चीज छोड़ी नहीं, लेके पेंटिंग, बॉलीवुड एंड आर्ट, आर्किटेक्चर, लिटरेचर, आपने क्या मतलब हर चीज को इसमें जोड़ दिया। And very interesting part was that hybrid, tree, that tomato, that is potato, and that आलू और टोमेटो को आपने जोड़ दिया, और इतनी सुंदर सुंदर मैंने आपने जितने मेडिसिनल प्लांट्स, हल्दी जो हमारे रोजमर्रा का अंग है, हल्दी का उपटन जो लगाते हैं, और आपने जो तुलसी, नीम, सारी चीजें जो आपने बताई, एक एक चीज, मतलब इस समय तो मैं उसको मतलब रिकलेक्ट ही नहीं कर पा रही हूँ। आपने इतनी चीजें बताई हैं, धारा प्रवाह बताई हैं, बहुत ही इंटरेस्टिंग वे में मतलब आपने प्रस्तुति दी है। वाकई में वी आर ब्लेस्ड टू हैव यू ऑन दिस प्लेटफॉर्म। ऑफ कोर्स डिजिटली अगर आप वहां पे होते, हमारे सामने होते, तो वाकई में इट नीडेड अ वेरी वेरी अपलॉड एंड यू नो बिग हैंड फॉर यू, रियली सर, रियली इट्स एन ऑनर एंड प्रिविलेज।
थैंक यू।
अब आपके लिए मेरे पास शब्द नहीं है, शब्द कम पड़ रहे हैं आपके इस लेक्चर को समाप्त करने के लिए। धन्यवाद सर, दिल से धन्यवाद,
और अपनी बात रखूंगा, ऐसे कोई बात नहीं है।
आपका आभार और तहे दिल से शुक्रिया सर।
तुलसी को दादी और नीम को बाबा की तुलना, ये भी बहुत बहुत विचारणीय और बहुत मजेदार था। इसने आपके वक्तव्य में बिल्कुल चार चांद लगा दिए, बहुत मजा आया सुनने में। थैंक यू सो मच सर। आनंद पांडे सर शायद मीटिंग छोड़ चुके हैं, अगर वो यहाँ पर हैं तो उनको भी बहुत बहुत धन्यवाद। आई एम रियली रियली वेरी थैंकफुल फॉर एन सच एन।
थोड़ा बिजी है प्रतिमा, मगर मैं उनको कन्वे करूंगी।
मैम थोड़ा फ्लोर ओपन करिए इंटरैक्ट करने के लिए। वोट ऑफ थैंक्स बाद में, पहले फ्लोर ओपन करिए इंटरैक्ट करने के लिए।
यस सर,
यस वो वेरी मैम, बिकॉज these are the very fortunate things to interact such learned people. हाँ सर मेरी एक रिक्वेस्ट थी कि इतना हम लोग आयुर्वेद और इसका एफर्ट जो कर रहे हैं, उसके पेस में जो एडॉप्टेशन है वो नहीं आ रहा है। लोग सुनते हैं, फिर पुरानी ही आदत पे चले जाते हैं और सारी चीजें। तो मेरी एक रिक्वेस्ट थी अपनी थिंकिंग से कि एक ये चीज हम कन्विंस करें कि आई एम नॉट द बॉडी, बॉडी इज ऑफ माइंड। मैं शरीर नहीं हूं, शरीर हमारी है।
एक्सीलेंट।
और इसी शरीर से हमको अपनी जिंदगी के सारे अनुभव, सारे उद्देश्यों को पूरा करना है और अनुभव करना है।
जी।
तो यदि हमारा ये इंस्ट्रूमेंट ही ठीक नहीं रहेगा तो फिर हम रियलिटी में अनुभव नहीं कर पाएंगे और वो कसक बन के रह जाएगी। तो शायद समहाउ माइंड में हम ये डालें कि आप शरीर नहीं हो तो टेस्ट जो है ना पीछे हो जाएगा, हेल्थ जो है आगे हो जाएगा। अभी तो क्या होता है कि मैं हूं, अब हमारी मर्जी, चाहे हम जो करें, हमारी मर्जी, चाहे हम जो खाएं जो पिए। लेकिन यदि हमारे माइंड में कॉन्शियस से हम ये कहें कि नहीं, यू आर नॉट द बॉडी, बॉडी इज द योर्स। तो जैसे मान लीजिए आपने मुझे 1000 रुपीज दे दिया, तो मैं 1000 रुपीज नहीं हूं, 1000 रुपीज मेरा है। तो हाउ आई शुड गो कॉस्ट इफेक्टिव टू मेक माई लाइफ सक्सेसफुल एंड इजी गोइंग। तो सर थोड़ा सा आप लोग एक्सपर्ट हैं, तो अपने नरेशन में इस पार्ट को कहीं हम ऐड करें, जिससे कि मैसेज ये जाए कि भाई आप शरीर नहीं हो, शरीर आपका है। तो आई थिंक इट विल बी मच बेटर, एज आई एम वेरी होपफुल and I am very grateful and I offer my gratitude to the great achievement, knowledge and spreading which is near to us.
आपने बहुत ही अच्छी बात कही। मैं बल्कि इस बात को कहता हूँ, और अक्सर ही हम लोग ये जो आपने कहा ना कि मैं शरीर नहीं हूँ, सारी समस्या यहाँ पे है दो तरीके की है। कि आप पता नहीं वेदांत दर्शन कितना पढ़े होंगे, थोड़ा सा वेदांत की अगर अलग से कभी बात करेंगे तो उसमें एक जगह आया है, द्वा सुपर्णा सयुजा सखाया समानम वृक्ष परिस्व जाते। मैं मतलब संस्कृत और उपनिषद, ये उपनिषद की बात बोल रहा हूँ। एक ही पेड़ पे दो पक्षी बैठे हुए हैं और एक पक्षी फल भक्षित करता है और एक पक्षी उसको देखता है। एक मन है, एक आत्मा है, एक शरीर है। तो ये दृष्टांत हमारे इसमें वेदों में है, उपनिषद में है। तो है क्या कि हमें ये इस चीज कैसे समझाया जाए। मान लीजिए माँ बाप ने या मेरे जो मतलब गुरु लोग हैं उन्होंने मुझे बता तो दिया, मैं तो लोगों को स्प्रेड करता हूँ, लेकिन हमारे यहाँ तो दूसरा भौतिकवाद शुरू हो गया। तो इस भौतिकवाद में क्या हुआ कि हमने अपने सुख को आगे कर दिया। अब इतनी बड़ी जबान है हमारी, है ना, उसके सारे की जबान होगी, इसमें टेस्ट है, यहाँ दो बातें आएंगी, है ना। इतनी छोटी सी जबान से मैनेज करने के लिए हम पूरे शरीर को बर्बाद कर लेते हैं।
राइट सर, राइट सर।
ये जबान में दोनों चीजें, हम उल्टा सीधा बोल करके मार भी खा सकते हैं, है ना। बात ही हाथी पाइए, बात ही हाथी पाओ, ठीक है।
राइट सर।
इतना ही मैनेजमेंट है। ये मैनेजमेंट जो है वो हमें यहाँ पे देखना है। हम लोगों की बात कभी खुले मंच पे होगी और
राइट सर, राइट सर, हाँ,
स्वागत है, या कभी हम भी आपके पास आ जाएंगे। तो ये समझना पड़ेगा हम लोगों को ही। और आयुर्वेद ये कहता है शरीर को क्यों महत्वपूर्ण माने, क्योंकि जिस दिन शरीर नष्ट हो जाएगा फिर आपका सब कुछ खत्म है।
राइट सर,
है ना। वो संस्कृत का एक और श्लोक है, आप लोगों को पता नहीं संस्कृत समझ में आ पा रही कि नहीं। सर्वम अन्य परित्यज शरीरम अनुपालयेत तद भावे ही भावानाम सर्वभाव शरीरनाम, है ना। सर्वम अन्य परित्यज, सब कुछ छोड़ करके, है ना, शरीर का ध्यान रखें। तद भावे ही भावानाम, उसी की वैल्यू है। वरना क्या आप मिस्टर सिंह नहीं रह जाएंगे, मैं डॉक्टर ओझा नहीं रह जाऊंगा, है ना। हम लोग को बॉडी कहेंगे, उसका भाव माने वैल्यू। तद भावे ही भावानाम, उसकी वैल्यू ही तभी, नहीं तो बॉडी है ना। उस बॉडी पे हम चाहे जो भी लगा लें कॉस्मेटिक्स, है ना, चाहे कितना सेंट लगा लें, चाहे कितना ये आर्टिफिशियल मीन्स है ना। ज्ञान को हम जीन्स नहीं पहना सकते।
यस सर,
है ना। या को हम कह दें कि आप अच्छा सा टी शर्ट पहनिए तो अच्छे स्मार्ट लगेंगे। अरे ज्ञान का बोझ अलग है, शरीर अलग है। तो ये सारी चीजें हम लोगों को समझना पड़ेगा, किस जगह क्या उचित है, क्या अनुचित है, और उस हिसाब से मान के हम लोगों को बोध कराना पड़ेगा। ये वास्तव में भारतीय दर्शन को हमें हर एक को बताना सिखाना है और उसमें मतलब अपनी दिनचर्या में लाना है, तभी हम लोग स्वस्थ हो पाएंगे। धन्यवाद।
थैंक यू सर, थैंक यू सर, थैंक यू।
मैंने थैंक्स गिविंग किया क्योंकि हमारे एक गेस्ट लीड कर रहे थे मीटिंग। मैं और भी प्रश्नों के लिए आमंत्रित करती हूँ, कोई और भी यदि प्रश्न पूछना चाहे किसी भी तरीके का, तो संजीव सर हैं हमारे साथ, उनसे पूछ सकते हैं।
And I think I am audible. डॉक्टर साहब आपको नमस्ते सबसे पहले तो।
हाँ। थोड़ी आवाज कम हो रही है, हल्की सी आवाज कम है। हाँ, ईयरफोन लगा के बोल रहे हैं वैसे थोड़ा। हाँ, बेटर है। नहीं हो पा रहा है, क्षमा करें। ये त्रिपाठी जी, आपकी आवाज हम तक नहीं पहुंच पा रही है।
शायद कुछ नेटवर्क इशू होगा सर, मे बी। और भी कोई अगर हैं जिनको अपनी क्वेरीज को इनसे सर के सामने रखना, प्रस्तुत करना है, तो दे
मेथी बात उसको उसे लौकी जो है इजीली, तो जीरा भी थोड़ा हल्का है, देसी घी के साथ कैसे करते हैं। हम पहले कान पकड़ते हैं, फिर नाक कैसे करते हैं, इसका नहीं मतलब है। मतलब यह है कि एक ट्रेडिशन है और हम वो ट्रेडिशन को किसी बुक्स में या किसी किताब में नहीं लिखा और हम उसको फॉलो करते हैं, है ना। हम उसमें गनीमत है कि मेथी डाल रहे हैं, कोई और चीज नहीं डाल रहे हैं, है ना, किसी और चीज का बीज नहीं डाल रहे हैं, या तो वो एक चीज है कि ये जो हमने फूड और मेडिसिन कॉम्बो बताया, ये उसका एक हिस्सा है। लौकी को आप देखेंगे फूड है, मेथी या अगर ये लेंगे, इसका कोई रिस्ट्रिक्शन नहीं है, मतलब रिस्ट्रिक्शन नहीं है। आप अपने स्वाद के हिसाब से, अपने हिसाब से जो बेस्ट सूटेड, कोई इसका वो नहीं है। लेकिन ये देखना है कि हमें उसमें मेथी पड़ रही है ना, जीरा पड़ रही है ना, वो जो इतना पड़ रहा है वो औषधि है, वो फूड नहीं है। फूड आपका लौकी है और जो जीरा है वो इतना सा है, है ना। पेट भरेगा लौकी से, इफेक्ट करेगा जीरा या मेथी। वो शरीर में नुकसान नहीं करेगा, मेथी भी नुकसान नहीं करती।
सर आपने एक अर्थराइटिस के ऊपर आपने 4 बीज की बात की थी, उसमें लास्ट वाला था शायद चमसुर करके कुछ वर्ड था, वो क्या है सर, उसको प्लीज बताइए।
आप प्लीज नोट कर लीजिए, बॉटनिकल नेम है लेपिडियम सेटाइवम। मैं तो बॉटनिकल इंस्टीट्यूट में हूँ तो सब चीज याद रखना पड़ता है। तो चमचूर का नाम है लेपिडियम सेटाइवम। सीड होता है राई की तरह, बहुत छोटा छोटा लाल रंग का, यहाँ पे मिल जाता है माता बदल पंसारी वगैरह में।
अच्छा,
बहुत सस्ता है। तो उसको साफ कर लें, धोने की जरूरत नहीं है। और साफ करके और मोटा मोटा दरदरा मतलब इसको पीस लेते हैं। मेथी, अजवाइन और मंगरेल जिसको कलौंजी कहते हैं, और चमसूर, ये 4 सीड है। ये कॉम्बिनेशन बहुत ही जबरदस्त है। मतलब अब तक मैं अपने मरीजों को देता ही रहता हूँ, अक्सर बताता रहता हूँ, तो दसियों हजार लोगों को बता चुका हूँ। ये मतलब आर्थराइटिस के लिए, बुढ़ापे के लिए, कॉन्स्टिपेशन के लिए, मोटापे के लिए, कई चीजों के लिए एकदम मुफीद चीज है और रेगुलर खाने लायक, कोई साइड इफेक्ट नहीं है ना।
कितना लेना चाहिए इसको?
1 चम्मच।
1 चम्मच मतलब दिन भर में 2 बार या 1 बार?
नीड बेस है। अगर आपको मतलब लगता है, हाँ, प्रेफरेबली रात में ले लें गुनगुने पानी से। अगर नहीं होता तो सुबह शाम ले लें। मतलब नीड बेस है, कोई भी चीज का बहुत कस्टमाइजेशन है। इसी में अगर मान लीजिए पेट साफ नहीं होता है तो मैं तो कहता हूँ थोड़ा सा हरड़ भून करके वो मिला लो। थोड़ा स्वाद डेवलप करना हो तो काला नमक मिला लो। तो आयुर्वेद में फ्लेक्सिबिलिटी बहुत है ना। ये नहीं है कि केवल हम इसी पे अटके हुए हैं। जाड़े का टाइम है, थोड़ी सी सौंठ मिला लो, है ना। लेकिन वो है गर्मी का टाइम है तो सौंठ हटा दो। तो ये कस्टमाइजेशन है कि हम कैसे, जैसे आप घर में लड्डू बनाते हो तो कोई भी चीज जो है उससे बना लो। अब कभी गोंद का बना लिया, कभी अलसी का बना लिया, कभी तिल का बना लिया, कभी सौंठ का बना लिया, तो वो अल्टीमेटली एक न्यूट्रास्यूटिकल फॉर्म में अंदर जाएगा। अलसी में ओमेगा 3 फैटी एसिड वो सारा जाएगा, है ना, या तिल में गया, तो वो बॉडी के लिए एसेंशियल कंपोनेंट है। वो हमें इम्पोर्टेंट चीज देखनी है।
डॉक्टर साहब मेरी आवाज नहीं आ रही थी, पीएम त्रिपाठी बोल रहा था, केवल 30 सेकंड लूंगा। एंड डॉक्टर सीमा माफी चाहूंगा अगर आपको कहने में
बिल्कुल कोई दिक्कत नहीं, आप बताएं।
मैम ने अभी पूछा, आई एम स्पेशली कोटिंग द वर्ड ऑफ डॉक्टर सीमा। डॉक्टर साहब, आई एम हेडिंग अराउंड 20 countries in one company that is called Okinawa, and I'm related to environment. Earlier I was heading Tata Motors, but I have left just because of some पर्यावरण के कमिटमेंट के लिए या इन्वायरमेंटल प्लेस के लिए मैंने वो जॉब छोड़ के अपने आप को स्विच कर लिया।
ठीक है, बहुत अच्छा, बहुत अच्छा।
जी डॉक्टर साहब ये पूछना था कि ये जो, जैसे मैं डायबिटिक हूँ डॉक्टर साहब, मैं बहुत सारी आयुर्वेदिक मेडिसिन कभी कभी लेता रहता हूँ, बट मैंने आज तक इसमें क्वांटिफिकेशन नहीं देखा। और प्रोफेशनली मैं एक इंजीनियर हूँ, तो मुझे ये लगता है कि जो चीजें क्वांटिफाई नहीं की जा सकती हैं उनके ऊपर कभी ट्रस्ट नहीं किया जा सकता है। इफ यू कांट क्वांटिफाई, इफ यू कांट डॉक्यूमेंट द थिंग्स, then you should not trust it. As an engineer I'm telling, मैंने आज तक कभी भी किसी भी आयुर्वेद की दवाइयों पे mg नहीं देखा कि वो कितने पावर की दवा है। अब डॉक्टर साहब जैसे मैं नीम के टैबलेट्स खाता हूँ, कभी कभी मूड करता है या गलती से 4 भी खा लिया तो भी कोई फर्क नहीं पड़ता है, 1 खा लिया तो भी कोई फर्क नहीं पड़ता है। एलोपैथ में अगर एक मेडिसिन रिपीट कर दे तो उसका साइड इफेक्ट तत्काल दिख जाता है। तो ये जो पावर डिस्क्रिप्शन है कि हमको किस पावर में खाना है, क्या खाना है, मैं दुबला आदमी हूँ तो भी 1 टैबलेट खाऊं, पतला आदमी हूँ तो भी 1 टैबलेट खाऊं,
नहीं,
100 केजी का हूँ। जी सर, आपका टाइम।
हाँ, बस थोड़ा सा ही कहना है। मैंने शुरू में भी बोला और अभी फिर से रिपीट कर रहा हूँ सर। एक तो पर्सन टू पर्सन, अभी जब आयुर्वेद के बारे में और जानेंगे तो कस्टमाइजेशन है, सबका शरीर अलग है, ठीक है। मैं आपको देख नहीं पा रहा हूँ, आपका वीडियो ऑफ है शायद, तो
मैं बहुत सिंपल सा दिखने वाला एक
नहीं काफी दिख गया, जितना दिखना था उतना ही देख रहा था।
बहुत बहुत धन्यवाद आपका।
हाँ, ये कहना चाह रहा था कि कस्टमाइजेशन है। आपने पढ़ा ही होगा कि मैंने थोड़ा सा वात पित्त कफ बोला, तो ऐसे प्रकृति टाइप होती है, वातज पित्तज कफज प्रकृति, और सब दवा सबको सूट भी नहीं करती, है ना। ये कस्टमाइजेशन सिर्फ आयुर्वेद में ही है। और जब आगे अभी की मॉडर्न टाइम की बात करते हैं तो मॉडर्न साइंस में फार्माकोजेनोमिक्स एक चीज है जो आई है कि एक पर्सन के लिए एक ही मेडिसिन बनती है। तो इसका कोई एग्जाम्पल भी मैं दे सकता हूँ, थोड़ा टाइम लगेगा अगर इन लोगों को सुनना हो। तो एक ब्रेस्ट कैंसर के लिए हर्सेप्टिन करके एक दवा बनी, तो वो था कि जब अमेरिका में ट्रायल लिया गया तो पता लगा बहुत ही कम लोगों में, 10% लोगों में फायदा करता था और 90% में फायदा नहीं किया। तो ये हुआ कि उसका बैक रिसर्च हुई, तो उन्होंने पाया कि आखिर एक पर्टिकुलर जीन है HER2 जिनमें प्रेजेंट है, उनमें ही अगर ब्रेस्ट कैंसर है तो उनमें ये दवा फायदा करेगी, हर्सेप्टिन। तो अब ये हो गया कि एक किट बनी, पहले अगर ब्रेस्ट कैंसर है तो ये किट से ये देखा जाएगा कि HER2 जीन प्रेजेंट है कि नहीं, तब फिर आपको हर्सेप्टिन दी जाएगी, तब वो फायदा करेगी, तो उसकी एफिकेसी 100% हो गई। तो ये डेटा जनरेशन और एविडेंस बेस और क्लिनिकल ट्रायल और तमाम सारी चीजें, ये अभी बहुत सारी चीजें जरूरत है। हम लोगों ने गवर्नमेंट लेवल पे बहुत सारी चीजें होती रही हैं, हो ही रही हैं, और अब तो बहुत ज्यादा ध्यान दे रहे हैं। पोस्ट कोविड में अभी आपने देखा कि आयुष काढ़ा, तमाम सारी चीजें ऐसी, जनरल इम्यूनिटी रेगुलेटर तमाम सारे आई हैं। एक है लाइफ स्टाइल डिसऑर्डर्स, जिसपे कि मॉडर्न मेडिसिन का कोई कंट्रोल नहीं है। मॉडर्न मेडिसिन केवल इमरजेंसी मेडिसिन के अलावा कुछ भी नहीं है, और अगर हम अपनी लाइफ स्टाइल मैनेज करें तो डायबिटीज शायद होने ही ना पाए, हो ही गई है अगर, तो तमाम सारे और ऑप्शंस हैं जिनको हम देख सकते हैं। आजकल दवाएं बहुत क्वांटिफाइड उसमें आ रही हैं, ऐसी बात भी नहीं है कि एमजी नहीं मेंशन्ड है, तमाम सारी दवाओं में मेंशन्ड है। और हमेशा मात्राबद्ध औषधि लें। वो मैंने पार्सेलियस वाली लाइन आपको पढ़ाई भी थी, बताई भी थी कि मतलब अगर आप मात्रा में गड़बड़ा जाएंगे तो पानी भी पिएंगे तो भी नुकसान कर जाएगा, है ना। ज्यादा पानी पिएंगे तो हाइड्रोसिस हो जाती है, किडनी फेलियर हो जाता है। ज्यादा नमक खाएंगे तो ब्लड प्रेशर हो जाएगा, है ना। ज्यादा चीनी खाएंगे तो शुगर हो जाएगा, ज्यादा घी खाएंगे तो कोलेस्ट्रॉल बढ़ जाएगा। तो वो आपको अति जो है सर्वत्र वर्जित है। हमेशा मध्यम मार्ग का अनुसरण करें और चिकित्सक और डॉक्टर की निगरानी में ही दवा लें। ये भी ना करें कि खुद ही मतलब मास्टर हो जाए, क्योंकि देखिए अगर मैं पुल बनाने लग जाऊंगा तो मैं पुल नहीं बना पाऊंगा, है ना। आप इंजीनियर हैं, हो सकता है आप पुल बना दें, हो सकता है आप कोई मशीन ठीक कर दें, है ना। तो जिसका काम उसी से साजे और करे सो थोथा बाजे। तो मेरा एकदम बहुत क्लियर कट कॉन्सेप्ट है, जिसका काम जिससे होना है उसी को होने दे, और जो डॉक्टर बताए वही दवा लें, है ना। वही आपको जज करके दवा देगा।
हाँ संजीव सर, मैं भी कुछ कहना चाहूंगा ये सर के क्वेश्चन में कि एक्चुअली सर आयुर्वेद में सबसे बेस्ट केमिस्ट्री ऑफ दिस यूनिवर्स इज विद इन द ह्यूमन बॉडी। और आयुर्वेद जो है ट्रीटमेंट नहीं करता है, ह्यूमन बॉडी में एक्टिवेट करता है ड्यू टू सम रीजन इफ समथिंग इज नॉट फंक्शनिंग। तो उसमें वो फ्लेक्सिबिलिटी जो है वो बॉडी के हिसाब से बॉडी अपना लेती है, बाकी डिस्कार्ड कर देती है। तो इसमें एग्जैक्ट क्वांटिफाई कर पाना भी मुश्किल है और करना भी नहीं चाहिए, क्योंकि मैथमेटिक्स में सर, मैथमेटिक्स में जब ये बाइनरी सिस्टम हुआ तो उसमें होता है 0 and 1, यस नो ट्रू फॉल्स। लेकिन सपोज आप रोड क्रॉस कर रहे हैं और कोई गाड़ी आ रही है, तो क्या आप एग्जैक्ट माप करके क्रॉस करते हैं और यू हैव सम आइडिया कि नाउ आई कैन क्रॉस इट, और आप स्टेप लेते हैं और आप सक्सेसफुल होते हैं। तो टेकिंग फ्रॉम दैट फजी लॉजी सिस्टम जो है कि बिटवीन 0 and 1 देयर आर मेनी पॉसिबिलिटीज एंड हाउ टू एड्रेस दैट। तो चूंकि हमारी ह्यूमन बॉडी अलग है, उसकी तासीर अलग है, तो इसमें हमारा ये जो फ्लेक्सिबिलिटी है कि यदि एक के बजाय हम 2 ले लेंगे तो नुकसान नहीं करेगा, तो दैट इज द सुपीरियरिटी ऑफ आयुर्वेद ओवर एलोपैथी।
बिल्कुल बिल्कुल।
तो दिस इज द ब्यूटी, क्योंकि वो बॉडी अंदर की केमिस्ट्री अपने आप इतना जागृत हो जाती है कि दैट इज गोइंग टू लुक आफ्टर अनलेस एंड अनटिल समथिंग वेरी एक्सेप्शनल। तो सर आई एम वेरी मच प्राउड ऑफ द ब्यूटी ऑफ आवर आयुर्वेद एंड द पर्सन। आई एम नॉट सेइंग
मेरा साथ दिया।
थैंक यू सर,
और
थैंक यू सर।
जो बाइनरी वाला आपने बताया, इस पे भी बात कर सकते हैं।
राइट सर,
इस पे थोड़ा समय की आप लोग बता दीजिएगा जहाँ पे दिक्कत होने लगेगी। बाइनरी वाले में ये है कि आयुर्वेद में हर चीज 2 ही बताई गई है, या तो ठंडा होगा या गर्म होगा। तो ये ठंडा गर्म का कॉन्सेप्ट भी ये है कि जब भी हम लोगों का जन्म होता है तो वो उष्णता का परिणाम होता है, और हीट बिटवीन 2 बॉडी से हम लोग जनरेट होते हैं और जन्म होता है। और ये शरीर में जब तक उष्णता रहती है तब तक हम लोग जिंदा रहते हैं। और ये उष्णता है जिसका मैंने कहा था एनर्जी, है ना, इसको हम लोग करेंसी फॉर्म में जोड़ते हैं कि हम जैसे किसी को भी लेबर हायर करते हैं तो हम उसकी क्या खरीदते हैं, हम उसकी एनर्जी खरीदते हैं।
इसके
बदले में उसे पैसा देते हैं। तो एनर्जी इज डायरेक्टली प्रोपोर्शनल टू करेंसी होता है। और जो शरीर का हमारे एनर्जी जब निकल जाती है, शरीर ठंडा हो जाता है ना, तो उसके लिए शब्द देखिए कितना खूबसूरत शब्द दिया है, निधन।
निधन, कोई धन नहीं। यस सर।
धन रहित हो जाता है, उसकी करेंसी खत्म हो जाती है,
खत्म हो जाती है। एंड दैट इज एनर्जी हैज गॉन। गॉन, राइट
है ना। अभी आपके पास परमाणु बम आता है तो वो आपका क्या है, वही करेंसी है। उसी से पेट्रोल क्या है, करेंसी है। आप पेट्रोल खरीदते हैं ना, पेट्रोल तो भाई हम लोग कहते हैं क्या का तेल है या पानी है ना, जो भी कुछ भी निकल रहा है तो सॉइल से निकल रहा है, अर्थ से निकल रहा है, कहीं से मिल रहा है, कोई प्रोड्यूस तो उस ढंग से है नहीं। फसल है, हमने 1 किलो गेहूं बोया और 1 कुंतल गेहूं उगा लिया, तो उतना हमने करेंसी पैदा कर ली। फिर जब गेहूं बेचेंगे तो उतना जो नोट छप के आएंगे वो मिलेंगे। वास्तव में इकोनॉमिक्स और ये जो बाइनरी सिस्टम है, ये हमारे, ये मैंने ठंडा और गर्म बताया, यही ठंडा गर्म हम पौधों में और तत्वों में देखते हैं कि कब आदमी के शरीर में ठंडापन कम है, कब गर्मपन कम है, वैसे ही चीज देते हैं। बुखार होता है तो बहुत ठंडा करते हैं, है ना, वो कोल्ड स्पंजिंग करते हैं, है ना। अगर मान लीजिए ठंड होती है ज्यादा, हीटर लगाते हैं, है ना। गर्मी हो गई तो एसी चलाते हैं। ये पूरा बाइनरी पे ही एकदम डिपेंडेंट है ना।
राइट सर।
तो ये बहुत थॉट है, कभी इस पे हम लोग बैठ के बात करेंगे।
यस सर,
बहुत ऐसा हो सकता है।
राइट सर, राइट सर। थैंक यू सर, थैंक
धन्यवाद सर। बहुत ही आकर्षक और बहुत ही मजेदार सेशन रहा ये। और अब हम लोग आगे बढ़ेंगे। इस सेशन ही का दूसरा भाग है।
एक्सक्यूज मी मैम, एक्सक्यूज मी मैम,
क्वेश्चन पूछना चाहते हैं सर से। ओके ओके, मे आई इनवाइट मिस दीप्ति टू आस्क क्वेश्चन टू डॉक्टर संजीव। ओके
सर, जैसे ही ठंड शुरू होती है हम लोग के सर में प्रॉब्लम होती है डैंड्रफ की। सर डैंड्रफ को कैसे कंट्रोल में कर सकते हैं? सर किसी किसी में सेबोरिक्स के रूप में हो जाता है, जो कि बहुत ज्यादा ब्लड में और इस काइंड में आ जाता है।
कई बार क्या होता है कि बॉडी टाइप ही ऐसी होती है जो बहुत ड्राई होती है स्किन, है ना, तो वहां पे डैंड्रफ हो जाता है, या कई बार फंगल इन्फेक्शन हो जाते हैं। तो ये देखना पड़ता है कि फंगल इन्फेक्शन तो नहीं है या बहुत ड्राई तो नहीं है। तो जो भी आजकल तमाम सारे ऑयल के एप्लीकेशन, हम लोग ऑयल कम लगाते हैं आजकल। तो शरीर में पुराने समय में ये था, ब्रह्म मुहूर्त में उठने के बाद है अभ्यंगम आचरेत नित्यम, रोज सुबह शाम जो है तेल लगा के मालिश भी करना चाहिए शरीर की। तो वो भी एक हिस्सा है नहाने के पहले और धूप को दिखाना। तो ये कहना कि शरीर में कहीं खुश्की हो रही है, रूसी हो रही है, तो उसको ये ऐसे ही है कि उसमें हम लोग वो उसको लगाया, उसमें ये डाल करके कपूर वगैरह। तो ये सभी तमाम सारे शैम्पूज आते हैं कीटाकोनाजॉल वगैरह के, मॉडर्न मेड्स में कोई भी स्क्राइब कर देता है। तो वो देखना पड़ेगा कि आपको मतलब किसी चीज से एलर्जी तो नहीं है, ये भी समझना पड़ेगा। हो सकता है किसी एलर्जी की वजह से हो। तो मेडिसिन एक अलग सब चीज है कि मतलब कोई मरीज ने अपनी प्रॉब्लम बताई, लेकिन एक ही चीज के लिए अलग अलग आदमी को अलग अलग दवा दी जाएगी। तो ये है कि बचाव में आप ऑयल एप्लीकेशन या लोशन या क्रीम या एलोवेरा जेल के आजकल बहुत सारे प्रिपरेशन आ रहे हैं, उनको यूज कर सकते हैं। मतलब नर्चरिंग करनी है, नर्चरिंग दोनों चीज हो जाए, मालिश भी हो जाए। जी
सर, इसमें मुझे भी कुछ कहना है।
बिल्कुल।
सॉरी सर, हाँ तो सेशन थोड़ा सा हो जा रहा है एक्चुअली। सर व्हाट एवर, अच्छा, शुड आई स्पीक और आई शुड फिनिश? मुझे एक्चुअली राइट, इस क्वेश्चन के आंसर में मुझे कहना है कि एक्चुअली मैम, व्हाट एवर इज द फिजिकल वर्ल्ड ट्रीटमेंट इन एनी फॉर्म, आइदर एलोपैथिक और होम्योपैथिक और आयुर्वेदिक, these are involving the physical material. इसका एक एडवांस पार्ट है कि whenever the season is changing or even every day, our every body cell has the mind. So if we will talk to them, being introvert.
एक मिनट में आपको इंटरप्ट ये कर रहा हूँ।
हाँ सर।
हाँ, कि हमें लगता है ये चूंकि कॉन्फ्रेंस है, यहाँ टाइम की
राइट सर, सॉरी सर, सॉरी
ये तो स्टूडेंट आपको मिल जाएंगे, आप भी डिस्कस कर सकते हैं और हमको भी कभी बुला लेंगे तो हम भी आपके यहाँ के पर्चे लिख देंगे।
ठीक है
हमारे नंबर ऑफ पेशेंट बढ़ जाएंगे। लेकिन ध्यान रखते हुए यहाँ पे सेशन को आप लोग
थैंक यू सर, थैंक यू सर, थैंक यू।
I would like to interrupt here. Actually, in fact, we are very happy to see this kind of response from the participants. But because of the time limits, we have to stop the session.
Yes, ma'am. We want to interact.
Yes. Questions in the chat box, and we will definitely convey it to the required speaker. I need the questions in the chat box.
There is one question, ma'am.
I
would like to ask you, sir, within 2 minutes.
Now, continuing our session, we have our the next part of our session comprises of oral presentation as well as poster presentations. And this session will be presided by Dr. Seema Joshi. I would definitely like to welcome Ma'am Joshi. Are you here? Dr. Seema Joshi was here a little earlier.
I'm here. Hello.
A very warm welcome to Ma'am Joshi. I would like to give a small introduction of Mrs. Joshi. Dr. Seema Joshi is the head of Department of Chemistry, IT College. She has 33 years of teaching experience. She is the faculty advisor, student government in IT College since 2007. She has organized various international and national conferences in chemistry at IT College and organized chemical popularization programs, research projects. In fact, research project on arsenic in drinking water was being done by her. She has presented papers in international and national journals. She has won many awards like Vaigyanik Samman, Bharat Raksha Dal Trust, Imam Jafar Isadeh Award for Educationist with Dedication, Sanjay Gandhi Gold Medal Awards, and many more.
I'm sorry, I think there is some technical issue from Pratima ma'am's side. Kindly bear with us.
Neha, if you please allow, we can continue with the session because already you are getting late.
Yes. Okay, so I would like to invite Mrs. Chandran Maurya to please call upon the name of the participants. Over to you, Mrs. Chandran.
I would like to, first of all, wish all the candidates.
Yeah. Ma'am Joshi, kindly give your address. Ma'am, I would like you to address here this session.
Yeah, I'm already in.
Welcome.
Thank you, Dr. Ghosh, for such a elaborate introduction. And good afternoon, everyone. And it's my proud privilege to chair this session of ICCHHE-2022, for which I extend my gratitude to Professor Manjula Upadhyay, Principal, Navyuga College, and Dr. Neha, Head Department of Chemistry, Navyuga College. Hygiene. We have heard about so many things about the hygiene and the good health, and the previous lecture was testimony to that. Hygiene has always remained historically important for maintaining both good health and good environment. In the ancient civilization also, the integrity of hygiene was maintained through our religions. As Dr. Oja just said, in all the religions, washing of hands, feet, and taking bath before offering any prayer was made mandatory. So this was the ancient time, and this was the method to sustain personal hygiene during that era. Later, at the times of Roman, Greek, and Egyptian civilization, along with personal hygiene, I must say that the sanitation also came into focus. And people started using personal as well as public washrooms for safe disposal of human waste. So this was the concept of the sanitation which gained importance in this era. During 19th century, understanding of communicable diseases established a relationship between contaminated water and the cholera, which was the epidemic at that time. During the same period, Louis Pasteur also discovered that the microorganisms are the cause for various diseases. And such small observations made the populations aware about the significance of personal hygiene and its scientific correlation with good personal health and good environmental health. We can say that environmental health is nothing, but basically it describes the human health in relation to the environmental factors. Industrialization and technology has already disturbed our ecosystem. And pandemic COVID-19 has shown its repercussions to us, all of us. Now, to restore the environment for good public health. Government is doing their role and need a positive participation from each one of us in such a scenario. And this conference in such scenario is a milestone. I appreciate the effort of Navyuga Kanya Mahavidyalaya and its organizing committee for choosing such a relevant topic for this conference. In this session, there are around total 26 presentations. And on behalf of the organizing committee, I welcome all the participants and request the speakers to stick to the time limit of 3 to 4 minutes, maximum of 4 minutes for their deliberations. 1 minute will be for the question answer session, and negative marking is for the extra time taken. So without wasting much time, I request Dr. Chandan to invite the first speaker for this session.
Thank you so much, ma'am. Am I audible?
Yes.
Okay, thank you so much, ma'am. Now, with your permission, I would like to invite our first participant, Dr. Rajiv Singh Yadav, PP01, to present his paper. Dr. Rajiv Singh Yadav. Are you here? Dr. Rajiv Singh Yadav?
I think Dr. Rajiv is not audible.
Dr. Rajiv is muted. He can kindly unmute himself.
He's not audible. Dr. Yadav, you are not audible.
आप अपने आप को अनम्यूट करिए राजीव जी.
नहीं दे रहा उन्हें. Message him.
All right.
Dr. Rajiv, please unmute yourself. Yeah, ma'am, you should give him full positive marks.
Ma'am, shall we invite our next participant?
Seema ma'am is here, so I'm nowhere to interfere in between.
Okay, our next participant, Dr. Renu Sharma, OP01. Are you here? Dr. Renu Sharma.
रेणु शर्मा जी को अनम्यूट कर दीजिए.
Please unmute. They are not able to unmute themselves.
Oh, I'll just check, ma'am. I'll just check.
Okay.
आई थिंक माइक के ऊपर क्लिक करने से अनम्यूट हो रहा है.
हाँ मैम, पार्टिसिपेंट्स अपने एंड पे अनम्यूट कर सकते हैं, हमने नहीं किसी को म्यूट अनम्यूट किया.
राइट.
यस मैम.
Some technical problem from their side. Maybe sometimes it happens.
She can rejoin. She can rejoin. And without muting herself, she can join.
Doctor, you can join later on again. So we can start with the third presentation.
Okay, ma'am. Dr. Basant Kumar, PP02. Dr. Basant Kumar.
Dr. Basant Kumar. Dr. Basant Kumar, are you there? I can't see him on the board. Chandan?
Ma'am, he is in the meeting.
He's in the meeting?
Yes, ma'am, he is here. Dr. Basant Kumar?
But his name is not among the participants list.
Dr. Basant Kumar has joined.
Okay.
Ma'am, we have checked. There is no technical error from our part. Participants have to unmute themselves at their end only.
Okay. Thank you, Neha.
Yes. Thank you. Basant Kumar ji, are you there?
He has shared his screen, so we are able to see his screen, but
बसंत जी आपकी आवाज नहीं आ रही है, आपकी एक बार आप बोलिए।
सुन ही नहीं पा रहे।
मे बी कि फोन से कर रहे हैं तो बैंडविड्थ की प्रॉब्लम है, ऐसा लग रहा है।
Sunil ji, who is handling the whole conference, he's saying that all participants can mute and unmute themselves. They have been given power to do so. So there is no technical error.
Actually, I think they are doing with the phone. So sometimes the phone and the microphone.
No, it's not there, ma'am. We are also using phone, but we can speak, we can listen. No problem is there at all.
So must be network problem, I suppose. So can we continue with the next person? Dr. Maurya, can we continue with the next one?
चंदन मैम अनम्यूट कर लीजिए।
Dr. Kumar Swami. Dr. Kumar Swami, OP02.
Yes, ma'am. Good morning to all. Am I audible, madam?
Yes, you are audible.
Thank you. Is it visible, madam? My screen is visible?
This is perfectly all right.
Yes.
Yeah.
Thank you, madam. Thank you, Anand. Myself, Dr. Kumar Swami, Assistant Professor of Mahatma Gandhi Institute of Technology, Hyderabad. Now, I'm going to take the oral session for one part multi-component synthesis of ferrol containing imidazole using tetrabutyl ammonium peroxide disulfate as a catalyst. So my research is related to synthetic organic chemistry. As of the real world demands how the synthetic organic chemistry play a very important role in particularly drug discovery and disease curing and all this. So in view of that, the scientists have put focus more on the eco-friendly synthesis and the high yield process. So in view of this, the medicinal chemistry put more focus on this. These are the common heterocycles focused on this. Now research is going on all nitrogen, oxygen, sulfur containing heterocycles. So my topic is related to benzimidazole and ferrol based. These are the biological activities of benzimidazole, various biological activities. And coming to my topic is one-part synthesis of ferrol containing imidazoles. So this one-part synthesis is one of the best method. It is an interesting tool in modern drug discovery because this method can avoid the isolation process and the large number of chemicals and multiple synthetic methods. So it can increase the cost also, thereby it can minimize the use of solvents and other. And we can also adopt the very simple conditions, reaction conditions, milder reaction conditions. So these are the methods we are adopting for the one-part synthesis. Coming to our topic, the present work. My work is the present synthesis of ferrol containing imidazoles using tetrabutyl ammonium peroxide disulfate. Here the tetrabutyl ammonium peroxide disulfate is a catalyst, is a very good oxidant. In this, we are considering our main focus is the synthesis of ferrol with containing benzimidazole using class and cause synthesis mechanism. It is a versatile method. You can see the synthesis process: 2-amino benzimidazole using dimethoxyfuran and alkyl halides by using tetrabutyl ammonium peroxide disulfate. By using various solvents, we are used acetonitrile and ethanol and even water also there. This is the possible mechanism for the target molecule. And this is optimization of the catalyst. In this, during the research process, we were used the following catalyst and different solvents. First, potassium persulfate, we are using the yield is obtained up to 54% only. Later, the potassium persulfate and tetrabutyl ammonium butyrate, using acetonitrile as a solvent, we got only 60% of yield. Later, we go to the our present catalyst, tetrabutyl ammonium peroxide disulfate. So by using 20% of the catalyst ratio, using different catalysis, different solvents like ethanol, acetonitrile, water, DCM and toluene. As for the data, we concluded that our catalyst is successfully working with using acetonitrile as the best solvent. So that in using acetonitrile, we achieved 85% of the yield. But the remaining compounds are 50, 45 and like that, the different solvent. In this, the solvent play very important role. So that is why we are choosing all our analogues are synthesized by using acetonitrile. And also we can, our derived products, the title compounds screen for antioxidant activity using DPPH radical scavenging method and nitric oxide scavenging method. This is the method we can adopt. So in this, all the solvents are dissolved, the substance are dissolved in DMSO. We can also observe the percentage of inhibition. So the compounds 4J and 4A2J are the analogues. In this, we observed that 4J and 4H have shown excellent activity due to the substituents that most trifluoro or trifluorine containing substance are gives more activity. Later, the 4H, that is also dichloro, 2 chlorines present on that. So all these substances can screen for their antioxidant activity using ascorbic acid as a standard drug. And coming to the experimental data, the melting points are measured by syntax and all the elemental analysis using percussion analysis and infrared spectra, means IR spectra of the compounds using FTIR, and proton NMR also using 400 megahertz C13 and mass spectra. This is the spectral data. And coming to our topic, my topic is highly challenging and novelty, and the one part synthesis is particularly, it is very good for present generation. So mostly I'm expecting this methodology is good and it is suitable for the well-reputed international journals. And this is my topic. Thank you one and all.
Thank you, Dr. Swami, for your presentation. Any questions? If there are no questions, we can continue with the next participant.
Okay, ma'am. Prabhakar Mani Tripathi, PP03.
Please unmute yourself. Mr. Tripathi.
Am I audible now?
Yes, you are.
Yes.
Oh, I'm very sorry. I have killed a few seconds. First of all, good afternoon and welcome to all this veteran society. My gratitude to all the teachers. And before going to my presentation, I think my screen is visible.
Yes, it is visible.
Yeah, thank you very much. So I would like to just tell background of my all those researches and what I'm doing. Earlier, I was heading one company that was called Tata Motors, and I was head of UP and Uttaranchal. Prior to that, I was handling Hyundai. My basic working was in automobile only. In 2013, I was the few of those people who were escaped and saved in Kedarnath disaster. From that point of time, I was very worried for all those ecological as well climatic changes. मुझे ऐसा लगा कि शायद शिव ने मुझे एक नया जीवन दिया है कुछ अच्छा करने का. Then I spent around 4 years traveling to many countries where I can get the solution, especially Germany, China, Taiwan, Australia, to understand how I can contribute in my field to the this pollutant and ecological changes. And finally, in 19, I took the admission in Singhania University as a research scholar. I left automobile industry, considering this is enemy to the society, and I shifted my career to an electrical vehicle industry. Now I'm heading around 20 countries with one company that is India's toppest performer in electrical vehicle, that is called Okinawa. I'm in India. My research will incorporate all those subjects. More than 40 lakhs of people every year, they are dying with just pollution. 7 lakhs of people are dying due to the in-house pollution, which is causing in the home itself. There are more than 15 childs which are dying before the age of 5 years just because of pollution out of each 10 lakhs of the people. There are 3 years of shortages of the life because of the pollution of an average Indian person. And there are so many impacts, so many respiratory disease and everything. I have gone through the discussion of all the people. Everybody is talking about something which has happened and now they are talking for the remedies. Ma'am, this is the time to work on the core. What is the problem? And we have to hit the problem. Many of the diseases, many of the issues, they are just because of the pollution only. India government is allotting 20% of their budget for hospitals and this human well-being, but out of those 20% of budget, 80% budget is just utilized in the respiratory disease. If you go by the ranking, out of 180 countries, we are ranking at the 188. If you just go by the polluted city globally, 6 cities out of 10, they are from here only. In one state, I'm talking, if you just go by one state, Andhra is emitting 22 tons of carbon per day. Where this society will go? Then I decided, okay, ma'am, the government, my just before 4 days I was Mr. Nitin Gadkari and I have presented all those things. And one fellow has talked about this ethanol. I would like to tell India is not ready for ethanol because in Angel's current infrastructure.
But the already 4 minutes have gone.
Okay, ma'am. So I will just share my last slide with your due permission. Although I have incorporated many actions. Basically, government plan to promote need more cohesive for the pollution to avoid respiratory disease, death of innocent child and citizens. Green energy and EV is the immediate solution, but government is failed to standardize the policies which immediate need of the time. Infra and subsidy is the key desirable actions from governments for the next 5 years. I will finish. I will not go. I will share my presentation for the audience and I will send to you also, ma'am. So thank you very much for giving this valuable platform. I'm highly obliged and thank you again. Thank you very much.
Thank you. Dr. Basant Kumar. Dr. Chandan.
Yes, ma'am.
Dr. Basant Kumar wants to.
Okay. Our next part, Dr. Basant Kumar, PP02.
एक मिनट. Okay, ma'am.
Yes, sir.
Ma'am, actually there is some network issue. I am unable to share my screen. I would like to just discuss my one PPT only orally as. My topic is
Please share your PPT.
Is it visible, ma'am?
No, it is not visible.
No,
please share first.
Ma'am, actually it is on my screen, but it is not visible. Can I only represent 1 minute in just 1 minute, ma'am?
Then you need to open your video also.
Okay, ma'am.
I request all the participants to be ready before.
Ma'am,
it will save our time.
Ma'am, my topic is toxicity of heavy metals, special reference to mercury. First of all, we should know what is heavy metals. Any element having atomic number more than 20 or having density 5 times greater than water is called as heavy metal. With reference to heavy metals, it is important to note that most of heavy metals are toxic to human beings, including lead, cadmium, mercury, and arsenic. Mercury is most toxic, which is harmful to human being for its health purposes. The sources of mercury toxicity is anthropogenic as well as natural sources, which goes into the environment from where by precipitation undergoes to aqueous environment from where fishes absorb there into the body and they further go to the human by their interaction. Mercury is exposed to human being by 6 major sources. First of all is air, second one is water, third one by eating the fish, fourth one is the amalgam fishing, fifth one is the cosmetics, and last one is the vaccines. These are the major sources which causes mercury toxicity. One disease which is called as mini matter disease, which is due to the poisoning of mercury to the human being. This disease was discovered in Japan, and in this disease the bones becomes brittle and twisted. As far as mercury exposure, among these 6 sources, amalgams, vaccines, and seafoods are the major part which are responsible for the toxicity. As far as human health is concerned, it affects the nervous system. It also affects the speech, vision, and muscle movement. Also, it causes some chewing and swallowing problem to the human being. As far as treatment of the mercury poisoning, the chelation therapy is important for mercury poisoning, and the removal of the source is also important. Thank you, ma'am. Thank you for providing the chance to me.
Any question? We can continue the next one.
Dr. Sonika Bhatia.
I am presenting just now. Is my screen visible?
Yes, it is visible.
Yes. A very good afternoon to all the participants and the organizers present on this platform. I'm Dr. Sonika Bhatia from Isabella Thoburn College, and I'll be presenting today under the disaster management subcategory. I'm presenting a review on oil spill disaster management and mitigation. So now the oil spill can occur because of natural calamities and man-made causes as well, like a tanker collision, a terrorist attack, a pipeline leakage, and an oil well blowout. They require an immediate response and a well-coordinated plan and strategy to contain the oil spill and to avert major disasters. I have shown 3 pictures. The first one is showing a bulk carrier which hit onto a coral reef. The second, the most major oil spills, it occurred due to an explosion which occurred on Deepwater Horizon drilling platform. And the third one is because of a corroded pipeline. So spills occurring close to the shore can be very, very catastrophic. And that can be seen by the pictures I have pasted on top. Thus, the best oil spill response is to collect it from the environment before it reaches the shoreline. There are various cleaning methods which we can use. I have shown some by way of some pictures, like booms. They contain the oil spill like a fencing. Skimmers have vacuum pumps which remove the oil from the surface. Dispersants are nothing but surfactants. They are sprayed on top of the surface and they emulsify the oil. Sorbents are adsorbent pads made of synthetic and natural substances both. In situ burning, this can be utilized if the oil spill is very, very far off from the shore. Otherwise, it will pollute the air. Bioremediation is mostly used near the shores. Stabilizers like polyisobutylene, it gelatinizes or solidifies the oil oil. And there are novel, many cellulose-based sorbents and dispersants which are being made from agricultural waste. Basically, the agricultural wastes are being modified by adding the adding hydrophobic chain so that the hydrophilicity of them can be reduced. So I have now given a systematic management of this disaster risk reduction plan by way of 5 steps. The first step which I have identified is risk assessment. A table of all the risks has to be made and the parameters, risk parameters associated with it. Once the response has been made, then comes the time of identifying equipments and maintaining them in a state of readiness. Regular maintenance and mock drills need to take place. So this is what I talk about sensitivity mapping. Environmental sensitivity index maps developed with the aid of remote sensing and geographic information systems are a compilation of information about reference. If there is an oil spill close to the shoreline, then and we cannot escape it reaching the shore, then what one needs to do is one needs to deflect the oil in such a manner that it reaches the shore at areas which are that is the dark blue areas, so that the damage is minimum. And that is what these maps are used for. The other very important thing, very important software which one can use is for advanced oil spill trajectory mapping. And this is a free software, MedSlick model, which is available on the net. Everyone can use it. Herein the inputs are the wind speeds, the bathymetric data, that is the depth of the sea surface temperature, and the sea currents, and the type of oil. That means the type of oil which we spilled. So we take the API gravity of the oil and add it to this particular software. And what we stick to the time. Okay, what we get out of this is this particular map where we see that in 14 days it is plotted, and we see that in 14 days it will hit the shore. So month-wise, location-wise, volume-wise, multiple maps for various volumes of oil leakage need to be mapped in advance for reference. These predictions for the path help in directing the resources to the shoreline. Now, if you see in India, it is the Coast Guard Headquarters, Directorate and Fisheries Environment, which is in charge of making a national oil spill disaster contingency plan. They have brought together all the resources, that is Government of India, state government, shipping, ports, and oil industries, and they've a clear concept of oil spill response. That means when it's a small response, it is managed locally, that is from the tanker from where spill is happening. Tier 2, the head would be the national coordinator, that is the Indian Coast Guard, and mutual aid partners. They will work together with the incident control. International help is sought. India is a member of OSR UK. So now I will like to conclude. We can see future oil spills can be mitigated by increasing preparedness for disasters, by doing regular trainings and mock drills. Though we see that the contingency plans are in place, but a stricter adherence of all stakeholders is needed. The annual updation of National Contingency Plan is very important. Concentrated efforts must be done to scale up the environmentally benign absorbent and dispersal technologies which are being made in the labs to large scale. And here I have showed the 5 ports where Indian Coast Guard have kept their oil spill response facilities. And in the left, if you see, I have given the detailed inventory of the Mumbai port. Thank you. And these are my references. Any questions? I'm open to questions.
We can have the next participant.
Dr. Meeta Rani, OP04.
Good afternoon. This is Meeta Rani from Department of Physics, Mahavidyalaya Degree College. Presenting Is the screen visible, ma'am?
Yes.
Thank you. Oh, sorry. Am I audible, ma'am?
Yes, you are audible.
Yes.
Thank you. So this is a review and it's being of a drug discovery and pharmacy development. And myself, Meeta Rani from Mahavidyalaya College. Actually, the discovery is a difficult process and expensive process. Identification of unestablished target to a new disease or an unmet clinical condition is to start with. Solid state properties, X-ray crystallography, mixture analysis, spectrometry, etc. monitor the modification of target after application of a new drug molecule and its quality performance. This in turn leads to further optimization and selection of a molecule for the selected target. More importantly, regulations and approvals through drug discovery and the development is achieved then. So initially there is a need of data to establish a selective target. And variety of biological entities such as protein and RNA is a need. The relation between target and disease should be monitored continuously. Target validation and multi-validation approach increases confidence levels. Following target validation, compound screening assays are developed, which confirm the modification of target. The prospective molecule becomes a hit molecule after its action is confirmed in retesting. High throughput screening is preferred as it checks the action of entire library of prospective compound against the target. Further, NMR technique looks for the hits confirming the action of prospective molecule. Secondary assays confirm the site of action. So, the assay reproducibility, cost and quality paves the way for progression in discovery of new molecule. This way, the new molecule is referred as a development candidate, which, if successfully marketed, is the medicine. Further formulation, development process, development, and a control strategy is established. So this is mixture analysis. Mixture analysis is actually material characterization techniques which play a significant role in the determination of a composition and phase purity of various ingredients and confirm action of the target site. Few of the methodologies used for doing quantitative analysis of mixtures are sample preparation, data collection, which leads to establishment of calibration standards. The qualitative analysis of mixture method leads to the evaluation of specificity, precision, robustness, etc. The use of analytical techniques such as Raman spectroscopy, NIR allows real-time process monitoring and controls, which is crucial for drug development. Next is physical stability. Actually, physical instability of drug being developed for modification of the target site must be thoroughly investigated prior to being administered. Physical instabilities such as change in crystalline structure, loss of order, phase transformation, discolor, melting, hydration, etc. may lead in nature of crystalline order, particle size, and morphology. These physical instabilities initiate a pharmaceutical interest to continuously improve until a physically stable drug product is achieved. Next is chemical stability.
Dr. Rani, sorry to interrupt, but you are exceeding your times.
I'm sorry, ma'am.
Already
Finding.
you are exceeding.
Yes, ma'am. So this is quality by design. Quality by design it can be done correctly in many steps, and this only reduces the time to market the developed pharmaceutical drug. Thank you, ma'am.
Thank you.
Thank you, ma'am.
Next participant.
Nagmilika Siddiqui, PP04.
Ma'am, is my screen visible?
Earlier it was visible, but now it is not.
Okay, ma'am, just give me a moment. Is it, ma'am?
No.
Yeah, show this.
No, it is
Yes.
No,
Okay.
it is
Great. So today I am here to talk about the kinetic and thermodynamic aspects of potassium bromide in understanding the metabolism of drug. Why this area of research, why I've chosen this as one of my favorite topics, it is because the drugs, how it is metabolized in the body. If I talk about particular one, which I've taken for now, it is isoniazid, also known as isonicotinic acid hydride. Much of my concern because oxidation of carboxylic acid wasn't much talked about earlier. And I've taken with the very common oxidant, which is KBRO3. So in here, first let me give you insight about the isoniazid, which is an anti-tubercular drug. It basically, if in active tuberculosis, it is sometimes mixed with streptomycin, and if in latent tuberculosis, it can be used as alone. So in this reaction and all these aspects of particular oxidant as well as the drug, the reaction was basically carried out to study what intermediate are formed. Obviously, if the oxidation is there, then the product is likely to be known. It very much is. But the intermediate, the metabolites, whether the toxicity is being created with the particular reaction, how well it is behaving in the body, and what negative impact it can give to the body. It is expected to be studied. Since it is the very first stage of my research, so not much I can give you insight about, but I've seen and the methods and materials I've used, I will try to elaborate as much I can. So for the particular of this reaction, we in the lab we have prepared all the solutions, reagents, and whatever we have used in double this for particular end of side reactions. And as the reaction proceeded, what I have seen, what I have understood till now, that the intermediates were formed. Obviously, they were, but in regular manner. Since for now we have studied the reaction at 4 different temperatures, that was from 25 to 40 degrees Celsius. So the observation was key for every 5 degrees in the temperature, the reaction, the rates were doubled, almost doubled. And it was in acidic medium for now. Alone perchlorate, the reaction proceeded quite very fast. So later on, it was mixed with the acid HCl, and the reaction was measurable in the presence of hydrochloric acid. So H plus ion was, and it was observed that the rate of reaction
was. Try to please wind up.
Yes, ma'am. I'm just at the end of my discussion. The rate of reaction was in negative first order with the increasing strength of the HCl4, which is acidic medium, and at the last, not much I have given insight to you about, but the discussion is all about that whether any free radical mechanism is being taking place alongside, or is there any kind of idiosyncratic toxicity in this particular combination, or whether what the what the further advancement we can do in this particular field in the class of anti-tubercular drug in oxidative process. So I think I am short of time. So is there any question? I'm open to all questions, please.
Any questions? No questions.
Okay, thank you so much.
Varsha Pathak, B0.
Yes, ma'am. Am I audible?
Yes, Varsha.
Okay, ma'am. Thank you. Can you please tell me, is my screen visible or not?
Yes, it is visible.
Okay, ma'am. Thank you so much. Only when the last tree is cut down, the last fish is eaten, and the last river is poisoned, man will know that he cannot eat money. A very warm good afternoon to all of you present here. I am Varsha Pathak, and the topic I have to present is disaster management and environment protection. Years and centuries before, our forefathers have the tendency to prevent the environment either naturally or sentimentally. He pointed out, there is enough resources in the world for everyone's need, but not for anyone's greed. The need of the hour is down the greed to the need. Arises what is disaster? So a disaster is a sudden event that affects the functioning of the society and results in human, economic, and environmental loss. Disasters of all kinds: natural disasters like earth, hurricanes, cyclones, environmental emergencies like forest fire, oil spills, complex emergencies in which Ukraine conflicts. These types of emergencies like wars, conflicts come under this category. Last is the pandemic emergencies, which means the sudden onset of any contagious disease worldwide. I mean, who doesn't suffer from COVID? Now, how can disasters? So disaster includes stages. First of all is prevention, because we all know prevention is better than cure. Second, mitigation. It involves the measures to prevent or reduce the negative impact of disasters. For example, seawalls are built to protect coastal areas. Third is preparation. We prepare to deal with disasters when it occurs. For example, conducting mock drills, training people to protect themselves and disaster. Then is response. It refers to the immediate assistance in times of disaster, like by government or NGOs, as moving people to safe areas, providing shelter and food to the affected people. And last is recovery. That is restoring the affected areas, rebuilding the livelihood. Not just disaster management, but disaster prevention is equally important. If we talk about India, NDMA, that is National Disaster Management Authority, was formed in 2006 under the Disaster Management Act and takes care of the disaster management in India. Now I will not take much of the time, and I will conclude myself by saying, although the countries have improved a lot in disaster response, but there is a need to put more efforts into disaster mitigation and recovery. Effective disaster management strategies will not only prevent loss of lives, but also help affected people in rebuilding their lives in the shortest possible time. So I hope you would have liked my presentation. Thank you.
Thank you, Varsha. Next.
Ayushi Singh. Ayushi Singh, PP06.
Hello, ma'am.
Yes.
Yes.
Ma'am, I'm audible.
You are audible.
Yes.
Okay, ma'am. I'm going to.
We can't see your slides. We are unable to see your slides.
Ma'am, I'm trying to network is not connecting.
No, you are not audible. The voice there, but not you are saying.
Well, ma'am, there is some network problem.
Try later on.
Okay, ma'am.
Next.
Dr. Usha Rani Singh, OP05.
Good afternoon, ma'am. I am audible?
Yes, you are audible.
Yes, you are audible.
Afternoon of all the organizing committee also. My topic, my name Dr. Usha Rani Singh, Associate Professor, Chemistry Department from Mahavidyalaya PG College. My topic is essential micronutrients in berry and their health benefits. Berry is a fleshy fruit without a stone from a single flower containing ovary. The berry is the most common type of fruits in which the entire outer layer walks into the potentially edible pericarp. Berry may be formed for one or more carpels from the same flower. Berry contain antioxidants which help keep free radical under control. Free radicals are unstable at a beneficial amount, but can damage our cell when their number get too higher, causing oxidative stress. There are more than 25 berry in the nature found. We show all the pictures, but the most common consumed berry are the strawberry, raspberry, blackberry, berry, black nuts, and red. The different berry we will show in the picture. Micronutrients in berry contain a range of micronutrients which are essential for health. Different type of berry contains a high level of vitamin C, antioxidant activity which act as a cofactor in hydroxylation reaction required for collagen synthesis, may have a role in hormone synthesis, in immunoabsorption, platelet aggregation, thrombus formation, preventing heart disease, a range of cancers. Berry and dietary source is a water soluble vitamin B, which is essential to prevent neural tube defect in newly born baby. It also may play a role in reducing risk of heart disease and cancer through a range of mechanisms, including lowering homocysteine level, catalyzing nitric oxide formation, and maintaining DNA stability. Berries are a rich source of phytochemical, only anthocyanin and flavonoids are glycoside for their red, violet, purple, and blue color. Phytochemicals are not and their absence does not result in a deficiency. However, they may have bio which could impact the health and disease. Berries are a rich source of such phytochemicals, in particular anthocyanin and flavonoids. Concentration of berries will influenced by many factors, including environmental condition, degree of repellence, cultivation, processing, and storage of fruits. The anthocyanin are conjugated anthocyanidin, which provide the distancing, and there are 6 and distributed throughout the plant kingdom, which again mainly 4 phenolic group. There is much variety of which are some fruits such as carbonberry, and there is only one type of anthocyanin, that is cyanidines. The anthocyanin of a tissue is characteristic as been a detection and identically show the structures R1 and R2 of anthracite. These are the benefits. Different have different type of from एंटीऑक्सीडेंट है, इन्फ्लेमेबल है, कोलेस्ट्रॉल डिक्रीज के लिए गुड है एंड प्रोटेक्ट कैंसर और and in jam form it is lost vitamin C in third. So it is a suggestion that the fresh food is more minerals than jam and jelly form. Berries are rich source of essential, particularly vitamin C and follicles. They have potential. Thank you, ma'am.
Thank you. It was informative. Any questions?
Time.
Yes. Quite interesting. But we need more time to explain us. Thank you.
Dr. Rekha, OP06.
Ayushi Singh is presenting. She had left earlier. Okay. She could not.
Okay. Ayushi Singh, OP06.
Ayushi, again you have. Ayushi.
Hello, ma'am.
It is not visible.
Yes, am I audible?
It is not visible. You are audible, but your presentation is not visible.
Ma'am, I had shared the presentation. Is it visible now?
Yes.
Yes, now it is visible. Now it is visible.
Okay, ma'am. Thank you, ma'am, for providing me the opportunity to present it after some time. Sorry, ma'am, for the delay. I am Ayushi Singh and I am going to present my work entitled Phase Control Solver Thermal Synthesis and Oxygen Absorption Reaction, OER activity of nickel sulfide nanoparticles obtained from a novel precursor 1,2-bis diphenylphosphinoethane nickel 2 acetyl isopronate diethylate. And we all know that we are aware that the fact fossil fuels has increased the demand of developing renewable energy sources. In this regard, electrochemical water splitting has emerged as a green and sustainable approach. Water splitting consists of two half-cell reactions, hydrogen evolution reaction and oxygen evolution reaction. Now, the overall practical application of oxygen, this water splitting is mitigated by the oxygen evolution reaction, which needs very high over potential. So, it is required to develop some electrocatalyst that can catalyze it so that it takes less time. First, in this preliminary work, we had synthesized a single source precursor, acetyl isopronate diethylate, and further it was decomposed into different elements, thiol and octadecylmine. It was synthesized by reacting this precursor nickel DPPCl2 with acetyl isopronate diethylate ligand to give this precursor. This precursor structure was characterized by crystallography. Further, there were different types of which was molecular framework, OH and nickel hydrogen and acoustic interaction was also visible. Further, the precursor was characterized by UV visible spectroscopy. These two bands were appearing which were arising due to intraligand charge transfer taking place in the ligand. Further, the supramolecular interactions existing in the structure were also assessed by Hirschfeld surface analysis. showing denorm plot as well as fingerprint plots. And then the different type of interactions existing in strong molecular architecture were assessed by energy framework calculation. Further, the decomposed products were characterized by powder XRD, which showed that the two decomposed products, NiODA and NiDT, have different type of phases. NiODA was having a single phase, Ni3S2, whereas NiDT was having mixed phases, NiS as well as Ni3S2. Then the particle size of two decomposed products were assessed by scanning electron microscopy, which showed that particle size of NiODA was smaller than NiDT. Then the OER or oxygen evolution reaction activity of the decomposed products were assessed by linear sweep voltammetry as well as electronic impedance spectroscopy, which provided the information that NiODA was having a better catalytic activity towards oxygen evolution reaction with a smaller overpotential of nearly 243 millivolts. Further, the mechanism of this overall oxygen evolution reaction was shown to occur by 5 steps. First, the hydroxide ion was formed, which was further reduced to oxide ion, and in next step, hydroperoxide ion was formed, which was then further reduced to peroxide, and in last step, oxygen was evolved. Further, the states and band structure calculations were performed, which provide information about the differences in catalytic activity of two materials that were obtained from decomposition carried out in two different types of solvents. So overall, I will conclude that we had synthesized a novel precursor. Then it was decomposed in different types of solvents.
Time is over.
That is, okay, thank you, ma'am.
Wind up fast.
Okay, ma'am. Two different type of solvents it was decomposed, and then we got two different type of decomposed materials which were used as electrocatalysts for oxygen evolution reaction. So we have shown that how the nature of phase and morphology plays a pivotal role in deciding the electrocatalytic properties of the obtained materials. Thank you, ma'am.
Thank you. Next.
Dr. Rekha Rani, OP06.
I'm here. Good evening, everyone. Namaste to Richa ma'am and Seema ma'am. I'm very grateful to give me the chance to present my some research work here. I'm sharing my slide. Just give me a single second. And here today topic is the assessment of coal fly ash impact on the environment. Especially I have taken this topic within two aspects. One is the environment and another is the human health. And I estimated this all study by estimated in the water with the fish. So these are the contents of this topic. And here the introduction of this topic is, as we all know, the energy is the essential part of living and non-living beings, and there is no any machine and life represent itself in the working form without the energy. For all the purposes, energy can be different. It may be the kinetic and it may be the potential energy. But for the different type of machinery, we used this form from where? From the fossilization of the plant in the form of the coal. So coal is the primarily used as a fuel to generate electric power. Power is the only source to operate any kind of machinery and the physical level and day-to-day living needs. Coal fly ash is a byproduct of coal that is used in the thermal power plant for power generation, is derived from the coal combustion. And the Indian scenario, the coal quality has a rich amount of fly ash content with low calorific value. So the fly ash is a waste product generated from the coal combustion in a large amount, consisting of the in the form of the ash, bottom ash, boiler slag, and fuel gas. Fly ash is the main component of the coal waste, is composed of the spherical particulate matter, which having the diameter in the range 0.1 μm to the 100 m. Fly ash mixture comprises with the silica, maximum in the iron, in the aluminium, iron, calcium, and the other heavy metals. this mixture contains some arsenic lead at the trace level. As a result of leaching of fly ash in the water, its noxious constituents hamper the aquatic ecosystem. Changes in the aquatic environment will seriously impact all over the world and the surrounding and badly affected all type of the lives. So in this, you can see that how we can use this fly ash in the different sector of the India and our economy. And but after using that, there is the 76 major coal fly ash pond accidental between we found here 2010 to June 2020 that have caused death, loss of property, and have resulted in extensive pollution of the nearby water sources and air and the soil. As you know well, the all life related to the water, air, and the soil. In especially the Madhya Pradesh, Odisha, Jharkhand, West Bengal, Tamil Nadu, Chhattisgarh, and the Maharashtra have the highest concentration of the coal-fired thermal power plant and the top the list of coal ash accident according to the report. And here you can see this Central Electricity Authority New Delhi report 2021, the use and utilization of the coal fly ash in the million as per the year. This is the fly ash you can see here, and this is the process how we can get this and the utilization and the material method how we achieve this result. Then here, as per this study, combustion is higher than the comparative use of the fly ash. So the exercise is also high to avoid the environmental degradation. That's why this is reaching easily into the water, air, and the soil contamination. So we have studied taking some fishes as per the weight 4, 5 to 8 inch. And here we study after the acclimatization, these fishes in the acute form continuously for 3 sections in the day. There is the morning, noon, and the evening. You can see here the sample of this coal fly ash. Here you can see the aquaria and the fish. And this is the observation of this particular study. Response of fish regarding the natural water and the contaminated water. Meal response you can see here. I'm not reading all that. This is self-explanatory because the time shortage here. In the fly ash, we found here the aluminium, copper, nickel, chromium, silica, heavy metals. There is the mercury, lead, and the iron. And this is the fish in the natural water where we can see the pectoral fin movement in the day. And here you can see the swim pattern of fish towards horizontal direction in the aquaria, in the normal water as well as the polluted water. And here you can see the next slide, the last. Okay, just I'm giving this result and discussion. I'm just giving the result and discussion, madam.
Okay.
Okay. So here you can see the impact of the on the human body in the form of the inhalation through the respiratory tract, and the wild animal in the form of the gestation, juvenile, and the matured during their life cycle, and the fly ash impact on the plant too. As we found, these all studied through the investigation on the upper side of the fish, and this is the impact of the heavy metals on all over morphology, anatomy of the fish, and this is showing by this graph. And this is my conclusion that this is badly impact our all the environment and the health. Thank you very much for giving me the time. I'm just removing myself. Any question, please?
Sorry, I keep on disturbing you all, but because the list of the candidates is so long that if
it's okay, it's enough to me. It's enough to me. Thank you, madam. Thank you, Seema, madam, and everyone. Thank you very much.
Thank you. Next.
Preeti Singh, PP07.
Preeti Singh is there?
Preeti Singh.
Is Preeti Singh there? No. Next.
Dr. Nishit Rastogi.
Good afternoon to all of you. Now I am sharing my slide. Am I visible to all of you?
Yes, Dr.
Yeah. First of all, I would like to thank the organizers for giving me this opportunity. The title of my paper is Synthesis of CYP and mannose bases of 5,6-dimethyl-indolinones and their biological evaluation. As we know that indolin 2,3-dione and its derivatives possess a wide variety of activities like cytotoxic, anti-tubercular, anti-malarial, anti-fertility, tyrosine kinase inhibitor, anti-conversion, enthalmatic, enzyme inhibitory, etc. Keeping this biological activity profile in mind, some new CYP and manic bases of 5,6-dimethyl indolin 2,3-diones have been synthesized. Sorry. One minute, please. Please bear with me.
It's okay.
I'm just trying.
It's okay.
Now, the starting compound was methylparaben, right? Which on reaction with 4-nitrobenzyl bromide gave 4-nitrobenzyloxybenzoate, which on hydrogenolysis resulted into 4-nitrobenzoxybenzoyl hydrazine. This hydrazine on condensation with 5,6-dimethyl isocytine and one substituted 5,6-dimethyl isocytines resulted into CYP bases. The simple CYP base, which was having hydrogen at the first position on being subjected to aminomethylation with secondary amines like morpholine, piperidine, pyrrolidine, dimethylamine, and diethylamine resulted into manic bases. These are these are CYP bases. Here you can see the bands, the band for this methyl here at value 2956 centimeter inverse, and in NMR you can see the peak for both the methyl groups at 2.30 delta, right? And benzyloxy for this methyloxy group, you can see the peak at 5.12 delta, and in the case of N-methyl substituted isocytine, you can see the peak for this N-methyl group at 2.65 delta, and in the case of N-ethyl isocytine, you can see the triplet for this methyl group at 2.14 and quartet for this methylene group at 2.60. And these three compounds gave molecular iron peak inconsistent with the molecular formula. There, similarly, in the case of N-acetyl isocytine, right, you can see the peak for this methyl acetyl, at 2.36 delta, and in the case of aminomethylated isocytines, you can see the band for this methylene group at 2830 centimeter inverse, and for this morpholino group, here you can see the triplet at the position of 2.64 for these 4 hydrogens, and a triplet at 3.67 delta for these 4 hydrogens. And similarly, in the case of piperidino group, you can see a multiplet between 1.42 to 1.59 for these 6 protons of methylene groups, and you can see a triplet at this for these 4 protons at 2.56, and for this methylene group you can see the peak at 4.44 delta. And all these compounds again showed molecular iron peak inconsistent with the molecular formula. In the case of pyrrolidine group, you can see a multiplet for these 4 protons and triplet for these 4 protons at 2.39. In the case of dimethylamino group, here you can see very well singlet for these 6 protons at 2.39 delta. And for this methylene group, you can see the singlet at 4.29 delta. And in the case of diethylamino group, you can see a triplet for these 6 protons and quartet for these 4 protons at 2.00 and 2.22 delta respectively. And all these compounds from 3 to 9 were screened for their antifungal potential against Candida albicans, Cryptococcus neoformans, and Candida parasites. These 3 are yeast fungi. And again, for dermatophytes, trichophyton, mentophytes, and Aspergillus fumigators, these 2 are dermatophytes or causing skin infection, severe skin infection, mostly local. And the testing method was used for tube dilution method at the concentration of 100 microgram per ml. And MIC was measured. Out of these, from 3 to 11 compounds, only 2 compounds having substitution at one position, acetyl group and dimethylamino and diethylamino group, they showed MIC 3.12. This was little more than the standard value, and in this case, I used the standard compound fluconazole. And in the case of morpholinomethyl group, I got excellent result. In the case of Candida parapsilosis and Aspergillus fumigatus, in which I got the MIC value more than the standard compound. Thank you.
Thank you, Dr. Rastogi, for sharing good information. Next.
Dr. Sonal Agrawal.
Yes, I'm here. Yeah. Am I audible and visible?
Yeah, yes, you're visible and you're audible also.
Okay, good afternoon, ma'am. Good afternoon, everyone. I'm Dr. Sonal Agrawal.
Do you want to share your slides?
No. Mine is an oral presentation. Yeah. So, the topic of my presentation is importance of maintaining a good environmental health for well-being. So, first of all, I want to say that the 21st century is witnessing such radical changes in every facet of human life on planet Earth that it has become difficult to distinguish between progress and hazardous. The rapid rise in industrialization, globalization, urbanization, and increase in all forms of pollution, be it air, water, soil, and even the threat of nuclear war knocking at the door in the current scenario, it has all affected health, not just the physical health, but also mental and psychological and spiritual health. The World Health Organization clearly lays forth that health is not just the absence of disease or illness or infirmity, but it is the proper physical, mental, and social well-being. See, it reiterates the importance of well-being in life. Our current scenario is flooded with pollution and environmental stresses which affect the physical health in the form of asthma, injuries, illnesses which are chronic and acute in nature, and also can precipitate a host of psychiatric problems, including anger, aggression, depression, irritation, frustration, annoyance, and so on. It is important to thus investigate the relationship between environmental health and psychological well-being in a scientific setting. So for this research, the aim of this research was to explore the relationship between environmental health and well-being among undergraduate students. The hypothesis, now on the basis of the available literature on this topic, on the two variables under study, the following hypothesis was formulated: that environmental health will be positively and significantly related to well-being among students. The variables, the two variables in the study were environmental health and psychological well-being. The sample of 29 students was selected. Purposive sampling was done for this purpose. The samples ranged in the age of 16 to 19 years. It comprised of both male and female respondents. And all the participants were well versed in English language because the questionnaires given to them were in that language. Next, the tools which were used in the study. There were two tools which were used in the study. First was the Environmental Health Assessment Survey, which consists of 17 items. And it covers various domains like pollution, crowding, traffic, and so on. And the second tool which was used was the Psychological Wellbeing Scale developed by Diener and Diener in 2009. This is a short summary item survey consisting of 7 items, which gives an aggregate score. Consists of 8 items, which gives a total aggregate score also. Ratings are done on a 7-point scale, and this scale has very high psychometric properties. Now, the method of data collection and analysis. For the sake of this study, the sample was contacted individually, and they were given the demographic sheets along with the 2 psychometric scales. Once the data was collected, the participants were thanked for their cooperation. The data thus received underwent statistical operations in the form of...
Dr. Agrawal, sorry to interrupt you, but 4 minutes have gone. And whatever result you want.
Can I just get 1 more minute? Yeah. Pearson product moment correlation was used. The result revealed that there was a moderate correlation of 0.43, which was significant at both 0.05 and 0.01 level, which shows that there was a positive relationship between the two variables under study, which shows that as environmental health increases, the well-being among students increased and vice versa. That's it. Thank you so much. Thank you.
Thank you.
Anshu Gautam, PP08. Anshu Gautam.
Anshu Gautam, are you there?
Call the next participant.
Dr. Ruby Yadav, OP10.
Yes, ma'am. A very good afternoon to everyone. I Dr. Ruby Yadav from Department of Zoology, University of Lucknow, and my topic is feeding behavior and dietary preferences of vultures. Now the introduction. Vultures are like nature's street sweepers and clear up messes that several of us would not dare to touch. And as carrion feeders, vultures play important role in our ecology. Sharp beak, strong talons, and neck are feeding helpers. They mostly consume soft muscles, tendons, eye, external ear, liver, heart, ribs, and mid-sized visceral organs. And they follow the hierarchical feeding system in order to arrive near the carcass. Firstly, feeding initiated by the king vulture, followed by migratory and residential vulture species. This is the image of the turkey vulture and a red-headed vulture, and they both locate the carcass on the basis of nostril and keen eyesight methodology, and research from the scientific and trade literature was cited and reviewed on the basis of feeding. And in this study, the feeding and foraging behavior of vultures was done using 2015 to 19, and by the direct observation of the group of vultures. The data was supported by 70D SLR Canon camera and binocular for photography and direct observation. And now the observation, total 13 carcasses were observed during the study periods. Out of 13, 7 were blue bull and 2 were sambar deer and 3 crows and 1 ox carcasses. And this is the vulture feeding helpers such as the pointed beak, strong talons, long neck, and lappets, keen eyesight. Now, the table of the vulture species along with the feeding helpers, and the first is the Indian king vulture, feed on the eye, heart, ear, and liver. Second, Eurasian griffon vulture, feed on the internal parts of the all mid parts. And third is Himalayan griffon vulture, mainly eat on the flesh and pine needle. And another is the Indian vulture and cylinder-billed vulture. It prefer the almost all carrion. And white-backed vulture feed on the pieces of old dry bones such as ribs and skull pieces. Another one is the Egyptian vulture. It prefers the excrement of the vertebrate, and human refuses eggs and other small invertebrate for the feeding purpose. And cinereous vulture mainly feed on the tendons, muscles, bones such as ribs or another flesh of the large animals. And last one is a bearded vulture. It prefers the bones and bone marrow for the feeding, and also on the small lizards and turtles. Now the conclusion, a nutritional and mineral deficiency problem has been observed in all the gypsy vulture species population because they exclusively feed upon the meat and soft tissue of large mammals, and therefore their diet contains very little calcium. Since the soft tissue contains very little calcium, most of the calcium is present in the bone tissue, and which has been collected by the contractor of dumping ground every week by the truckloads. And I recommended the establishment of the captive breeding centers and vulture restaurant, and establishment of the artificial feeding stations, and construction of the small drinking ponds, and provide the diclofenac-free carcass to the vultures near the breeding areas. Thank you.
Good. You completed within time. Good.
Thank you, ma'am.
Where you have studied all this? In Lucknow only?
Yeah,
No, ma'am.
I can see the photographs on your last slide. Is it from Lucknow? Next.
Samriti Dwivedi.
Good afternoon, ma'am.
Please start.
Yes, ma'am. Is my screen visible, ma'am?
Yes, it is visible.
Okay, good afternoon, everyone. I'm Samriddhi Dwivedi, working as a research scholar in the field of atmospheric chemistry, and I'm here to present my study on assessment of indoor and outdoor superfine ultrafine particulate matter in addition to gaseous pollutants in Lucknow city. The increasing developmental activities and rapid urbanization have resulted in a massive stress on environmental quality. Every year, millions of cases are encountered of COPD, asthma, and other respiratory disorders in the country. India's capital city, Delhi, was also declared as the most polluted city consecutively 3 times in a row in the year 2020. Lucknow, being the capital city of the most populous state of the country, comprises of a population of approximately 35 lakh people in the year 2021. While growing congestion in cities, a trend which is most likely to increase in future, we need to also look at transport sector and infrastructure investments which may contribute to the environmental factor. Although people are aware of ambient air pollution, yet indoor air quality is still neglected. The outdoor pollution concentration has direct impact on indoor air quality. It has been estimated that approximately 2 to 5 times more pollutants are found indoors compared to outdoors. Also, people on an average spend 90% of their time indoors, which has been further increased due to COVID-19 lockdown from the past 3 years, making assessment of indoor air quality highly recommendable. The objective of my study included assessment of particulate matter, including PM 2.5, PM 0.1, 0.25, and less than 0.25, along with some gaseous pollutants like sulfur dioxide, nitrogen dioxide, carbon dioxide. Apart from that, meteorological parameters like wind speed, wind direction was also evaluated during winter months in the city at different residential, industrial, and commercial areas. Now, what are superfine and ultrafine particles? Ultrafine particles are basically particulate matters having aerodynamic diameter less than 100 nm, whereas superfine particles have aerodynamic diameter between 100 to 300 nm. As displayed in the figure, we can easily conclude that although the particle mass of ultrafine particles is small, yet the particle number is far more than fine and coarse particulate matter, which makes them more easily absorbed and highly toxic in the human body. One of the anticipated medium of transference of such particulate matter has been displayed here. For instance, if a person sneezes, the nearby ambience is occupied by small droplet which act as a medium of transference of such particulate matter to a healthy body. Some of the principal pollutants recognized in indoor and outdoor environment include sulfur dioxide, sulfur oxides, nitrogen oxides, particulate matter, pHs, and heavy metals, which pose certain kinds of short-term effects like irritation in eyes, nausea, sneezing, to long-term effects such as asthma, COPD, decreased fertility in men and women, etc. Coming to the instrumentation of my assessment, we assessed ultrafine particles using Leland-Legacy sample pump with 5-stage Sauter's cascade impactor. Each stage used to collect finer particles than its predecessor. The instrument was set both at indoor as well as outdoor environment at a flow rate of 9 LPM for 24 hours. For PM 2.5, we used Envirotech APM 550 at a flow rate of 17.57 for 8 hours in both indoor as well as outdoor environment. The air in this system entered through omnidirectional inlet, providing a cut point for particles greater than the required diameter. The gaseous sampling was done for NO2 and SO2. The gaseous sampling was done using handy impinger, and the absorbing solution was made as per the CPCB norms. Other than that, CO2 and ventilation rate was measured using ES 206 monitor. Here is the result.
Try to finish fast.
Yes,
already you have taken more time.
Yes, ma'am. Here the result has been shown. Although the average concentration of PM2.5 is more in outdoors than indoors, yet the difference between the two is not very much, indicating that indoor air is also as polluted as outdoors. Whereas the concentration of ultrafine particles shows totally opposite result. Here the indoors possess more concentration of ultrafine particles than outdoors. pHs was also assessed, which were associated with the particulate matter. Here Pearson correlation was determined, where some pHs showed positive correlation, the others shows negative correlation. That means increase in the concentration of one pHs led to the increase in the other. Also, the 7 recognized carcinogenic pHs contributed more than 60% to the total contribution. Despite how much it needs to be accomplished, we have come to a long way in the past few years. We have started of getting science and technology right in the terms of monitoring of air quality. We are working to approach more budget-friendly and easily accessible remedial devices. Thank you.
Thank you, Samriddhi. Any question? Next participant.
Akriti San, OP12.
Yes, ma'am, just presenting. My slides are visible?
Yes, these are visible.
Very good evening to all. I am Akriti San, Department of Chemistry, Isabella Thoburn College, and going to present on molecular docking of epigenin with various reported biological activities. So, first of all, phytochemicals are the compounds produced from the plants with different pharmacological properties. And among phytochemicals, flavonoid top the charts, along with other polyphenols, anthocyanidins, limonoids, phytosterols, glucosinols. And these flavonoids are responsible for multiple functions in both plants and as well as in the humans. Basic flavonoid structures are the oxygenated heterocyclic with the skeleton structure of 15 carbons, and they are made up of 2 aromatic rings connected by the oxygen and carbon chains that form a close spiral ring. Therefore, therefore structures also referred as C6, C3, C6 structures. And now why I have chosen the epigenin, as the epigenin is a promising biological compound showing activity for anti-cancers, anti-tumor, hypotension, hypoglycemia, anti-osteoporosis, antioxidation. And it is mainly found in the plant including chamomile, wheat sprouts, parsley, onion, oranges, tea, and some seasonings. The basic structure of epigenin is a trihydroxyflavonol with molecular formula C15H10O5, and it is substituted with 3 hydroxy groups at position 4-5 and 7. And these hydroxy groups can easily be modified or substituted for designing synthetic analogs to increase its biological activities. And I tried to do molecular docking to see the interactions between the proteins of reported biological activities and the epigenin. So first, in the contrast, I selected anti-cancer, and for this I chosen CDK6 protein. And while docking, I got the best binding energy pose for -6.64. Same as for anti-diabetic, I chosen adiponectin receptor protein 2. And for this, I got best binding energy pose at -8.95 kilocalorie per mole. That is a good binding energy. And for antimicrobial, I have chosen hepcidin, and for that I got -6.28. At the end, I chosen plastine 3 for
antiosteoporotic activity, and for that I got binding energy at minus 6.69 kilocalorie per mole. And from these, all the docking results reflect that binding energies of the dock conformations for all the proteins that are CDK6, adiponectin receptor protein 2, hepcidin, and plastine 3 were of good values. And the best binding energy I got for the antidiabetic property, that is minus 8.89 against the protein adiponectin receptor protein 2. And the interactions were also visible for hydrogen interactions and some non-covalent interaction too. So that synthetic analogs can be developed in this direction, hoping for improvement in binding energy values and drug likeness properties. These are some of my references. Thank you.
Well in time, Akriti, I must appreciate. Next, any question?
Thank you, ma'am.
Dr. Seema Sarkar.
Good evening, and on this auspicious occasion of Mahashivratri, due greetings to the chairperson and all present over here on this digital platform. I belong to this college, the Department of English, Seema Sarkar, and purely, literature person, nothing to do with chemistry, but the sub-theme that is hygiene to save lives and combat COVID-19. That, created some bubbles of thoughts, and that is what my humble presentation before you entitled Revisiting Traditional Indian Practices in Times of COVID-19. So I can tell you that I have nothing to do with chemistry. It's just some of the expressions that I can correlate with the topic. So my father was a general physician, but I am a pure literature person. So I can tell you that revisiting how this idea cropped up in my mind, because this COVID-19 has transported me to the things that has been very much related to our lives. That is why I'm talking about revisiting traditional Indian practices in times of COVID-19. For the last 2 years, lifestyle has changed globally. And COVID-19 may be a crisis period, but has some flickers of positive effects on us. And in 2022, we are searching for a ray of hope in our lives, and we have exhausted the meager resources of Earth and find ourselves dwindling away. Something entirely different is happening. The first and foremost positive side effect of the pandemic is bringing back family togetherness in the lockdown period. Home-cooked food was preferred to tinned, canned, and junk foods. Sanitization has become a habit now. Washing hands frequently is a part of life now. These things are our cultural ethos, which we were losing in the post-modernist approach to life. And I can tell you that these things, took back to nostalgia to my childhood, to my grandparents' home, my nani ma, who was very much restricted to, we used to say this chua-chut kind of thing, but this is what, these are the cultures, what we have already there in our lives. In my childhood, I remember washing hands and feet from coming outside, keeping shoes outdoors, and changing clothes immediately as part of life. Outside food was available at functions only. So fresh cooked delicacies were enjoyed at home. And summer vacations were mostly spent at maternal grandparents' home with cousins, uncle, aunts, strengthening, family bonding. Big joint families had no scope of loneliness like the modern times. So nature has given us a message to stay in coordination with it. As the priest of nature, William Wordsworth has wonderfully commented on materialistic approach of life in his poem, The World is Too Much With Us. Little we see in nature that is ours. We have given our hearts away a sordid boon. Thank you, ma'am.
Thank you, Dr. Sarkar. It was really nice to have the positive picture of the pandemic. And this is much needed because it will bring all of us from sadness to happiness.
Ma'am, I have written a paper, full paper on, that effect of Bhagavad Gita on in this crisis time, pandemic, which is very soon means publishing. Okay. So that's what I felt in this time, that this is, maybe this is very dismal time, but I have
got very positive things. I have improved myself. So I don't feel that this is very bad time. Bad time has its positive, things into it. Thank you, ma'am.
Next.
Dr. Renu Sharma, OP01.
Actually, Dr. Renu Sharma was not able to present.
I'm audible?
In the beginning.
Hello.
Renu Sharma was not. Yes.
Yeah, audible.
Hello, I am audible?
Yes, you are
Yes, ma'am.
audible.
Good afternoon, everyone. I am Dr. Renu Sharma from Gokul Das Girls Hindu College, Muradabad. The word disaster has been originated from two French words. First is das, which means bad or unwanted, and second is astre, which means stars. Etymological, the word disaster comes from in the sense of bad star. India's geoclimatic conditions make it one of the most disaster-prone country. About 58% area of India is earthquake-prone, which occurs in a middle or high-risk zone. 12% of India's land is affected by flood and erosion. 4% of the land of India is vulnerable to disaster. In Uttar Pradesh, about 27 lakh hectares land are affected by annual flood, and the annual loss due to flood is approximately 432. Flood and cold wind cause about 386 human losses per year. A disaster is a sudden calamitous event that has the potential for physical, social, economical, and environmental damage. Disaster may be natural or man-made. Both types of disasters are serious disruptions occurring over a short or a long period of time, but a disaster is different from hazards. Hazards have the potential to harm, but a disaster is a serious disruption of the functioning of a community. For example, in 26 December 2004, tsunami is a hazard because in this case, there is no loss of life, but a disaster occurs when a hazard has an impact on vulnerable people. Disaster risk reduction is a systematic approach to identify, accessing, and reducing the risk of disaster. In 2005, the Disaster Management Act was passed by Rajya Sabha and the Lok Sabha. The main focus of this act is to respond and recover people from disaster events. Under DM 2005, lays down a multi-layered system such as National Disaster Management Authority, State Disaster Management Authority, District Disaster Management Authority. National Policy for Disaster Management 2009 to build a safe and disaster resilient India. Sendai Framework for DRR, which 7 targets and 4 priority, and Paris Agreement 2015 aims to strengthen the global response for climate change and to pursue efforts to limit the temperature further increase to 1.5 degrees centigrade up to 2050. National Disaster Management Plan 2019's main feature are understanding risk, interagency coordination, investing in structural and non-structural measures in DRR, capacity development, and climate risk management. Disasters are unpredictable and are extremely destructive to human life. There is a need to work quickly in the field of disaster management so that more people can be benefited in lesser time. A country should be prepared for disaster management. Remote sensing is an extremely important tool for disaster management. Remote sensing is the science of acquiring information about the earth using remote instruments such as satellites, which can be inherently useful for disaster management. Satellites offer accurate, frequent, and almost right data over large areas anywhere in the world. When a disaster strikes, remote sensing is often the only what is happening on the ground. Remote sensing data helps to make strategy actions to assist risk reduction initiatives. Remote sensing is widely used by a meteorologist for weather forecasting and warning to severe weather events are released, which can be very helpful for deciding within a short period. Remote sensing data is a valuable source of information which is presented in print, television, and the internet. Visual impacts makes a more lasting impact on the effect of disaster. Reducing the impact of any disaster is always the top priority, and the technology and result offered by remote sensing are unmatched in the field. Geoinformatics technology is very important in disaster management. This technology is based on remote sensing and geographical information system. Geoinformation technology can be used pre-disaster by early warning, monitoring, risk modeling, etc. With the disaster to public warning systems, search, rescue plannings, etc. and post-disaster to damage assessment, habitation. Thank you. Thank you, everyone.
Thank you, Dr. Sharma.
Sushmita Bajpayee, OP14.
Hello. Hello.
Yes, ma'am.
Good evening, ma'am.
Good evening.
मैं सुष्मिता बाजपेयी ब्रह्मानंद कॉलेज कानपुर केमिस्ट्री डिपार्टमेंट से हूँ. मेरा टॉपिक है इंस्ट्रूमेंट टू कंट्रोल वाटर पॉल्यूशन. ठीक है, मैं आपको थोड़ा सा आइडिया ये दूंगी. वाटर पॉल्यूशन तो सभी जानते हैं कि वाटर पॉल्यूशन, लेकिन इसमें हम क्या इम्प्रूवमेंट करते हैं, कैसे हम इसको कंट्रोल कर सकते हैं. मेरा मेन पेपर का टॉपिक है इंस्ट्रूमेंट टू कंट्रोल वाटर पॉल्यूशन. तो हाउ टू कंट्रोल वाटर पॉल्यूशन. Water pollution control is usually specified address in connection with the establishment of environmental legislation and action plan, but also within the framework of water resources management planning. Moreover, documents related to public health aspects may also consider water pollution. These three interacting areas are often administered in different line ministries, typically a Ministry of Environment, a Ministry of Water, and Ministry of Health. यदि ये तीनों मिनिस्ट्री आपस में मिलकर कोरिलेट होकर कोई भी काम करेंगे, तभी हम इससे वाटर पॉल्यूशन से निजात पा सकते हैं. In addition, the policy making process, if it exists, may often takes place independently. Water quality degradation caused by erosion and sedimentation, municipal and industrial discharges, and agricultural runoff threaten the fish and wildlife. अब नेक्स्ट वन इज व्हाट इज वाटर रिसोर्सेज मैनेजमेंट. किस तरह से हम वाटर मैनेजमेंट कर सकते हैं. वाटर रिसोर्स मैनेजमेंट अकॉर्डिंग टू द वर्ल्ड बैंक aims to maximize the benefits of water by ensuring that there is enough water or sufficient quality for drinking water and sanitation, food production, energy generation, inland water transportation, and water-based recreation, as well as sustainable, healthy water-dependent ecosystem, and protecting the aesthetic and spiritual values of lakes and rivers. Floods, drought, and pollution all are the example of water-related hazards that must be managed. The interconnection of water, families, ecosystems, and economics necessitates integrated management that takes into consideration the thread of the water many uses and values. What are the water policy? Commercial instruments for water policy are tools that are incentives and disincentives to alter conditions in order to facilitate economic transaction or minimize risk, with the goal of improving environmental quality. In the world of even increasing water demand and declining water supply and dependability, where water-related risks are on the rise and where climate change threatens the undermine decades of development gains, the only path to sustainability is a well-balanced policy mix. As a result, all line ministry involved in water resource management as well as the organization responsible for overall economic development should be consulted and consensus sought during the policy making process. Why are the water policies being promoted? During recent years, there have been increasing awareness of and concern about water pollution all over the world, and new approaches towards achieving sustainable exploitation of water resources have been developed internationally. It is widely agreed that a properly developed policy framework is a key element in the sound management of water resources. A variety of potential aspects for such policies have been recognized, particularly during the development of Agenda 21 and subsequent actions. There are some instances of policy aspects that promote the long-term management of water resources. Next is objective of water resources management. Water security is one of the objectives of water resource management. For fast increasing and urbanization global population, it is impossible to anticipate and design a single path for water security. कोई भी एक सिंगल रास्ता आपको पूरी वाटर सिक्योरिटी से नहीं दे सकता. तीनों मिनिस्ट्री को, हेल्थ को, एनवायरनमेंट को और हाइजीन, सभी को मिक्स होके अपना एक प्लान बना पाएगा, तभी हम इस वाटर पोल्यूशन के कंट्रोल को हम बैलेंस कर पाएंगे. फॉर फ्यूचर जनरेशन, for future planning and management of water resources is necessary to enhance the water security. However, one of the most common water resources management goals is to promote circumstances that allow for ecologically sustainable, economically efficient, and fairly distributed water resources uses. They also involve increasing an advantage of current hydraulic infrastructure while lowering the risk. The common goal of these initiatives to incorporate policy approaches into other sectoral policies in a broader region across the country. This frequently entails the creation of social, technological, and administrative instruments for water resource management. Economic instrument to control water.
Dr. Bajpayee, please try to.
मेरा कंक्लूजन में यही कहना है कि मैडम कि सभी को मिलकर इस वाटर पोल्यूशन को कंट्रोल करने के लिए सभी लीगल जो अपने एस्पेक्ट है उन सबको मिक्स होना चाहिए और इसके लिए प्रयास करना चाहिए. थैंक यू.
Thank you for your presentation. And sorry for interruption.
Mrs. Bimla Bin, OP15.
मैम स्क्रीन विजुअल हो रही है?
हो रही है.
Yes, ma'am.
Thank you, ma'am. A very good evening to Seema Joshi ma'am and are present here. Today, I will represent a review on the topic of environmental risk of nanoparticles used in cosmetic industry and its regulation. International Organization for स्टैंडर्डाइजेशन. और इन्होंने क्या डिफाइन किया कि जो उनकी ये साइज के आधार पर डिफाइन किया कि जो भी नैनो पार्टिकल हैं उनकी 1 से 100 नैनोमीटर की साइज के वो नैनो पार्टिकल के अंतर्गत आएंगे, जो कि आप जो है या नैनो स्केल में इनकी साइज जो है आपका एक एंगस्ट्रॉन से जो है 100 तक शो हो रहा है. फिर जो है हम कुछ नैनो पार्टिकल्स के स्ट्रक्चर देखेंगे, नैनो स्ट्रक्चर जो कि नैनो स्केल पर दिखाए गए हैं, जैसे आप देख पा रहे होंगे लाइपोसोम, डेंड्रीमर, गोल्ड नैनो शेल, क्वांटम एंड फ्लूइड. तो ये सारे जो नैनो पार्टिकल के स्ट्रक्चर हैं जो कि आपको नैनो स्केल में दिखाए जाएंगे.
अब यहाँ पर जो है आप देख पाएंगे कि ये जो कुछ एप्लीकेशन इनके हैं नैनो पार्टिकल्स के, जो आपको हर जगह देखने को मिलेंगे। जैसे कि मेरा टॉपिक है कॉस्मेटिक, तो कॉस्मेटिक के अलावा जो है आपको इलेक्ट्रॉनिक्स, बायो इंजीनियरिंग, डिफेंस, सिक्योरिटी और इसके साथ साथ इन्वायरमेंट में भी इनका बहुत देखने को मिलेगा। अब यहाँ पर हम देखेंगे कि कुछ नैनो कैरियर्स हैं जो कि नैनो पार्टिकल को कैरी करने में यूज होते हैं, क्योंकि इनकी साइज इतनी माइन्यूट होती है कि इनको जो है ये इंडिविजुअली खुद से ये जो है कहीं भी क्रॉस नहीं कर सकते, इसलिए जो है यहाँ पे कैरियर्स का यूज किया जाता है। ठीक है, यहाँ पे कुछ कैरियर्स दिखाए गए जैसे लाइपोसोम्स हो गया, ट्रांसफेरोसोम, तो यहाँ पे ढेर सारे कैरियर हैं जो इनको कैरी करते हैं टारगेट ऑर्गन तक, और वहां पर जाकर ये अपना इफेक्ट दिखाते हैं। अब यहाँ पे हमने देखा कि कॉस्मेटिक्स जो है जहाँ पे नैनो मटेरियल्स का यूज हो रहा है, तो हालांकि इसका जो है हमें बेनिफिट तो होता है। जैसे अभी यहाँ देखिए माइक्रोब इन्फेक्टेड स्किन में अगर हम जो है कोई एंटी माइक्रोबियल क्रीम लगाते हैं तो उससे हमारा स्किन अच्छा हो जाता है, बट इसका साइड इफेक्ट भी आपको देखने को मिलेगा। सनस्क्रीन हम यूज करते हैं तो उससे भी हमें अच्छा तो हो जाता है, प्रोटेक्शन तो मिलती है, यूवी प्रोटेक्शन, लेकिन इसका कहीं ना कहीं साइड इफेक्ट देखने को मिलता है। अब यहाँ पे कुछ नैनो मटेरियल्स ऐसे हैं और इनके टाइप्स हैं जो कि मोस्टली हम लोग दैनिक जीवन में यूज कर रहे हैं, जैसे जिंक ऑक्साइड हो गया, एल्यूमिनियम ऑक्साइड हो गया, आयरन ऑक्साइड, टाइटेनियम ऑक्साइड, ये सारे जो है नैनो मटेरियल्स आपके यूज हो रहे हैं। जैसे आप देखिए फाउंडेशन में, कंसीलर में, यूवी प्रोटेक्शन में, तो ये सारी चीजें जो है यूज हो रही हैं इनमें, और इसका हम पर्टिकुलरली हम लोग यूज कर रहे हैं, जो कि आगे जो है हार्मफुल इफेक्ट क्रिएट कर सकता है। और ये जो कुछ नैनो पार्टिकल्स ऐसे हैं जो कि आपका एनवायरनमेंट में भी एक्सपोज हो रहे हैं। ये ग्राफ के थ्रू आप देख पा रहे होंगे कैसे प्रोडक्शन के समय, ट्रांसपोर्टेशन के समय और कंजप्शन के समय या फिर डिस्पोजल के समय जो है ये हमारे एनवायरनमेंट में एक्सपोज हो रहे हैं और ह्यूमन के हेल्थ को प्रभावित कर रहे हैं। फिर जो है ग्लोबली हार्मोनाइज सिस्टम दिए गए हैं, जो कि इन्होंने क्या दिया कि एक्वाटिक टॉक्सिसिटी को डिफाइन किया, कि जो है इनको 5 क्लास में इन्होंने डिफाइन किया, और ये जो है बताया गया कि किस तरीके से एक्सट्रीमली टॉक्सिक और वेरी टॉक्सिक, टॉक्सिक, हार्मफुल, नॉन टॉक्सिक। तो ये जो है नैनो पार्टिकल्स के कॉन्सेंट्रेशन पर लिया गया एक्वाटिक मीडियम में, कि किस तरीके से ये जो है कम इनका जो है बहुत लेस अमाउंट ज्यादा टॉक्सिक है, और जो है और ज्यादा उनके और इनके ज्यादा कॉन्सेंट्रेशन जो है आपका कम टॉक्सिक है। ठीक है, तो इन द केस ऑफ नैनो पार्टिकल अब यहाँ पे ये भी देखेंगे हम लोग कि किस तरीके से हमारी बॉडी में एंटर करते हैं। ये रेस्पिरेशन के थ्रू, डाइजेशन के थ्रू और हमारी स्किन के थ्रू हमारी बॉडी में एंटर करके और हमारी बॉडी को अफेक्ट करते हैं। अब यहाँ पे देखेंगे कुछ जो है रूट होते हैं, डायरेक्टली रूट और इनडायरेक्टली रूट के थ्रू ये हमारी बॉडी में एक्सपोज होते हैं, और इनडायरेक्टली में जैसे आप देखेंगे प्लांट और एनिमल के थ्रू ये हमको अफेक्ट कर रहे हैं, और डायरेक्टली में आप देखेंगे जिससे कि एयर का एक्सपोजर हो रहा है, सॉइल का, वाटर का, तो ये सब जो है इस तरीके के नैनो पार्टिकल हमारे बॉडी में एंटर कर जाते हैं। अब यहाँ पर ये देखा गया है कि जो नैनो पार्टिकल होते हैं मोस्टली वो रेस्पिरेटरी ट्रैक्ट में एंटर कर रहे हैं, जिनकी साइज मेनली जो है बताया गया है कि 10 से 20 नैनोमीटर की हो सकती है। अब यहाँ पे 2 पाथवे दिए गए हैं कि किस तरीके से हमारी बॉडी में वो रेस्पिरेटरी ट्रैक्ट में एंटर करते हैं। तो ये जो है एंडोसाइटोसिस फर्स्ट है और सेकेंड है डायरेक्ट पेनिट्रेशन। एंडोसाइटोसिस में देखेंगे तो यहाँ पे आपको अबाउट 5 पाथवे दिए गए हैं, जो कि फैगोसाइटोसिस, माइक्रोपिनोसाइटोसिस और अदर भी प्रोसेस हैं, जो कि ये जो है नैनो पार्टिकल हमारे बॉडी में एंटर करते हैं और अक्रॉस हम सेल मेम्ब्रेन। डायरेक्ट पेनिट्रेशन में आप देखेंगे तो यहाँ पर आप देख रहे हैं डिफ्यूजन, परमिएशन, पोर फॉर्मेशन, इनके थ्रू हमारी बॉडी में वो एंटर कर रहे हैं थ्रू द सेल मेम्ब्रेन। फिर यहाँ पे देखेंगे कि अभी मैंने बताया
मिस बेन प्लीज ट्राई टू।
जी मैम, बस हो गया। तो ये नैनो पार्टिकल की साइज छोटी होने की वजह से क्या हो रहा है कि कैसे हमारी बॉडी में इंटर कर रहे हैं और हमारे नर्वस सिस्टम पर अफेक्ट कर रहे हैं। ये कार्सिनोजेनिक भी होते हैं, और अभी कुछ एग्जांपल हमने बताया संस्कृत में टाइटेनियम डाइऑक्साइड, जो बहुत ही ज्यादा पहले इनर्ट था, लेकिन अब माना गया कि नहीं, अब ये एडवर्स इफेक्ट कॉज कर रहा है। और इसके साथ साथ ये रॉस का फॉर्मेशन करता है, जेनोटॉक्सिक भी है और कार्सिनोजेनिक भी ये होता है। और भी आप देखेंगे कुछ रिप्रोडक्टिव डिसऑर्डर भी क्रिएट कर रहा है, डिजेनरेटिव डिजीज भी क्रिएट कर रहा है। तो इस तरीके से हमारी बॉडी को हर तरीके से अफेक्ट कर रहा है। अब यहाँ पे भी कुछ डिग्री दी है टॉक्सिसिटी की, जो कि जब आप रेफरेंसेस कुछ पेपर देखेंगे रिव्यू, तो आपको वहाँ बहुत अच्छे से डिटेल में पता चल जाएगा। अब कुछ प्रॉब्लम ऐसी हैं जो कि लीगल हैं और एनवायरमेंटल रेगुलेशन के थ्रू हम देख सकते हैं। यहाँ पर ये देखेंगे कि जो लो कॉन्सेंट्रेशन है वो ज्यादा इफेक्टिव है, ज्यादा टॉक्सिक है, और जो लार्ज साइज का है वो कम टॉक्सिक है, क्योंकि जो है ये जो लो कॉन्सेंट्रेशन है वो बहुत इजीली हमारे बॉडी में एब्जॉर्ब हो जाता है, हमारे स्किन के थ्रू एब्जॉर्ब हो जाता है, और बट इनके लिए जो है कैरियर की जरूरत पड़ रही है। तो और इसके साथ साथ जो है ये जो है डिफिकल्टी क्या हो रही है कि नैनो पार्टिकल के डिटेक्शन में डिफिकल्टी हो रही है, और इस तरीके के टेक्नोलॉजी डेवलप करने में डिफिकल्टी हो रही है, जिससे कि हम इसका जो है प्रॉपर क्योर नहीं कर पा रहे हैं, तो और ना ही इससे हम उबर पा रहे हैं। तो ये जो है इनका जो है डिफिकल्टी है जो कि हमें फेस करना पड़ रहा है। चलिए मैम, थैंक यू सो मच मैम।
गुड शेयरिंग। नेक्स्ट। Next participant is Sangeeta Shukla, OP16. Sangeeta Shukla.
Dr. Sangeeta Shukla, OP16.
Hello, good afternoon.
Dr. Shukla, please mute your one of your plans.
Hello.
You are using, I suppose,
two. To all the dignitaries present in this digital platform. Am I audible?
Yes, now you are.
Yes,
audible.
ma'am. Am I audible?
Yes, you are audible.
Yes, ma'am, you are audible.
I'm Sangeeta Shukla, serial number 0P16. I'm talking about hygiene and sanitation as depicted in Arthashastra of Kautilya. The ancient Indian society was very much conscious regarding health and sanitation from the very beginning of civilization. Plant, township, drainage system, and creation of roads and grid system made the Indian civilization a unique one among the oldest civilizations of the world. The people of the Neolithic culture lived in the circular oval huts made of wattle and daub, inside which there were the cooking hearths. Sometimes circular pits surrounded by low walls was also found, which may have been used for the storage of the grains. The disposal of the dead is one of the major concerns to prevent a health hazard. Example of two sites are discussed here. These are Abhaipur in Pilipit district of Uttar Pradesh and Bhagwanpura in Kurukshetra district, Haryana. At Abhaipur, two graves were encountered, one each of an adult and a child. Both burials was extended. The adult, lying on its back, was oriented along northeast, southwest. Here I highlight the direction northeast and southwest, which is this practice is continued till date. With the face turned to the right, he measured 1.70 m in length and has been estimated to have been 45 to 50 years old. Some parts of a pot were found a little away from the waist. On piecing together, these formed a large-sized bowl of painted graveware. No other grave goods were found. The child's skeleton, 71 centimeter in length, was poorly preserved. It lay in a north-south direction with the head up to the north. The estimated age was between 2 and 2.5 years. In this case, too, no grave goods were found. It may also be added that these burials lay in the habitation area. This practice is also seen in Mesopotamian civilization, but there were grave goods with the body. ये adult burial हड़प्पा का है. Sanitation was taken good care of inside the houses. The description of the two devices is relevant here. In one, large size jars were placed one above the other. The bottoms were broken so that the silage could easily pass down. In the other, deep vertical pits were lined with terracotta rings and the silage was passed down through these ring wells. In one example, as many as 11 rings were observed made of terracotta. The rings measured about 60 centimeter in diameter, 15 centimeter in height and 5 centimeter in thickness. There were street public drains too, made of campfire bricks. These were fairly large with the bottom paved. This is the picture of ring well. These are the four. In this well, we can see 47 rings as counted by Dr. B.B. Lal. Though the river was close by and thus water could be fetched from it, some of the houses had their own wells. These were also lined with terracotta rings so that the sides may not collapse. In one case, as many as 47 rings were counted without reaching the bottom. The site of Shishupalgarh near Bhubaneswar in Odisha was never reoccupied after it was abandoned around 3 to 4th century. In 1948, when Dr. Brij Lal excavated Shishupalgarh, there was very little encroachment and he was able to get a good idea of early historic settlement. It is surrounded by a fortification wall which forms a square on plan. Each arm measuring a little over 1 kilometer, a moat fed with water from a nearby rivulet girdles the fortification. There are 2 major gateways through each arm, making a total of 8. Joining the opposite pairs of gateways, there are streets which run along with the cardinal directions and form a grid pattern on a plan. In one area, Dr. B.B. Lal exposed residential buildings made of laterite, flanking a stone-paved east-west street. Incidentally, the street had rut marks created by bullock cart passing over it. Most of the major early historic sites in the Ganga basin were fortified, but the plans varied in most cases. It seems that once the people started understanding the benefit of living in the floodplains of river and of course the constraints of the flood too, for which they devised the technique of erecting high artificial platform of mud and mud bricks to establish permanent settlements enclosed by a fortification wall and its components. It is pleasantly surprising to note the people honor the authoritative roads and divert public drains in favor of their own houses. For example, at Lothal, the city was extended beyond the southern walls in phase 3 when it was 2 kilometers in the circuit, but the plan of the city remained unchanged in phase 3 and 4. Similarly, at Mohenjo-daro and Kalibanga, evidence of road encroachment is not available. These are terracotta cards. Illustration of Rigved verified that the Vedic society was neither nomadic nor even in a mere rural stage, as has been assumed by many. It had long passed those stages and was dealing with kings and kingdoms. It had organized administrative machinery, had fortified towns, along with high health and sanitation norms. This practice of sanitation specific lifestyle to remain healthy and enjoy a prolonged life goes on to the later periods, which can be correlated with Jatakas, Puranas, and the early medieval works of literature. In this paper, I aim to highlight the progressive state of health and sanitation of the period depicted in Kautilya's Arthashastra, and I have used Kautilya's Arthashastra English translation by Dr. Sham Shastri and Sanskrit translation by Vachaspati Gorala and another edition by Ganpati Shastri ji. I am taking one part of this city system. Construction.
Sorry to interrupt, Dr. Shukla. Please, already you have exceeded the time.
One point I will discuss to you in such a long paper. Construction for force on all the 4 quarters of the boundaries of the kingdom, defensive fortification against an enemy involved shall be constructed on ground best fitted for the purpose. I will cite an example of this building within the fort. Demarcation of the ground inside the fort shall be made first by opening 3 royal roads from west to east and 3 from south to north. This plan is of Shishupal city, and you will see it is resembling with the illustration of Kautilya's. Demarcation of the ground is the fort shall contain 12 gates, provide with both a land and waterway kept secret. Here you can see 6 principal gate, 1, 2, 3, 4, 6 इस तरह से, and ये देखिए this is a subsidiary gateway, and 2 gateways are hidden that will be in water and land inside. The building shall be constructed on a strong ground in the midst of the houses of the people of all the 4 castes, and to the north from the center of the ground inside the fort, the king's palace facing either the north or the east shell. This is the king's palace complex. Now, what will be in this direction? This is illustrated in here. This is east north. This is eastern. And this is south. You can read this. And this is the south direction. South direction is illustrated pink and gray, and the west direction is illustrated orange and light pastel. This is north side. To the north by east, the treasury and the stables of cows and horses. To the north, at this section. To the north, the royal tutelary deity of the city, ironsmith, artisan, working precious stone, as well as Brahman shells. So all the paths are demarcated. These are the area to live there. Either to the north of the east, burial or cremation ground shall be situated, but that of the people of the highest caste shall be to the south of the city. Violation of this rule shall be punished with the first immersement. Families of workmen may in any other way be provided with site befitting with their occupation and field work. And this self-reliance system, all grain, sugar, salt, medicine articles, dry, fresh, metal, skin, charcoal, tents, weapons and stones shall also be stored in such quantities as can be enjoyed for years together without feeling any want. Of such collection, old things shall be replaced by new ones when received. Never shall Bahirakas, outsiders, who are dangerous to the well-being of cities and countries, be kept in force. They may either be thrown in country part or compelled to pay tax. Sovereignty is possible only with assistance. A single will can never move. The king, who is well educated and disciplined in sciences, devoted to a good government of his subject, and bent on doing good to all people, will enjoy the earth unopposed. Righteous and unrighteous acts are learned from the triple Vedas, wealth and non-wealth from Varta, the expedient and inexpedient, as well as potency and impotency from the science of government. When seen in the light of these sciences, the science of Anvikshiki is most beneficial to the world, keep the mind steady and firm in will and go alike, and bestow excellence of foresight, speech and action. I think you people of science can't understand Anvikshiki, so I add one more sentence. The academicians must participate in the discussion frankly on the issues of public interest and have good care of the nation's welfare. The practice of disobedience of laws for the fulfillment of the greed of persons shall be disregarded by the society itself. My motion is Akt Dharma Akt Falaya Rajyaya Namah. Thank you.
Thank you, Dr. Shukla.
I thought you should must have some idea of these things, so I shared this topic on to you.
Okay, thank you.
Dr. Pramila Pandey.
Yes, ma'am.
7.
Yes, ma'am. I'm presenting my slide.
Please try to be within time.
Yes, ma'am. Yes, ma'am. Yeah. Ma'am, is it visible?
Not yet. Yes, it is.
Yes.
Yes, good evening, ma'am. And I'm very thankful and grateful to the chairperson of this session, Dr. Seema ma'am. And ma'am, my topic, I'm Dr. Pramila Pandey from BSND PG College, Department of Botany, and I have chosen the topic Tulsi as an elixir. Ma'am, why it is an elixir? First, I will define by the topic. Why it is an elixir? Because whole parts of the Tulsi is used in medicine like leaf, stem, flowers, seeds. Even we have seen in this COVID, most of the families are having in the form of kadha for boosting their immunity. Because of this, I have chosen this topic. Tulsi, a member of Lamiaceae family, is a herb well known for its spiritual, religious and medicinal importance in India. The common name of Tulsi is, this plant is C. Its leaf, it has essential and volatile bioactive oils like phenols, terpenes, aldehyde. Here I have varieties of tulsis are found, and in my college I am developing one Aushadhi Vatika. For these I have collected 6 varieties of tulsi. In this slide you can see. Here you can see the morphological difference in all 6 varieties. Like here I have only 5 varieties: Rama, Shyama, and Karpuri, Ban Tulsi. Here you can see these are the parts which are used in medicine, like seeds we are using medicine, flowers, leaf, stem and root also we are using. So these are the substances, phytochemical substances which are extracted from the holy basil plants. Like from whole plants we can get mineral, from leaves we can get alcoholic extracts, from seeds we can get a lots of fixed oil. These here in this slide I am showing you the uses of phytochemicals, like eugenol in this plant. It is antiseptic and flavoring, antioxidant. Thymol, it is strong and antimicrobial. Beta caryophyllene, it is used in relieving of anxiety and depression. Rosmarinic acid, antioxidant, and carcirol, it is protective effect from the liver. Here in this slide you can see I have for like leaf in cough and cold, leaf and tulsi seeds we are using good antioxidant, anti-ulcer and antidiarrheal properties, and as a condiments in foods. See Taj Mahal. It is very interesting to so now relaxed tulsi is about to planting, and it is why, because it has a very oxygen, like all plants evolve oxygen, but tulsi evolves a lot of oxygen in comparison to other plants. So because of this tulsi is planted Taj Mahal, and its medicines will protect the marble mausoleum from this pollution, and it is one of the best plants to purify the environment in cleanness, high oxygen, which minimize the adverse effect of industrial and refineries.
Thank you, ma'am, for having a patience.
It was
Sakshi Gupta, OP18.
Yes, ma'am. Am I audible?
Yes, you are audible.
Wait, ma'am, sharing it.
Meanwhile, Sakshi is.
Is my screen visible?
Yes, yes. Yes, it is.
Okay. Shall I start?
Yes.
Good afternoon, respected teachers. Myself, Sakshi Gupta. I'm a research scholar from Department of Chemistry in IT College. The topic of my presentation is role of metal in enhancing the biological activity of the compound. So here I'm going to discuss the compound which shows biological activity, but the introduction of metal in these compound enhances its biological activity. So moving on to the first compound, which is the chalcones. This is the general structure of chalcones. The chalcone ketones that form central core or ligand with different types of metal ion complexes. This is formed by the aldol condensation reaction between benzaldehyde and acetophenone. These chalcone exhibit in two forms, Z and E forms, where E form is thermodynamically favorable form. And these chalcones have excellent pharmacological properties. And compound has discussed is carbazone, which is the derivative of Schiff basis. Carbazone acts as a ligand by doing its electron pair from the sulfur and nitrogen, while in semicarbazone, the oxygen and nitrogen. Now I will discuss this. Metals have to be very beneficial drug development and chemistry. These form the coordination complexes, and these metal ligand complexes have been used to treat several human diseases like infectious diseases, diabetes, etc. The d block elements have partially filled d orbitals. These form the coordination complexes with the electron-rich ligands, and the cisplatin and ferrocene exhibit anti-tumor and anti-cancer activities. And the metals involved in this are platinum and iron. So these are the d block elements which are useful in antimicrobial activities. Now the lanthanide, the F block element, is also very useful in antimicrobial activities, like the examples, and these are active against these bacteria. And these lanthanide complexes exhibit luminescence and fluorescence properties, which the use of these properties I will discuss it later. The actinide complexes are also very useful in nuclear power and nuclear weapon production. And as these have high positive charges, so these metals can easily achieve high coordination number. Now the mechanism of these metal complexes is according to the Pearson by HSAB principle, which says that hard acid reacts with hard bases to form a stable complexes, and soft acid react with soft base to form a stable metal complexes. So these metal complexes works on this principle. Now here I have shown, this is the chalcone compound. On the introduction of iron metal, its activity increases and it results into its antimicrobial activity. Whereas here ruthenium metal is used, which acts as a soft acid, and here oxygen acts as soft base. So these complexes form, which results in antifungal activity. Same wise, the thiosemicarbazone with nickel, cobalt, and copper, I have discussed it here. And this is the thiosemicarbazone, and metal I have nickel and copper. So these are the biological activities complexes I have mentioned, and these are the biological activity of the chalcone metal complexes I have mentioned. So the conclusion of the presentation is that these metal complexes and comparative evaluation of biological activity can be beneficial in terms of producing novel and less toxic and more effective anti-cancer and anti. Any questions to answer?
Thank you, Sakshi.
Okay.
The good completed.
Thank you, ma'am.
Dr. Two Preeti Singh, PP07 and PP08, Anshu Gautam had not reported. Please confirm. Preeti Singh.
Anshu Gautam ji.
And then Dr. Raj was not audible. It was the OP1.
There was some problem on his part. Actually, we checked.
If Dr. Rajiv Yadav is here, he can repeat, he can give his if he wants. If he is
Dr. Rajiv Yadav, I think he has left.
I think he has left. So I conclude.
But please.
All the participants for their presentation. Some of the presentations were good and very informative, and it was quite difficult for me to give marks or judgment. Suggestions were all equal and they were good. So this is to spread awareness, and this has been it has served the purpose well, and I congratulate Dr. Neha Agarwal and her team. Innovative also, but because of the paucity of the time, I could not give much time to them. So I apologize for that. And with this, I again convey my congratulations to the team and thanks to the organizing committee and the principal for inviting me.
Thank you so much, ma'am.
Thank you, ma'am. Thank you, Mrs. Joshi.
Thank you, Dr. Pandey.
Last, I'd like to convey my sincere
Thank you, ma'am.
Thank you, Seema, ma'am.
Respected
Thank you, ma'am.
Dr. Seema Joshi as the chairperson of presentation session. And I would like to thank all the participants, all the delegates who are present here. And that made a and I think I won't be taking much of time.
Thanks, Seema.
Thank you, ma'am.
Bye.